Good morning, everyone, and welcome to the 8th Single Cell Proteomics Conference. It's a great delight to see you here and open the meeting for this year. As we all know, what brings us together is our interest in understanding biology, not just at the level of the genome and the transcriptome, but also embracing the complexity at the level of the proteome with the myriads of proteoforms, modifications, dynamics in time and space. And of course, paying very close attention to all of the other relevant layers of biology, including the regulation of transcription, RNA metabolism modifications, and all of these other processes that ultimately impinge on and shape the proteome. In this meeting, we are focused on solving biological questions, and we are agnostic to the methodologies that we use. Of course, I'm very enthusiastic about the capabilities of mass spectrometry proteomics, but I'm equally appreciative and very enthusiastic of the awesome capabilities of imaging that until this day has allowed us the highest time resolution of being able to see protein dynamics. And as Galit Lahav is going to tell us, that's really, really important to understanding much of what makes us alive. And as customary, we are going to have presentations in this meeting from colleagues working in the realm of single molecule proteomics, emerging technologies. We are going to have presentations from mass spectrometry proteomics, and of course, from methods using affinity reagents, fluorescent proteins, and so on. And ultimately, I think that there is a lot that we can learn from those different domains, from these different technologies. As we made the argument in this perspective a couple of years ago, together with Mike McCoss, many of the challenges that those different technologies face and try to resolve are similar, such as the immense scale of the proteome. And approaches that have been helpful in one domain might also help in another domain. So in this particular case, we suggested that using separation that has long been the mainstay of mass spectrometry analysis can also be very beneficial for single molecule proteomics. And I think that there are many such examples. So one thing that I'm very proud of this meeting is that it brings together people who rarely attend the same meeting. And I think that's a catalytic capability there for us to all try and understand the developments in another area. We started in 2018 on this campus, and I think it's worth taking just a minute to recognize how much the landscape has changed. What precipitated the first meeting was our excitement around the capabilities to start making more specific, more scalable measurements with mass spectrometry. And at that point, there were some enthusiasts like us and Ryan Kelly who thought that this was indeed feasible, and we were excited to start developing it. And there was a lot of skepticism that maybe all that we are measuring is noise and proteins are still beyond the sensitivity of mass spectrometry. And by now, all that skepticism has dissipated. So in 2018, part of what we were trying to do is to demonstrate the quality of our measurements in vitro grown cell line cells. And today we are going to have presentations from colleagues analyzing many thousands of primary cells from complex mammalian tissues and measuring not just protein abundances, but rates of synthesis and degradation modifications, and not only measuring things, but really interpreting modes of regulation. And I think that's amazing progress for somebody who has witnessed that, who has seen this meeting changing from 2018 to now. It's remarkable. It has certainly taken a lot of effort from our community, from all of you and many, many people beyond this room. But for me as a participant of this movement, it's really amazing and encouraging what we have been able to do and how much more we have ahead of us, because the rate of progress, if anything, is accelerating. So as we took picture in 2018, we're going to take another picture today, and that is going to be just before lunch. As I already mentioned, we have colleagues joining us with very different backgrounds. Some people are very focused on biomedical research, biology, medicine. Other people are the foremost experts in their technology realm. Other people are computational biologists or mathematicians. And our very lofty aspiration, and I'll confess, it's very, very hard, but our aspiration is to make the presentations as accessible as we can possibly make them to everyone. Try to explain things conceptually, and ultimately, we are going to need assistance from the audience. You need to ask questions. That will be relatively easy because you'll see that everybody does that. That's usually the ethos of this meeting. We like to keep it very interactive, very engaging. And this is just for those of you who are first-time comers. This is just the first time that you're going to hear it, and soon you're not going to need to hear it because it will be obvious. But the goal is really to make everything accessible and to discuss. It is not so much to impress with all of the amazing results that we have. I know that there will be a lot of amazing results, but what I think is very special about being here together is that we can have discussions. We can read our papers. Many people can watch us online, which is wonderful. We have a large audience to which we are streaming the meeting, and I'm very happy to make it accessible. But what I think is very special for us being here is that we can interact with each other and ask very spontaneous questions. And that's why we have plenty of time after presentations, during breaks, in between breaks, to talk, and lots of good food. I'll get to that in a minute. So the goal is to be able to create a community that is very interdisciplinary and that as a whole possesses all of the skills and expertise that we need to advance this field forward. None of us does, but together we can help each other to make progress in what is admittedly very challenging, very daunting area of research. There's just no getting around that proteins and their interactions and modifications are very, very complex, and we still understand very little of that, which is exciting. I already emphasized how much we love questions. For those of you who are joining us virtually, I would ask you, if you're not asking questions or presenting, to turn off your video and audio so that there are no interruptions. We're going to have many excellent meals throughout the conference, including dinner for all attendees tomorrow evening. That's at 5 Horses, a short distance walk from campus. We can all go together. And that will be a very nice informal location where we can mingle and interact with each other. It's going to be a buffet-style dinner, and I think we're going to have a lot of fun with it. We also will have a number of posters presented today and tomorrow, and we are going to have poster prizes. I'm not going to tell you who are the judges, so that you try to impress everyone, not only the judges, but I'll tell you that the prizes are cool. All right. And of course, it's very, very important to acknowledge the support of our sponsors who have enabled all of the lovely meals that we're going to have throughout this conference. Thank you for supporting us. And I'm looking forward to a very exciting meeting with lots of biology and lots of technology and lots of interactions between those different domains. So I'd like to invite Keller to introduce our first speaker in our morning session, Professor Galit Lahav, from not far away, just across the road from Harvard Medical School. Keller, please.
Awesome. Yeah, can you hear me? Awesome. Okay. Hi, I'm Keller. I'm a research associate at Parallel Square Technology Institute on the immunology team. And I have the honor of introducing our first speaker today. Dr. Galit Lahav received her PhD in 2001 from the Technion, Israel Institute of Technology. In 2003, she completed her postdoctoral fellowship at the Weizmann Institute of Science in Israel. She then spent a year at Harvard's Bauer Center for Genomics Research, and in 2004 joined the Department of Systems Biology. In 2018, Dr. Lahav became the chair of the department. Dr. Lahav pioneered computational and quantitative experimental approaches to studying the fate and behavior of human cells at the single-cell level. Her work has yielded critical insights into the function and behavior of tumor-suppressing mechanisms and their role in cellular destiny. She has been recognized for excellence in both teaching and research through several awards, including the Smith Family Award, Vilcek Prize for Creative Promise, and Excellence in Teaching and Mentoring Awards. Lahav is now leading a department that works across traditional disciplinary boundaries to create new formulas, conceptual and mathematical models that enable moving from observing biology to predicting and engineering it. Please welcome me in welcoming, please join me in welcoming Dr. Lahav to the stage.
Thank you, Keller, and thank you for the invitation. It's a pleasure to be opening this very exciting meeting, and a meeting that is so easy to get to from, as you said, across the road. I'm very excited to share with you a recent story from the lab, and a story that I think the next steps of this story will really benefit from what we're all going to hear here in this meeting. But I will start by providing a little bit of background on the main goals of the lab and some of the critical information and the things that we learned in the last 2 decades. And do you have a clicker? There you go. Thank you. All right, so I think we will all agree that this clicker doesn't work. It's okay.
Oh, it's because I don't know why it's not working.
Is there another way to advance the slide? I will start just talking while we figure this out. So what I was about to say is that we all agree that in the last 2 decades, it became clear that cellular information, and I mean both information within the cell, but also information outside the cell, can be transferred through what we call protein dynamics. And by the word dynamics, I mean anything that we can measure over time. It can be concentration of protein, localization, modification, but it's really the idea about the shape of the curve, how these levels change when you look at them over time. And when you start to look at these measurements, you realize that it's actually very rich, and you can look at parameters such as frequency, if there's oscillations, amplitude, area under the curve, duration, and the computer is restarting. We are quite obsessed in the lab in trying to understand how information is encoded in these parameters and how these parameters are decoded by cells to trigger the right output in response to different external and internal inputs. And we've been using this umbrella to study a lot of processes in biology, cell cycle regulation, cell size, DNA damage and repair. What I will show you today is one of the main pathway that we've been studying, which is the p53 signaling pathway. So what do you need to know about p53? The p53 biology is huge, but let's try to summarize it and make it very simple. p53 is a tumor suppressor. It is the gene that is mostly mutated in cancer, and it responds to stress from the environment. And so when cells encounter cellular stress, or in our case, we mainly focus on DNA damage, p53 levels go up, and then it activates many target genes. It's a transcription factor. And what's quite interesting about p53, it has like these bipolar personality or dual personality. On one hand, it can activate programs that will allow the cell to cope with the stress, to cope with the damage, and resume growth. So it triggers more like survival. On the other hand, it activates genes that participate in terminal fates such as senescence and apoptosis. Okay, and the notion is that if the damage is too big, it's too difficult to cope with, it's better for this cell just to die or to arrest forever, and in order not to continue to grow, because when p53 is mutated, these damaged cells grow and can develop into tumors. Okay. So a lot of effort has gone in the entire p53 community to understand how this one single transcription factor chooses between these different outcomes, right? Sometimes it activates survival and repair, sometimes arrest, sometimes apoptosis, sometimes senescence, and so on. And a lot of beautiful studies suggesting it's cofactors that bind p53 or the level of p53. And when we entered the field, we were wondering whether dynamics matter, right? Whether different inputs trigger different dynamics of p53 and different dynamics of p53 leads to different outcomes. There we are. Okay, so here's our p53 responding to stress and activating different downstream targets. And the question that we had was...
Right now. Nope. Sorry, like...
The question we had is, do cells use modulation of p53 dynamics to achieve specificity in this network so that one dynamics will trigger one outcome and another dynamic another outcome? And so what you need to know about when studying p53 dynamics is that the regulation on the protein level is through the stability of the protein, okay? So there's no, in response to DNA damage, it's not changes in the mRNA of p53. It is, it is changes in the protein stability. So p53 activate the transcription of a target gene called MDM2. MDM2 is the E3 ubiquitin ligase. It binds to p53, ubiquitinate it, and send it to degradation, right? So we get this very classical negative feedback loop with two different arms. There is a transcription arm, which is a slow rate response, and protein-protein arm, which is a fast rate response. And even with this simple negative feedback loop, depending on the parameters, you can get different behaviors for p53, you can get oscillations, you can get just single pulse, you can get accumulations, and so on. Okay, so it gives you an array of dynamics that may be used for p53 function. And one of the things that we discovered many years ago is that in response to gamma radiation that leads to double strand breaks in the DNA, p53 show these beautiful, this series of beautiful pulses in cells. So if we look at the individual cell level, we see oscillations in p53 of a period of about 5 hours, 5.5 hours. When you average all them in the population, it seems like they're damped because with time cells lose synchrony. But within each individual cells, we get these pulses of p53. And we also saw that we don't always get oscillations. It depends on the input, right? So gamma radiation leads to these oscillations, but if we give different kind of damage through UV, we get one single pulse. If we give a drug like cisplatin, we get accumulation and so on. So we tested different ways to activate p53. We got different dynamical behaviors, hinting that maybe dynamics is used here in order to transfer information. And this kept us busy for many years, understanding the molecular mechanisms of all these different dynamics. But the real question that we have is, do cells care, right? Do cells care if p53 oscillate or doesn't oscillate? And in a series of studies, we looked at perturbation of p53 dynamics and look at the impact on target genes of p53. And here is one example. If we just irradiate cells and we get these oscillations, this allows recovery and growth. But if we use a small molecule that inhibit MDM2 and sustain p53 levels, then it pushes cells towards senescence, right? So that was the first example we showed that just by tweaking protein dynamics, just that, very accurately, right, we can activate different target genes genes, and we can push cells toward different fates. And a couple years ago, we also took it in vivo and measured p53 dynamics in different tissues and found at this point correlation that the more sensitive tissues showed sustained p53, the more resistant tissues showed oscillations. Again, so oscillations are more related with recovery and growth, and the sustained more with permanent cell cycle arrest. And we also used a xenograph we measured p53 exactly as we did in cell line. We found the exact same thing. If we use a small molecule to change p53 dynamics from oscillatory to sustain, right, then we actually lead to a better control and shrinkage of tumors. So basically, in all these studies, we measure total p53, right? Because we use a reporter, we take p53, we fuse it to a fluorescent protein, and we just follow it over time, right? So it's total p53. But we know for many years that p53 can modify a lot. And a lot of acetylation and simulation and phosphorylation and any type of modification that you can think of, p53 has it, okay? And so a few years ago, we started to wonder whether different pulses of p53 have different modifications. So if we look at the first pulse, maybe it's modified in a few sites, and then if we look at the second and the third, maybe it's modified in other areas. So even though total p53 look the same, these pulses have the same amplitude, the same period, the species of p53 in terms of modification, does it change between pulses? And the best way to do that is through mass spectrometry, right? And in the last, 15 years, at least more than 10, we've been collaborating with many across the US to try and use mass spec to probe modifications on p53 for reasons that I don't fully understand. p53 is one of the most difficult proteins to do mass spec on. I'm happy to talk with each one of you during breaks and coffees and tell you about all the different things we tried and what didn't work. But at some point, one of our collaborators from Northwestern, Neil Keller, I know we have another talk from this group a little later today, suggested that we do something different, which is a system that he was working on at the time, which is top-down mass spec, which is basically you don't digest the protein, you take the entire protein and you run it through a mass spec, and you can identify things that we call mod form, different modification forms of p53. So we don't know where the modifications are, but we can tell how many different species they are and what is their abundance, what's the distribution of these. And so we said, you know what, that's good enough. Let's start with that, right? And we can compare the first and second pulse of p53. If the mod forms are exactly the same, then we're done. That means that they're similar and we don't need to dive deeper. But if they're different, then we'll try to find new ways to understand what these modifications are. So this is how data from mod form look. Here, this is the least modified protein, and the green go here is the more modified, and you get distributions of species in different modifications. And so we did this for p53. We took samples from the peak of the first pulse and the second pulse. And this is the spectrum that we get, first pulse and second pulse. Again, here is the least modified p53, and here is the most modified p53. And when you compare both of these, they look pretty similar. But if you look carefully, you see that there are a few places in which there is a species here of p53 that doesn't exist in the second pulse. And again, here's another example, exists in the first pulse and not in the second pulse. So that was enough to motivate us in order to try and identify which potential modifications exist in the first p53 pulse and not in the second. And then we went into the good old Western, and we used all the possible existing antibodies to find which modification may change between the first and second pulse. Here is an example of few that we tested. We look only at first and second pulse of p53. And many of them don't change, they look the same, or they're just not there. But we realized that two of them, acetylation at 373 and 382, appear in the first pulse and disappear in the second pulse, right? Even though we load similar levels of p53. And if we look at them over time, we see the same behavior, right? This is total p53. We see the first pulse and the second pulse. Remember, here it seems a little damp because cells lose synchrony. But when you look at the acetylation, it exists on the first pulse, disappear on the second pulse, okay? So this is one modification in which the species of p53 is different between first and second pulse. What could lead to this difference? Oh, here, if we quantify it, we see this is total p53, and this is acetylated p53. And so we thought, let's see if we can actually change this by using small molecules and inhibitors to understand the function, the potential function of these modifications. And the idea was to take the same experiment in which we have total p53 having two pulses that are the same, and acetylated having one pulse and a smaller pulse, and then to add a deacetylase inhibitor between the two pulses, right? And see if we can enhance the acetylation on the second pulse. And the answer was yes, with one particular inhibitor. We saw that when we use an HDAC inhibitor, then on the second pulse, then we can enhance p53 acetylation on the second pulse, okay? So telling us that during the second pulse, there are deacetylases that remove this acetylation from p53, and this is why we see the difference between the first and second pulse. So what we can do now, we can use this similar setting and look at target genes of p53 and see whether they care at all, if it's acetylated or not acetylated. So we started with just a set of what we call canonical target genes and compare their levels in the same setting I showed you during the second pulse, right, without HDAC inhibitor and with HDAC inhibitor. And you can see that some target genes, like this is P21, a cell cycle arrest protein, GAD45, that's a DNA repair protein, another DNA repair. Some of them don't really care, right? The induction is pretty similar, but there is a group of genes that really like P53 to be acetylated, because when we add the HDAC inhibitor, that leads to much more acetylated p53, we see a strong induction in the level of these genes. And this is an apoptotic gene, PUMA, and these are two genes participate in senescence. Of course, HDAC inhibitor does a ton of things to cells. We could show that if we knock down p53, we don't see that difference, but this means it's p53 dependent. It doesn't mean it's the acetylated dependent. So in order to really prove that, we had to go and mutate these two sites and see if we see differences in target genes of p53. So we created mutation in one of the residue that has chain 373. In the second one, we also, as a control, created a mutation in another acetylated site that did not change, 381, and we created the 2KR that has both of these. And just in order to squeeze everything in short time, I will just show you the final results that we got for the 2KR and how it impacts target genes of p53. So basically what we did, we had created two cell lines. One is expressing the regular wild-type p53, and one is the mutant that cannot be acetylated on these two sites. And we did RNA-seq and collected samples for the first 3 hours after radiation and compared, okay, globally, all target genes of p53 and basically all mRNA between these two. And I would just say what we expected to find and then what we find. So I was under the impression that what we'll see is either genes that don't care if it's acetylated, like what we saw before, and many genes that will love it when it's acetylated, that will prefer it's acetylated. But the picture was a little bit different. And let me show you this piece of data. So what we see here, this is the expression level of a target gene if p53 is mutated. And this is the expression of a target gene if p53 is wild type. Okay, the wild type can be acetylated, the mutant cannot be acetylated. Everything in gray here are genes that don't care, right? They are induced similarly, whether it's acetylated or not, right? Whether it's wild type or mutant. Everything in red here, these are genes that have preference for the wild type, meaning that p53 can be acetylated. These are the ones that we expected to find, right? That they prefer acetylated p53. But then there was this group here of these blue genes that actually prefer non-acetylated p53, okay? That they are induced much better when we put the mutant that cannot be acetylated during the first pulse, all right? So we realize, and as you can see, this is really gradual, right? It's not like a complete separation. You can see that there are genes that care a lot if it's acetylated, and genes that care a little bit, and those that don't care at all. So, the emerging picture from everything I told you is that there is an interplay between the dynamics of p53 and modification in order to temporarily control the different programs downstream of p53, okay? There is a gradual, if you look at p53 target genes, some of the genes prefer acetylated, some don't care, some prefer deacetylated. And so in the first pulse, when p53 is acetylated, it pushes more the programs that prefer acetylated p53. And then in the not acetylated, in the second pulse, it activates a different program that prefer the non-acetylated p53, okay? And in a way, this study has been very satisfying because for many, many years, we're asking ourselves why p53 oscillate, like at all. And this suggests, if you think about a goal for a cell to temporally order something, if the gene doesn't oscillate, if the protein doesn't oscillate, then you are left with protein from the first pulse that may have modification from that pulse. But if it's oscillating, then you get rid of all the protein that was there and you're expressing a new fresh protein that can be modified in different areas. What we would love to do here is to take these modifications in the single cell level and to combine our live imaging with measuring modifications with single cell proteomics. So I'm very excited about what I'm going to learn from this conference here. So this is what I wanted to tell you today. These are the people in the lab and our main collaborators. The postdoc who was leading the work on p53 modification is Dan Liu, and this is in collaboration, as I said, with Neil Keller from Northwestern, Jeremy Guanadena at HMS. And thank you so much for your time. I'm happy to take questions. I love questions, so happy to take them. The timing of each pulse of p53 is about 5.5 hours in human cells.
So, do you also see the modifications of p53 within the same pulse, like how it changed? Because as you said, it's a 5 hours, which is very long time for a protein to change the dynamics.
Right.
And also, when you take the samples from different pulses, what is the criteria? Are you taking exactly the same time for the both pulses to compare, because apple to apple comparison? So I'm just curious, like.
Yeah. So the total p53 we can measure at the single cell level through dynamics, right? So we sample it every 10 minutes, right? So we get a really high temporal resolution. When we look at the modifications with mass spec or with Western, we look every hour or every half hour. So basically the temporal resolution is different. And when we compare them, we take the peak of the first pulse and we take the peak of the second pulse, right? So we take when the protein is highest. But remember that when you do it at the population, the first pulse is the most synchronized. The second pulse is already a little less synchronized. So it's probably not going to be the peak of all cells, right? On average, it's the peak, but probably some cells are still on the way up and some already starting to go down.
Show the data for first and second pulse, use about how many pulses you have been quantified with PTM.
We normalize this to total p53 to make sure that because the levels are different, that's not the reason that we see less acetylation. Yeah. Yes. Hello, Katie.
Hello. Thank you so much, Galit. It was a really beautiful talk. I'm wondering for your acetylation modification, do you think that you're actually getting differences in binding to those target genes, or is this a more indirect effect?
Oh, I love this question. So I think we know the answer to that because a few years ago, we actually looked at p53 binding to its target genes, to DNA, during the first and second pulse, and it was exactly the same. Okay, so it's not binding. We also see p53 bonds in tons of areas in the genome, but the activation is much, much smaller. So I think it impacts the ability to bind some cofactors or that impact the transcription itself and not the binding.
Do you think these modifications are affecting the transcription component more, or is it affecting the stability of interactions of MDM2? And it's actually, kind of affecting the pulses, like the acetylation may encourage degradation or not?
Yeah, that's a great question. I think because the mutant still oscillates, I don't think it impacts the stability or the feedback with MDM2. And it is known that the stability with an MDM2 is mostly affected by phosphorylation on p53. So I think these are more impacting the ability to transcribe DNA, mRNA. I cannot hear you.
Very beautiful work. I
Thank
have
you.
a very general questions. There are a lot of mutations on p53, and some are functioning to impact the patient's outcome. But in terms of the time scale, the pause may affect the binding of or recruiting of other partner or the protein modification, which is faster. The change of the modification or the interacting partner, which may come first, and how will the mutation also change the dynamics?
Yeah, so you're asking about the mutation that we use, the acetylation, or mutations in cancer? Both, okay. So as I just said, the modification on the acetylation, the inability to acetylate, doesn't change the dynamics. They still oscillate. Hotspots in cancer are usually mutations in the DNA binding domain. So you get a protein that cannot activate target genes. It cannot activate MDM2. So it totally disrupts the oscillations, right? You get completely different dynamics. And in terms of the speed, so the induction in the level of p53 is relatively quick. And it's a little hard to distinguish between the speed of modification in protein, because first you need to have a protein in order to modify it, right? What we do see in the first pulse, it basically mimics each other. So the same kinetics that we see from the protein, we would see for the modified protein.
Hey, wonderful
Hi.
talk. I'm Hong from Chinese Academy of Medical Science. So I have two questions. So the first is a technical question is when you do the optimum aspect, you see the two peaks which are different, and then you did Western to see the acetylation, does it match? That's the first question. And the second question is you measured the first pulse and the second pulse. How about the following ones? What do you think is every pulse mean different biology or just
Yeah, no, they're great questions. So we can't tell if they match because the mod form, the top-down, doesn't tell us where the modifications are. Neil's lab group are working on that, and I think we'll get to a point that we'll be able to figure this out. But for now, we just know that from the top down, they're different. We don't know where. In terms of the subsequent pulses, that is a super interesting question, whether they look like the first, the second, something different. Technically, we're a little stuck in our ability to do it until we can do single cell proteomics and measure these modifications at the single cell in a way that is not antibody-based, because these antibodies don't work well for immunofluorescence. So please develop a method so we will be able to answer this very exciting question.
This is Patrick from TGen. So I think you already answered the question, but you don't really know where it's happening, right, on the DNA. So I was wondering if you did any kind of ATAC-seq and looked at chromatin states between the pulses. Would you imagine it changing a lot? I mean, that's what I would suspect.
Yeah, we haven't done ATAC, we haven't looked at that. I think looking at chromatin state and see how it changes will be fascinating. So that's certainly something we would love to do. I'm at Harvard, we can't hire anybody, right? So we're a little stuck in our ability to do any of these projects, but that is on the list. This is like the next most obvious exciting things. I agree. Yeah, maybe someone wants to collaborate. Yeah.
I wonder how the oscillations happen. So is this transcriptionally driven, or this is purely on protein, like the protein is changing, but the mRNA is not changing? And maybe also how common is this mechanism? So the oscillation, is it a p53 specific thing, or you can think of many other regulators behave the same way?
Yeah, so on p53, the oscillations are at the protein level, not the transcription. mRNA is flat, it doesn't change. It's really just the stability of p53 through MDM2. And you're absolutely right, p53 is not the only one that oscillate. The upstream kinases that activated, like phospho-ATM, phospho-CHEK2, they oscillate as well because there is a loop from p53 back to them. So there are a lot of things that are oscillating, right? It's a little hard to say what is the cause and what is the result, but a lot of protein at the DNA damage response oscillate. We actually did a mass spec to look at everything every hour, and we see a lot of DNA repair protein that oscillates, phospho protein that oscillates. So this is just probably one of them. Mm-hmm.
Nikolai Slav from Northeastern and PTI. Wonderful talk, Galit. Very much enjoyed it.
Thank you.
And one expectation that I have based on it is that if you're able to measure nascent transcripts, you'll be able to see that the first pulse is associated with higher increase of those genes whose transcription was more induced without acetylation, and conversely, the second pulse would be associated with more nascent transcripts corresponding to the different mod form. Can you measure this? Is this also a way that you can use to identify differences in transcripts induced in more pulses?
Absolutely. Yeah, I think that is possible. We have, again, from a few years ago, another study that we look at mRNA and nascent transcript, but we haven't connected these two, but I think that is very feasible. Yes. Yeah.
Thank you.
Thank you.
Any other questions? I think there might be one online.
I think we have that.
Yes. As we move forward with more and more data, can we turn it into music? The trained ear can detect a wrong note.
Oh, as we move forward with more and more data, can we turn it into music? The trained ear can detect a wrong note. We could. I think if we want to train oscillations into music, I'm up for it. I love music. So yeah, reach out, whoever wrote that question, and we'll turn. We have a lot of movies that we collect, and I always feel like we need to have some soundtrack to put it in the background. So that could be a way to do it. All right, thank you.
Get it?
What's the plan? I think I sent it.
Morning, everyone.
Presentation, and I didn't know.
It's a great pleasure to be able to introduce our next speaker, Andrew Leduc. Andrew comes to us from Northeastern, and he's made important contributions to moving single-cell proteomics forward as a field. Andrew developed an approach that massively scales up our ability to prep samples for single-cell analysis, an approach called NPOP, that allows us to collect and prep thousands of cells in a single day. Really exciting development. And Andrew's used that approach and other approaches to make important discoveries in the field of single-cell biology, one of which he'll talk about today. But with the time that I have to introduce Andrew, I also want to highlight another thing that I think is an important aspect of Andrew's character, which is his willingness to help others get into the field of single-cell proteomics. So Andrew has traveled around the country, around the world, training others in how to use NPOP. He's readily shares data. He's actually come on site to our lab and helped us by sharing data, by helping us learn the protocols that he's developed. And I think that's really important because single-cell proteomics moving forward as a field depends not just on people, individuals making great discoveries like Andrew's done, it also depends on empowering others to innovate and to make their own discoveries. And Andrew's done a great job with that. And in doing so, he's really established himself as a great leader and a colleague in the field of single-cell proteomics. So please welcome Andrew.
Thank you. Thanks so much, Malin, for that really nice introduction. I think this is my 7th single-cell proteomics conference. The first one I was an undergrad coming just out of undergrad in chemical engineering, and I think I thought proteins were mostly a dietary supplement. So hopefully I seem slightly more knowledgeable in my talk today.
Go.
So today I'm going to be telling you a little bit about our recent work quantifying modes of regulation for protein abundance in single cells from primary tissues. Get this guy off the screen. You move that box. Maybe it won't get in the way too much. All right, so multicellular life, as you know, has this amazing ability to create a diversity of cell types from a common set of genetic information. And of course, these diversity of cell types arise because these cells regulate their genomes to express different subsets of proteins and express these proteins at different levels that give rise to these diversity of different functions that these cell types have. So it goes without being said, if we want to better understand the diversity of cellular phenotypes that arise in multicellular organisms, we'd like to be able to measure the proteins that these different cell types express so we can learn a bit more about their function. And one of the early technologies that allowed us to get an idea of what proteins are expressed in single cells for thousands of genes across thousands of single cells was, of course, single-cell RNA sequencing, which lent us insight into the transcriptional processes that lead to the production of different proteins that allow cells to carry out different phenotypes. Yeah. Sorry.
Hold on one second.
And more recently, of course, single-cell proteomics has emerged, and I'm sure you'll hear a bit more about the development in this of these technologies from others in the talk, and they're technologies that I've been lucky enough to have a chance to work on in my time in Nikolai's lab over the past 7 years. And of course, while it's great to measure protein abundances to get a better idea of how these cells work, we also want to know the methods these cells use, the ways in which these cells are able to regulate the protein abundances. And while this is interesting, not just because it helps us learn about the biological circuits that control cells, of course, this information can also be practical if we want to manipulate cell states and devise treatments. As we just heard in the last talk from Galit, that if you wanted to modulate P53 levels, you might be interested in modulating them at the level of protein degradation, where the pathways are really controlling the abundance of this P53 protein. And if we think about methods that we are really commonly used to study the ways in which protein abundance is regulated, a lot of these work at the level of methods that kind of control transcriptional processes. So we have methods to explore kind of transcription factor binding and DNA interactions and methylation and histone modifications. And ultimately, these are a really important set of regulatory processes that give rise to differential transcript abundances across single cells, which ultimately influence the proteins they produce. But as we know, the regulation of protein abundance doesn't end at transcription. There's many translational processes and regulation of protein clearance that play a role in kind of developing and contributing to the ways in which the proteome is shaped across different single cells. So if we want to get a better idea of what the contribution of these kind of post-transcriptional processes are, translation and protein clearance, to determining protein levels, one approach that we could take would be to sequence single cells at both the level of mRNA and at the level of proteins. And recently, this work was done in our lab led by Sa Khan, where he took cells from the human testes and measured single cells of the protein and at the mRNA level and integrated this data. And he could use this data to assess the extent of discordance between protein abundances and mRNA abundances in single cells from a given cell type. And from these measurements, you can kind of infer a difference between the protein and mRNA levels and determine the amount of post-transcriptional regulation that's kind of going on past just the level of mRNA abundance. But you might be interested in taking it a step further and narrowing this down and learning whether it's really translational processes or processes in regulating protein degradation and protein clearance that contribute exactly to the discordance that you're seeing between these mRNA and protein abundances. And a really useful technology for being able to do this is the metabolic labeling of proteins with heavy amino acids. And this technology works by essentially at time point 0, you spike in these heavy amino acids and proteins are slowly synthesized. And you can measure these on the mass spectrometer with these kind of peaks that pop up, the number of Daltons heavier for They're being cleared as the cell kind of divides more rapidly than the protein half-lives allow. So to try and answer this question for tissues in the kind of a wide array of tissues in the human body, we applied this metabolic pulse labeling strategy to in vivo in mice, which you can do simply by, instead of kind of changing the cell culture media to have heavy amino acids, you can just feed the mice this different food that has these heavy amino acids, and this allows you to measure these protein synthesis and degradation rates in this kind of wide array of tissues. And when we measured synthesis and degradation rates in the brain, one of course, a tissue where there's really slow cellular turnover, of course, neurons mostly kind of never dividing in the life of an organism. What we saw was that there was actually a really strong relationship between the rate at which proteins cleared and the kind of overall abundance of these proteins in the cell. And thankfully, since we measured other tissues that had a kind of variety of growth rates, when we looked at these trends as a function of the average cellular division time of the tissue, we found this strong relationship where the faster cells divided in tissues like kind of the bone marrow, we found very low control by protein degradation, and that kind of gradually increased as the doubling time of the cell got longer. And here I just wanted to talk out about, point out kind of the type of regulation we're talking about is within these samples, the ways in which the abundance of kind of different proteins are controlled within a cell. But of course, it's also important to think of the kind of converse, which is for a given protein, how is its abundance controlled kind of across different tissues, across different cells by these kind of mechanisms of degradation. And what we saw was when we compared the rates of protein clearance for two tissues that kind of divided more slowly, the brain and the pancreas, what we found was that kind of off scatter points here are going to suggest differences in degradation rates between these two tissues. And when we compared the fold changes for clearance rates to the fold changes for differences essentially in protein abundance between these two tissues, we also found that the relative differences in protein levels for the same gene were kind of strongly controlled by differences in protein abundance. And if you want to hear more about this kind of brief story on the role of kind of dilution in determining the ability protein degradation has to determine protein levels, you can read a bit more about it in our recent paper. So back to single cells, are we also able to measure these mechanisms and also kind of transcriptional mechanisms in single cells and not just at the whole tissue level? So we recently set out to do this again, and we were able to kind of feed these mice this special diet, and we were here analyzing cells from primary kind of murine tracheal epithelial samples. And so we took certain cells for proteomics, and then from kind of the same type of dissection, we took cells to sequence the mRNA through just 10x genomics. And for the protein prep, we utilized kind of these multiplex mass spectrometry technologies where for each run on the mass spec, we were analyzing two single cells at a time, and we achieved this with kind of these heavy isotope tags that we are able to kind of put onto the end of the peptides we're analyzing. And when the peptides come into the mass spec, that you kind of get this trend where you see a peak corresponding to the first cell, which has its light and heavy component. And then you have a kind of this 16 Dalton separation, where now you can kind of see, again, the differences from the second cell. And these technologies allow us to kind of increase the number of cells that we analyze and infer information kind of for more cell types. And so after collecting this data, we generated a data set that had about 4,000 single cells at the protein level and about 36,000 single cells at the mRNA level, with roughly 2,000 kind of shared genes quantified between these. And when we looked to try and see the cell types that we were identifying in these two samples, what we found is that there was kind of, of course, as we expected, we found kind of similar cell types between the two samples. And we wanted to compare to make sure our assignments were kind of confident and similar between these two data sets. And so to do that, we can just compare the relative levels for kind of like cell types at the protein and mRNA level. And of course, mRNA is a really important part of the regulation of protein levels. So we expected to see some strong agreement. And here you're just looking at, I'm just visualizing two cell types, and we can see is kind of the relative average mRNA level and average protein levels across cell types kind of agree strongly for the two cell types that I'm showing. And we can extend this visualization to all the different cell types that we are able to measure. And on the diagonal there, it's kind of the agreement between the kind of the average protein levels within a cell type at the protein level and at the mRNA level. And this kind of gave us confidence that our integration was faithful and that we were kind of comparing apples to apples when we were comparing two cell types at the protein and the mRNA level. We can also look at the kind of reproducibility of the measurements we're making. So here we're just taking kind of split pools of cells from within a cell type and comparing kind of the agreement. And we see that kind of we have strong reproducibility for our mRNA and our protein measurements. But again, we wanted to be able to kind of measure these translation and clearance rates for these proteins as well. And again, so we can apply this metabolic labeling technology to also infer kind of the degradation rates. And then with this equation, with these kind of 4 parameters, we have the leftover variances going to give us the ability to understand the translation rates in this for these proteins as well. And so we can also assess the reproducibility of the degradation or clearance measurements, and as well as the translation, which is going to be defined kind of as translation number of molecules produced per copy of mRNA. And so in a given cell, we can quantify these degradation and translation measurements for a little over 500 proteins, and we can quantify abundance measurements for about a little over 800 proteins per cell, with the total set being about 1,500 for degradation and translation measurements and a little over 2,000 for abundance measurements. And you might be wondering about the variation in these distributions of box plots, why some cells might have more proteins quantified than others. And a lot of this comes down to kind of the size of the cell. For bigger cells, we're able to kind of quantify more proteins. And you can see here this really nice scatter between kind of the volume of the cell, which we can measure through our sample prep method, and the total amount of protein that we're measuring, which kind of gives us another layer of faith in the reproducibility of our sample prep for these samples. And so earlier I was talking about the ways in which kind of growth played a role in determining the influence of clearance in these cells. So ideally, we would also like to be able to measure directly the rate at which kind of cells are dividing in these samples. In addition, so we can kind of disentangle influences from protein degradation and growth kind of due to dilution. And so this growth, again, is just kind of proportional to the doubling time of the cells. So really, if we measure the cell cycle time, this gives us the ability to infer kind of this growth parameter. So how can we kind of measure the cell cycle time for these populations? And when we look at the turnover of histone proteins from these samples, what we can see is we get these actually really discrete distributions, because histones mostly are not really degrading very quickly, except for that one kind of point in cell division where they have to kind of double the amount of histones produced. So it leads to these discrete bins here, kind of reflecting cells that have divided once or twice. And we can use this information to compute the doubling time or the growth rates of the cells. And you see that there's a nice amount of kind of variation across these cells, with some cells like these basal cells, which are kind of these stem cells that replenish the epithelium and the dividing quite quickly, where other cells like these tissue resident chondrocytes dividing quite slowly. And so what effect does this have on the kind of differences in measured clearance rates? You can see that when we compare cells that divide quite quickly with cells that divide quite slowly, it kind of shallows the slope of the measurements between the clearance rates. And essentially what this means is that for cells that divide more slowly, you're going to have a lot more variance in the clearance rates compared to cells that divide kind of quickly, and that high growth term is going to kind of limit the cell protein to protein difference in clearance rates that can kind of be obtained. And you see that kind of relationship as you compare cells that kind of have a smaller growth differential, that slope kind of narrows closer to 1. And another way to kind of visualize this is to kind of just plot the variability of these clearance rate distributions as a function of the growth rate. And you can kind of see we have this nice trend. The question is, do we see these same trends recapitulated in the single cells that we found in the tissues, where cells that kind of grow more slowly have more control across proteins due to degradation? And what we can see in the cells that chondrocyte cells, which are some of the slowest dividing ones we measure, there's quite a strong association between clearance rates and, again, this protein abundance, much kind of more so than that 5% that was originally measured in these rapidly dividing cancer cells. And so we can see this trend again, we can see this kind of similar trend with the correlation to the growth rate of the cells. And the question you might have next is, what processes are kind of compensating for this, picking up the slack, and, for cells that divide faster, that are contributing kind of to a greater extent to the amount, to the regulation of proteins. And what we can see is in particular, the kind of translational control per mRNA seems to kind of increase considerably with the rate of the cell and, potentially making up for that variance that's kind of taken away as the differences in protein degradation throughout the cells. And of course, we didn't just want to measure kind of within a cell type the protein abundance control. We also wanted to look at this across cell types. And so one way we can do that is for a single protein, we can plot the change in kind of concentration across different cell types. And so in kind of, you can see the mRNA in the black line, and you can see the protein levels in the orange line. And here we have quite strong control for this protein by transcription. And so we can compute a correlation from this trend, and we can kind of visualize that distribution of correlations, and we can see that on average, this trend is quite strong. But of course, for other proteins, we see that there's kind of a less degree of discordance, and we might kind of suspect that other modes of regulation are important for this protein. We can also visualize the same trend for degradation rates. Here, I've plotted kind of the protein half-life in blue and not the degradation, because the kind of half-life is going to vary with the inverse of the protein. So, but the half-life, think protein degrades more slowly, you'll have more of it. So you again see kind of a strong correlation for this particular protein to the protein half-life. And then when we look at the distribution again of correlations between protein abundance and this degradation rates, we see on average this negative trend, which suggests that maybe to a lesser extent than the mRNA, but there's still some kind of control going on by protein degradation. And so for this particular protein, we can, it turns out that that protein that we just visualized with poor concordance with the mRNA is strongly controlled by degradation. And so you can kind of see how we're able to unpack these modes of regulation for kind of individual proteins that might be regulated similarly across single cells. And we can visualize this not just for one protein, but we can kind of see this trend generalized across the whole proteome. And what we can see essentially is for proteins that have high negative correlation with their degradation rates. So that's to say, as the protein degrades faster, there's less of it. We find that the correlation between the mRNA is also decreased. So kind of we're seeing this trend generalized past just this kind of one single example. But of course, we don't necessarily need to assume that this degradation, the modes of regulation, are going to be kind of continuous across all the cell types. We might have cases where a protein is degraded by degradation in one cell type, and then translation drives the protein abundance in another cell type, and transcription in another cell type. So we want to also look at these trends kind of on the individual cell type level. And so take these chondrocytes that these cells rely heavily on glycolysis. And you can see for this particular protein, glycogen phosphorylase, on average, it's about twofold higher in these cells. And so these cells are kind of they're tissue resident cartilage cells, and they kind of live in low oxygen environments, of course. So these cells really kind of rely heavily on glycolysis versus kind of respiration. So when we look at the control by mRNA, we see that the mRNA levels are kind of quite not very differential for this protein and not kind of contributing strongly to the increased abundance of this protein. And from the previous work, we would maybe look at the difference between these protein levels and mRNA levels, and we would infer some difference, and we would say that kind of post-transcriptional regulation is happening kind of in the cell type on some level. But now we can take this a step further, and we can look at the contribution of translation, and we see that that also is not strongly contributing to the increase of this protein. And we can remember that these chondrocytes, when we were kind of looking at the regulation across the proteome, we found that degradation was quite important for these kind of slowly dividing cells. And in this case, we can see that the slower degradation of this protein in these cell types allow them to have higher abundance of these proteins that participate in glycolysis and kind of allow the cells to kind of carry out this important metabolic function and provide energy to these cells. We also measured, again, these kind of basal epithelial cells. And one of the markers of this cell is kind of this keratin 5 protein. And we can see that this protein is kind of massively upregulated in these cells. It's about 6.6 on a log 2 scale, so almost 40 times upregulated kind of versus the other cells where keratin was measured in this population. And these keratin proteins are quite important for all these kind of different epithelial cells that are kind of constantly proliferating and kind of moving up towards the surface. They need to express different keratins to kind of hold different shapes. And so if we're interested in learning about kind of the regulation of this protein, we might suspect, of course, the mRNA abundance needs to be high in order to kind of produce a 40-fold increase in this abundance of these kind of keratin proteins. But it seems like the mRNA fold change is kind of not sufficient to kind of explain that really 40-fold kind of high abundance. So you might think that maybe the protein is degraded slower so that it kind of can accumulate in these cells, but we also find that that's not the case when we look at the contribution of degradation. And that allows us to kind of understand that not only is the increase in transcription important for these cell types, but also the translational kind of per mRNA translation of this protein is important to kind of allow these cells to have high abundance of this kind of keratin protein. And the fast turnover might be because, these cells kind of need to divide quickly and be able to kind of differentiate into cells and kind of lose that keratin protein so they can take on different shapes. Okay, and so with that, I think I'm about at time. I lost track a little bit, but yeah, okay. So I'll just say thanks so much to the organizers and to my colleagues in the Slavov lab, and also our collaborators at MGH who allowed us to kind of do all these animal experiments and helped us collect the data that we talked about here in this presentation today.
Someone asked a question on the chat. So thank you for the excellent presentation. Could you please provide an estimate of the minimum number of cells required to reliably annotate the population?
Yeah.
Use cell markers derived from mRNA data or from?
Yeah, so there was a question online about kind of the number of cells needed to quantify different populations at the protein level and how we were able to kind of assign cell types, whether the markers were coming from mRNA data. In terms of having enough cells to kind of reliably assign a cell type, I think we pretty commonly find that we can identify these cell types with kind of quite a small number of cells. I mean, of course, you want to measure enough so that you can kind of compute robust statistics. So, as long as you analyze enough single cells to get, 10, 20 cells from this population, you're usually able to identify them. And these markers came from some at the mRNA level, but also some kind of that have been studied in measurements with there's been some that have kind of these markers have been validated through antibodies. Here you're seeing these kind of keratin 13 plus subset of these epithelial cells, which we were studying that kind of, you can kind of clearly see these markers with antibodies and fluorescent-based proteins. So it kind of came from an array of sources. Thanks.
So I think you may have just answered it, but these are hand-curated marker proteins for each cell type?
Yeah, so in the original assignment, when we kind of originally assigned these populations,
You map way back in the middle.
yeah, we had. Yeah, so to be honest, there's like a host of different methods you can do to integrate these data. In the past, we've kind of relied on popular tools like Liger to do the integration, which kind of also facilitate them. In this particular case, I found that the kind of clusters were quite clear, and you could, from kind of just doing a quick literature search and seeing what proteins were expressed in the different clusters, it was easy to kind of differentiate what cell types were where. And then, of course, mRNA abundance, while maybe not the complete story, is obviously kind of like one of the really most important modes of regulation for protein abundance. So you should have a default expectation that there's kind of a really strong concordance between the two. And I think with these kind of integrated analyses where we're kind of taking, not measuring mRNA in the same cells as proteins, it's of course like really important to make sure that your integration is kind of reliable, because any discordance in the integration of cell types can kind of lead you to misinterpret variation. Yeah, from it might just be a noisy integration versus, yeah.
Thank you so much, and thank you for persevering through your talk. We really appreciate that. Do this. So you mentioned that you can actually measure volume when you're in the process. So that potentially gives you the opportunity to then actually make very precise measurements about concentration as well. Is that something you guys are doing or can integrate? And can you tell us a little bit how you measure volume?
Yeah, that's a really good question. So doing the sample prep, we rely on this like cell sorter that's imaging based. And so for every single cell, we have a picture. And it kind of gives you this rough estimation of the diameter of the cell, and then we kind of just use that to compute the volume. I think like that's a really exciting analysis too. Generally, we have like the assumption that the concentration scales pretty well with size, but I think looking at the variation, like in general, is a really fruitful area of analysis. There's a slight question of whether the pictures that we get are like how quantitatively you can measure the volume, but I think like as that technology improves, it's a really exciting kind of way to analyze the data. Mostly I'm using it for like, if there's a massive disagreement between the volume computed and the cell, you kind of can suggest doublets that you might have sorted and things like this. But yeah, it's that and like image analysis of the morphology of the cells correlated with protein levels. There's kind of a lot there for people who are more inclined with image analysis.
So this question may not exactly relevant this one, but I just want opinion. Degradations. Are you referring to the proteasomal degradation or something, a different way of degradation? And the second thing is like, even if it is a proteasomal degradation or different degradations, whether the degradation is giving degrading the protein into individual amino acids or the fragments of the like peptide remnants. If they're producing a peptide remnants, when you do a quantification on the mass specs, how do you differentiate that the peptides that we are measuring are coming from these remnants or the intact proteins? So, and also a question is like, because when you're referring, sorry,
Yeah.
I want to complete the whole.
Mhm.
So, when you say protein degradation, I'm just also curious, did you ever try using any PROTAC to target a protein of interest to just compare this one with the real protein degradation stuff?
Yeah, that's a really great question. So, of course, this is like, you're getting slightly closer to understanding the regulation of these proteins by knowing kind of the main mode in which through they're regulated, be it degradation or translation or transcription. But of course, there's like a lot of different ways a protein could be degraded. And I think this is like a really powerful tool for kind of hypothesis generation for you understand that. So before you do any like perturbation experiments, I think there's kind of a lot of fruitful analysis to be done, just seeing like, the abundance of certain ligases increases in these tissues where you see the corresponding like relative increase in degradation. And this can suggest, whether it's kind of through an E3 ligase proteasome pathway, or maybe you can look at kind of different lysosomal proteins that are increasing on a lot in a certain cell type where you're seeing, faster degradation of other proteins. So I think kind of, just through like these associative correlative analyses, you can start to kind of develop more fine-grained hypotheses that will then allow you to kind of do better experimental tests to kind of exactly understand the precise mechanism through which these proteins are degraded. Through using kind of things like PROTAC or kind of even degrading certain E3 ligases that you found might be upregulated in a certain cell type, and seeing kind of how that influences these degradation rates and whether you can kind of pin down a more confident mechanistic relationship between a protein who's degraded faster in a cell type that happens to have more abundant a certain E3 ligase, and whether there's kind of a direct interaction there. So it's kind of closer to mechanism, but also maybe a good method for kind of generating hypotheses for more specific mechanisms.
All right, we have a couple online questions too, and I think what you just said is a good segue to the first one, which is for cell types that rely more on degradation, do you also see an increase in the abundance of the respective machinery? For example, the ubiquitin system enzymes.
Yeah, that's a good question. Certainly the abundance of the proteasome kind of varies a lot across proteasomal proteins varies a lot across these cells. I don't have any kind of data exactly to speak to this, but we see that proteins of the proteasome themselves are kind of controlled a lot through degradation. So I think their abundance is higher in these kind of chondrocyte cell types that divide more slowly. And I think that kind of reflects the fact that kind of specificity of degradation are more important in regulating the proteome of some cells versus other cells.
And then the next online question is says, amazing work. You've measured protein data for 4,200 single cells. 2 questions. How many proteins are you able to measure per cell? And how long did it take to measure 4,200 single cells? Is the throughput 2 cells per run with mass tags?
Yeah, great question. So the throughput was 2 cells per run. I think the active gradient was about 10 minutes. All in all, it took about kind of like roughly 2 months of mass spec time to acquire the data. And the reason that there's kind of difference in these two numbers, whether it's kind of proteins where we can measure synthesis and degradation or proteins where we can just measure abundance, is because we're only pulsing in like a heavy lysine, but we're using a tryptic enzyme to cleave the protein. So we also end up measuring a lot of proteins where we just have kind of arginine containing peptides where we don't get that kind of heavy light split. So yeah, I think there's like also exciting work to kind of pulse in other non-essential amino acids and increase the number of proteins, which you can also infer this for. And you can also use like other enzymes like lyse, which don't always perform as well as trypsin, but might give you a slightly higher number of clearance and translation rates quantified.
Andrew, thanks for the great talk. I'm curious if you have considered looking at aging, as a lot of age-related diseases are degradation dependent or related.
Yeah, good question. So this research was actually funded as part of a grant to study cellular senescence. And we have here some of the mice are like really young, just a couple of months old, and some of the mice are kind of 24 months old. We were really excited to study this. These experiments kind of are a bit expensive. And sadly, we had 2 of our old mice that were like 24 months old pass away like 9 days into feeding them with this expensive food, which like unfortunately like lost 2 data points in the experiment. So we don't like have great statistics for looking at age with this particular data set, but we definitely plan to kind of look at this a good amount.
Other questions for Andrew?
Just quickly, how do you take into account the dynamics of lysine in the body? Do certain tissues absorb it differently, and metabolism happens differently, and how does that impact?
Yeah, that's a good question. So we're able to get essentially like there's a parameter in the equations, which is like the free pool of lysine that's available. And we can get all right estimates for this in kind of on an individual single cell level by looking at peptides where there was a miscleavage. And so they have 2 lysines. And essentially, with some probability, they'll be translated with kind of both the lysines heavy or both the lysines light or one light and one heavy. And through like these, through kind of leveraging this information, we are able to compute the kind of roughly what the free lysine pool is, and then adjust the kind of measurements to understand these rates. And one like kind of quick sanity check for us is that here you would expect kind of cells that have divided once to have like roughly 50-50, and it looks like, maybe slightly more heavy than light histones because there's probably also kind of some turnover of the histones themselves in that process. But kind of doing the raw heavy to light intensities, these ratios are like kind of way slimmer, kind of much kind of closer to it's something implausible where like if a cell were to have divided, of course it has to renew at least half of its histones, like it can't still have 80% light. So yeah, that's kind of that's a bit of a sanity check, but also we have this kind of method for we're certainly not the ones to kind of develop. People who are doing protein turnover studies in vivo have kind of looked at it for a long time.
Hey, Andrew, very beautiful work. I'm just curious. So you listed like one representative protein, HSP9, if I remember right. It's like degradation contributed a lot to the measurement of abundance. Just wondering, I'm just curious, have you summarized like global protein level, like how the degree of measurement difference contributed by degradation?
Certainly. So I didn't kind of get to everything, but so like these kind of correlations don't kind of provide the full picture. Of course, you kind of still have to look at the magnitude of the trends. And so, of course, there's kind of 2 ways to answer this question. One is like the kind of magnitude of regulation within a cell type. And like kind of these numbers are going to sum to one. So, within a cell type across kind of the proteome, we see that some cells rely, much more heavily on degradation and like less so on translation in mRNA. At the level of kind of average protein across cells or kind of within a specific tissue, you can kind of look at can look at these different magnitudes. So kind of this gives you a sense, like for this given protein in this given cell type, you'll have, something like 80% degradation. You can kind of look at the average of that across all cells for kind of the relative control. And generally what we see is like, depending on the cell type, close to like somewhere in the order of 20% on average, like control of relative changes by degradation. And, but I don't have kind of the quantitative plots for that. But yeah, certainly a good thing to look at on the relative level as well as absolute.
So we've got one more online question, and then I think we're all ready for a coffee break. So it says, did you measure the percentage of dead cells in your cell suspension? Does it affect your measurements?
Yeah, that's a really good question. We actually had an entire article that arose spinning off that issue. Originally, when we were measuring these cells, we had like a weird subcluster that I was analyzing for months and like thought something interesting was going on. And then later realized that part of these cells from this kind of dissociation were kind of their membranes were permeabilized and they were leaking proteins. And so when we're sorting the cells, we stain them with like Cytox green and kind of do a negative stain for any cells positive with Cytox green to ensure that we have kind of all intact cells. But this is a really important thing to watch out for. And if you're doing a sample prep that doesn't have fluorescent capabilities, we made like a tool to detect dead cells. Essentially, they like leak smaller proteins and cytoplasmic proteins. And you can find that on our kind of SCP Slab Off Lab and our GitHub.
Thank you, Andrew. Let's thank Andrew. So we are ready for a break, but I can't resist saying one thing that relates to a question that Andrew received and that we are going to hear more throughout the conference. That while it has taken a couple of months for Andrew to acquire this data, you're going to hear in the course of this conference several talks that are going to showcase technology that can make it a couple of weeks or even less. So we should all keep in mind that what we're showing here is capabilities that we had a year ago when we started doing that project, and I think for a lot of, and that is true for a lot of the projects across the board. This is a field that developed so fast that in some ways, almost the price or not yet hit the bioarchive. It's starting to age. It's wonderful that we have a conference where the technology forward and is going to speed up progress. So with that, I'm very much looking forward to talking with you and enjoying some coffee and snacks in a short break. And we will return at 11 o'clock for our second morning session.
All right, well, good afternoon, everybody. It's a great pleasure to be able to introduce our first speaker of the afternoon session, Ryan Kelly. Ryan's been a longtime leader and innovator in the field of single-cell proteomics. He's developed a number of different innovative sample preparation methods, analytical approaches, and he's used those tools to come up with really great data sets that have really moved our understanding of single-cell biology forward. I was particularly excited by a paper he published last year, taking individual motor neurons from patients afflicted with ALS and looking at the proteomes of neurons that have pathology and do not, the sort of data set that you could really only dream about a few years ago. So really incredible work. And I think in the context of single cell proteomics more generally, one of the things that's been very exciting in the field is to see gains in throughput. And in that regard, Ryan's title, which you can see here, Single Cells in 5 Minutes or Less, I think is a really exciting title. And I'm sure it'll be a great talk. Please welcome Ryan.
All right. Thank you very much. All right. Well, I'm really appreciate you acknowledging that we did some biology because there won't be any of that in this talk. So I loaded my slides onto a USB drive to load onto this computer. And let's see. I don't want to. Okay. Okay. And I found a PowerPoint slide with this in it, and it's from August of 2016. So it's like an old USB drive that I just pulled out of the pile, and all the files in there were from 2016. And I saw this and I was like, oh yeah, 125 cells, and we saw a chromatogram. I have no idea what we identified, but we were clearly excited enough to put this screenshot into a slide in August 2016. Okay, so I am involved with this company, Micromics Technologies, and I just have to show this slide to show that yes, we make these really great columns and emitters and heaters and things like that. And we're working on an LC system that should be very flexible. Okay, so as an outline, I said no biology in this talk. Pretty much, I think I'll stick to that. Okay, so simple sample prep for label-free single-cell proteomics. We are very interested in at least TMT based single cell proteomics, and we are so bad at it that I think we need to borrow Andrew for a weekend or something and get us up to speed. So everything we'll be talking about here is label free. Then we'll have talk about our multi-column nano LC system that gets us to a throughput of 288 samples per day, or one analysis every 5 minutes. We'll look into some trap columns versus direct loading or trap columns, see how much we're losing there on the trap columns, because that's been a concern of ours. The impact of post-column dead volume on single-cell proteomics. I'm really stepping in it here, doing some comparison between the timsTOF Ultra and the Orbitrap Astral. And then we'll look at going to higher throughput, like 1,000 samples per day, label-free, potentially. And then how we're working to automate the data analysis as well. Okay, so getting started, sample prep, and this is nothing really new here, but just kind of a reminder that as we started with the nanopots approach, it was a lot of steps. Adding a reagent, incubating, putting it back into the system, adding the next reagent over and over and over again, like maybe 5 steps. And so this is a manual process. It involved a homemade robot, homemade slides. And so things have gotten a lot easier for sample prep, at least in the label-free space. We've kind of collapsed all of these steps down to ditch reduction, ditch alkylation, and since we're using this rapid trypsin from Promega that's stable at 70 C, that's also a good temperature for lysing cells and things like that. So we can essentially, one pot, 1 step, 1 hour, same proteome coverage, and we're doing this in the sometimes in the TCAN Uno or in the Cell-in-1 system, we can dispense, but we've kind of favored well plates rather than our old custom microfabricated slides, because the bottom of these well plates, the surface contact is similarly small and it works just fine. So we really don't see any compromise in doing this over a nano well. And with the exception that, at least for spatial proteomics with our laser microdissection setup, that is hard to shoot all the way. The collector is upside down and it's hard to shoot all the way to the bottom of that. So for that, we still use nanowell chips. All right, so yeah, that's basically get a cell in, dispense a reagent, incubate, do a low flow separation, and a high resolution mass spec analysis. This I've also been talking about for a little while. So it's our 2-column high throughput nano LC platform. And It uses a storage loop concept similar to what EvoSep uses, but there are 2 LC columns. So you can see if we fill a storage loop with sample plus its gradient, we can just use an isocratic pump to push that sample through the column, while at the same time we're regenerating the other column. And at the same time, we can be pushing from a binary pump through the trap column to load the next sample and gradient into the storage loop. And while that's happening, we can be queuing up the third sample and getting that ready to run. So besides 2 columns, we've got kind of 3 samples moving the system at any given time. And so Xiaofeng started kind of the original geometry of this, and Chow more recently has reduced this down to a 5-minute analysis. And you can see this is running actually on a LTQ, not even an LTQ Orbitrap, just an LTQ, because why would you want a good instrument collecting boring data for a week, right? So what we have is we're just looking, here's 1, 2, 3, 4, 5 peptides eluting from the system over the course of a week, and green is eluting from one column and blues from the other column. And you can see they don't perfectly line up, but it's kind of easy to kind of align those after the fact. And we've run here 2,000 analyses with the same setup, and we've gone to about 6,500 working on the Astral with the 2-column setup without changing the LC columns. So if you're going fast, you need to be concerned about if you're having a lot of sample carryover from one analysis to the next. But since we can, with this setup, we can sort of selectively elute the good stuff here and then direct the rest to waste. So the super hydrophobic stuff doesn't ever see the LC column. And so you can see we can elute in from column 2 here, and then from column 1, that didn't have any sample on it before anyway. So that is clean, but then going back to column 2, it's also clean. There's just nothing eluting there. So that's working well. Working with this system, Lavender Lynn, who's one of 3 women in my group that had babies in the last month, which is great. Here's 2 of the new lab members here. So she has kind of done a lot of the data analysis with the single cells that were run at this 288 samples per day. And you can see if we sample just Pierce Gila Digest wet from a well, we get about 4,300 proteins detected. And then if we allow that same sample to dry and then we reconstitute it and analyze it that way, and I'm not sure, maybe we didn't have DDM in the system for this, but we took a big hit, and that's kind of how we also analyze our single cells. But still, about 3,000 proteins per cell every 5 minutes is pretty good. These are looking at CVs of technical replicates here and biological actual single cells here. So it's good that single cells aren't all the same, or there would be no point of single cell proteomics, right? And this is very unexciting, but I guess we actually had a nice biological application in there, and something went wrong. So it's like the treatment didn't happen or something. So we're like, they don't look different. So something we have to work on. So if you want to test your quantitative performance, often in the proteomics field, we make these mixtures of human, yeast, and E. coli samples, and then we play with the ratio of yeast and E. coli relative to human. And so again, this is in that 5-minute analysis. The human, the yeast, and the E. coli together make about 385 picograms total protein, 5,900 proteins per sample. And This is just, what part is the human, the yeast in their different ratios, and the E. coli in their different ratios. And the observed to expected ratios look pretty fine here with that. Okay, so moving on to trapping columns, how much sample are we losing? So you can do direct loading onto an LC system using a 6-port valve, but the sample loading flow rate is restricted by the back pressure of this column. So one trick that like the Vanquish Neo does is just go to really high pressure for that. But I'm like, if we have a lot of pressure accessible to us, I want to use that for the actual separation too. Or you can work with a trap column, and our 2-column system's pretty dependent on trapping, where you have essentially a short LC column here that has a much lower back pressure, so you can do faster loading, and it does keep some of the nastier stuff away from your analytical column, which is nice as well. So this is looking with 10 nanograms HeLa, looking at no trap versus a trap column, and we really see quite similar coverage, quite similar hydrophobicities across those. So no deal breaker there. And then we looked at different trap columns themselves, like what do we make the trap out of? These could be 5-micron Dr. Maisch particles, 3.5 micron waters, symmetry particles. And you can see the peak width distribution here is pretty similar. So you have no trap in blue, and that 3.5 micron one is almost identical. And we get just a little bit wider peaks with these other two. And then we can analyze 200 picograms or 10 nanograms in 1 microliter or 10 microliters, and that all works quite well. We were surprised that we could, after prepping, dilute a single cell size sample into 10 microliters and not suffer appreciably from that. So that worked well. And then here, this is just looking at proteome coverage with these different particles and different loading flow rates. 10 microliters per minute, which would allow us to do really fast sample loading. Doesn't seem to hurt anything there. Okay, so next little vignette here is post-column dead volume and its impact on label-free single-cell proteomics. And as Jared from Ion Optics mentioned, they've been screaming about the benefits of an integrated LC column for a long time without like a union and a separate emitter. There are benefits to having a union and a separate emitter, which is when you bump that emitter into something, you just throw the emitter away and you still have a good LC column. So there are reasons to want an integrated column and reasons to need to be especially careful there. So what we've been doing for our 2-column system is essentially in front of the thermo systems is modifying the flex source with this 3D printed adapter that allows us to point 2 columns at the inlet of the mass spectrometer. And then we flip which one is this electrospraying and which one receives the high voltage. And that works well on a thermo system, but on our timsTOF, what do we do for 2-column LC? We can't, right? Because we've got the captive spray source there, and so we haven't had a good way of doing 2 columns into one mass spectrometer. So here's a photograph of an integrated emitter. The packing material goes to about here in our case. Then we have a little frit, and then we leave it open to minimize the back pressure there. A column with a separate emitter would be something like this. Here's your column, here's a union, here's the emitter, right? And then for 2 columns with 1 emitter, we thought, well, maybe we could put a 4-port valve after the LC separation. So column 2 would go here and might be directed to waste while column 1 is spraying. And then we would switch the valve. So column 1 effluent's going to waste while column 2 is going to the emitter. So that's what we were interested in doing. especially to get in front of the timsTOF. And so, and by the way, going from an integrated to or a non-integrated emitter to an integrated, up to this point, we had not seen much of a difference. This is with Gary's, where's Gary? There he is. So this is from a 2020 paper where we used a pico clear union and polished the column and polished the emitter. And we didn't see any change in kind of peak widths with or without an integrated emitter or a separate emitter. But these peaks were about 30 seconds wide. So you're not going to notice a big difference. But now as things get faster, the peaks are narrower. And so this is what we're seeing. If we have a separate emitter versus a valve with a 150 micron column operating at 1.2 microliters per minute, we see a little bit of increase in our peak width, and a little bit more when we go to the 100 micron column. And these are all 15 centimeters long, so the linear velocity and kind of the separation efficiency should be the same. We're just adjusting the flow rate by changing the column diameter. And at 75 microns, we tested an integrated emitter, a separate emitter, and a valve after the fact. And you can just see that we get narrower peaks when we use an integrated emitter versus a separate emitter. And about 2 seconds wide with an integrated emitter, maybe 4 seconds, 3 to 4 seconds wide. And so it doesn't seem huge, but this is what in terms of proteome coverage, a couple of features here. So we are going, this is 2 nanogram HeLa digest here, 10 nanogram HeLa digest on the right. And then in blue, this is going from a 5-minute to a 10-minute to a 20-minute gradient with a valve, or in orange with a separate emitter. And you can see it really is fine if you're working at 1.2 microliters per minute with this large column. It's also interesting that you can go from a 5-minute gradient to a 20-minute gradient, and the proteome coverage is very similar, right? But even going down to a 100 micron column operating at 530 nanoliters per minute, if you have a large sample, they're still not that different. But as you go to smaller samples, there's clearly a benefit to not having that extra post-column dead volume imposed by the valve there. Still interesting that the coverage is fairly flat. And then this is with a 75 micron column. Now we're looking at single cell sized samples, 10 cell size samples, and 50 cell size samples. And the smaller your sample, the bigger the benefit of keeping those peaks narrow and tall, right? A skinnier peak is a more intense peak. So we're getting about 4,000 proteins quantified in 5 minutes with this system. And now the last one is going down to 50 micron. And the difference is especially huge from a integrated emitter column to a non-integrated emitter column. And now we're up almost to 5,000 proteins every 5 minutes for that. So, and then you should rightly be concerned, like how much you might be undersampling at some point, right? And that would lead to really large CVs, right? But they're actually not too bad. So this is for the 50 micron column, 200 picograms, 2 nanograms, and 10 nanograms. Even at 5-minute analysis, if you have a larger sample, your CVs are, median CVs are less than 10%, and they're in the 13 to 15% range here. So that's for a 5-minute analysis. So it looks like we're able to go pretty fast. Okay, so now let's get political, right? Okay, so I think there's really good things for the community in general here, and for both Bruker and Thermo here. And the huge caveat is that we just used the same acquisition setting all the way across this, and same acquisition setting all the way across this. I think it was a 150 millisecond ramp time here, and this was a 4 Thompson isolation window on the Astral. But you can see, maybe you can see that the timsTOF has better coverage that we saw under these settings at 10 nanograms than the Astral, and then the situation is reversed at 0.2 nanograms. But not a night and day difference by any stretch here. It's also interesting to see in both of these that, you know, you can go 5 times more sample and your proteome coverage increases by this much, right? And on both instruments. And that's something that, this is a thermo poster, I think from last year's ASMS, where you can see that, right? So going from 10 nanograms to 20 nanograms, you get like 4 extra proteins identified, right? Again, looking at CVs for both instruments, they're great all the way across from 200 picograms to 10 nanograms. And it's interesting that going longer in our hands for both of these systems just does not provide tremendous gains in what we're able to quantify. In fact, with the timsTOF Ultra, we're maxing out at 10 minutes, and that would be 144 samples per day. And it's essentially flat up here for the Astral as well. One area that we're seeing is kind of the linearity as we go from 0.2 nanograms up to 10 nanograms. Our signal on the Astral goes up like exactly 50 times, and it's just super linear as you change the sample input. It's a little bit less so. This is still pretty good, 0.95 here for the timsTOF. But if you drop that 10 nanogram and just look from 200 picograms, 500 picograms, 2 nanograms, the linearity is improved on the timsTOF, and that is indicative of some saturation that's happening. I'll show you that. Okay, so this is just CVs. Again, these are pretty good CVs, even at 5 minutes on both instruments there. Here's where you can really see some saturation happening. And it is true, like the timsTOF Ultra 2 is tuned for single cells. And so to throw 10 nanograms at it, and especially where we are working at these very low flow rates, so it's even brighter ion source. So the 10 nanograms probably looks more like 50 nanograms. It just starts to saturate here. So, and I think this is a really good story for the proteomics world as well, is when you compare intensities that you observe on the Astral to intensities that you observe on the Ultra, they look pretty good. 0.83 at 200 picograms, 0.86 at 500 picograms, and then you start to see a little bit of saturation effects that drop it down. But isn't it nice to know as a field that these are both good instruments and the intensities that you measure on one agree with the intensities that you measure on the other? And there's always going to be some spread, right? Because 1% of these are not even correctly identified if you set your FDR to that. Okay, so this is just kind of cool. Okay, so Chow comes to us from the Dalian Institute of Chemical Physics in China, and he's like, well, why aren't we just making our own particles, our own chromatography media? And I'm like, well, because I have no idea how to do that. But he's like, oh yeah, it's easy. This has been around for 100 years. So here, and not only does he make these particles, but they are beautiful, right? So these are non-porous particles, so you have to worry about the sample loading capacity of these, but not if you're doing single cell proteomics or low nanogram proteomics. You still have plenty of sample loading capacity, but man, I could just pin that up on my wall. Look how pretty those are. And so with this, he has been going crazy fast with the LC. This is not like multiplexed, so it's not like we have, so 2,000 880 samples per day per se, but what this does show is if we had a system capable of 2.5 minute separations or more interface, you can actually, so this is 200 picograms, 1 minute, 2,800 proteins, 5 minutes or 10 minutes, and then with 2 nanograms, so this is just amazing to me that with label free, we can start to think about high throughput proteomics without multiplexing, because like I said, we're bad at it, right? But we can maybe do some fast chromatography to mediate that. Now the CVs are not great at 0.5 or 1 minute here, but they get pretty reasonable at 2 minutes and 5 minutes and 10 minutes. And we think we have some things we can do to maintain the proteome coverage while improving the reproducibility there. And what we found in a hurry is that if we do really fast analyses without updating how we're doing the data processing and analysis, we've just moved the bottleneck one step down without really solving the overall throughput issue. So Xiaofeng, the same guy that did that first 2-column system, and the same guy who's basically running Micromics, he was like, what you guys are doing with your data here is just disgusting to me, like moving things on hard drives. So he came up with this system where we have an automatic file uploader that goes to this server, backs things up, can distribute different jobs to different computers, and can provide kind of a simple readout of things. And this is very extensible, right? So right now the functionality is pretty limited, but I just feel like we have these awesome tools out here that we still have to, like, our data are being analyzed by computers, but you still often have to sit there telling the computer what to do. And this is what we're trying to avoid. We want a robot to tell the computer what to do, or a computer to tell the computer what to do. All right. And so this also helps us because it's analyzing the data as fast as it's collected, so we can see if something's going wrong there, and trying to make this as easy as we can. You can see, this is from our we had about 6,000 runs just straight, about 250 plus samples per day. This is back in October. So, and then the MS Connect can just kind of give us a quick readout, seeing how things are going. So hopefully I've shown some advances across the workflow for label-free single-cell proteomics. I think it's becoming easier all the time. Still not as easy as I would like it to be, but we're making progress. We have a throughput right now of about 288 cells per day, about 3,000 or so proteins per cell. And then we have this MS Connect to automate data management, analysis, and visualization. And with that, I'd just like to thank our sponsors. I hope these funding agencies are still there in a year. And thank my wonderful research team. And thank you for your attention.
Great. Well, I'll handle the questions, and I'm going to abuse my power as the holder of the microphone to ask one myself. So I think the sample preparation that you described is really exciting. We get rid of those steps. We hopefully minimize some sample losses. And so the plots you presented show that we don't see differences in the number of proteins we see. What I wonder is if we see differences in, say, the sequence composition or the subcellular localization of the proteins by those two different prep methods.
Yeah, absolutely. So we're probably missing, we're not doing anything to decross-link disulfide bonds, right? And so we're probably losing the specific peptides that are involved there. But that's, unless you really care about that, and there are great reasons to care about that, but for global proteome profiling is probably not one of those. So that's okay. In terms of we were concerned about hydrophobicities and things like that, but looking at the gravy score of each prep method, it doesn't show any bias that way. So
thanks.
Yeah, thank you.
So a very interesting talk. So when comparing the different gradient lengths, you were mainly looking at the total protein identifications, and also I saw coefficient of variation. Did you also look into the accuracy? I saw that you mentioned
Like
you
with
had the mixed species.
Yeah, the mixed
samples
species
there.
proteome.
Did
Yeah,
you-
these are pretty early, so we haven't. Even if we collected that data, I don't know that we've analyzed that data. So it's stuff to look at for sure. Yeah, and we love that human yeast E. coli mixture for that. I even saw it, and you can buy it. You don't have to prep it yourself because there's somebody in Europe that's selling it now. So yeah. Yeah, hi, Yuju.
Hi, very exciting development. I have a question about the peak width. When you convert the retention time in such a short gradient, how will that impact the peak width and the peak shape? And is it okay also for quantitation?
Yeah, so the peak widths, as you go to shorter gradients, the peak widths kind of naturally get narrower, right? Because they just have less time to disperse on the column. And so I'm not sure I followed the rest of the question. Yeah, so that's why we're looking at the CVs, because at some point you're going to be undersampling those peaks, right? And so that's a concern for sure. There are things you can do to ameliorate that. For example, match between runs is one way that you can do that if you get enough MS1 points across the peak. There's also we've also looked at the peptide CVs, and they're not very bad either, which is kind of surprising. But you're rolling up a bunch of peptide measurements into these protein measurements, which I think can effectively give you more points across the peak than you have at the peptide level. But it may also be, because certainly this is going to be a concern, right? If you go to sub-second peak widths, you're going to have to change your acquisition method. But experimentally, we're not seeing, if you have a 15% median CV, that's not that much worse than a typical 30-minute analysis that you would see. So there are other sources of variation, and peak sampling, satisfying the Nyquist theorem and getting enough points per peak is one of those. So I think there are other things in there, but it's definitely something we are certainly keeping an eye on and looking for alternative strategies to ameliorate that as we get faster and faster, right? So yeah, does that sort of answer the question? Okay, thank you.
I think we had an online question as well.
Yeah.
Okay, so enjoy the talk. Why have any particles use ploy columns, no back pressure, ASMS presentation?
Okay, so plot columns probably, porous layer, open tubular columns. Yeah, so we have for a plot column to work well, or for a fully open tubular column to work well, this is where you completely get rid of particles and you just rely on sorbent coating the walls. I think that's probably what they're talking about. So we did one kind of proof of concept paper led by Ying Zhu about 5 years ago. But to get effective mass transfer, you need that capillary to be about 2 microns or less. And so it becomes very difficult to do sample loading there. And your flow rate is about 1 nanoliter per minute there. So all the challenges that we have in going to lower and lower flow rate get really severe with that. So it's been shown that the separation themselves can be absolutely beautiful when working with a plot column. It's the sample loading and the electrospraying that haven't totally been solved yet for that.
And then our second online question says, do you believe in 1% FDR for 2-minute gradients?
Do I believe in 1% FDR?
Do you believe in 1% FDR?
We are relying on tools that other wonderful developers have created. So a lot of false discovery rate algorithms don't rely on retention time at all, right? And so their retention time is essentially irrelevant for a lot of these things. So yeah, it's things where none of these stories are completely set, but there are certainly ways you can experimentally test for FDR to complement the readout of your software, right? With multiple species, decoy databases and things like that. So
Right, yeah.
absolutely.
Okay, and then we'll do one more online question. Where do you foresee single-cell proteomics going in 5 years? Will we be able to do all human proteins, and maybe some challenges that you see in those goals?
Yeah, so I think it's already amazing that there are workflows. The Nikolai's group has shown these throughput of 1,000 proteins or cells per day, and I don't think he would say we're at our limits yet at all, right? And then going a little bit slower with label-free single-cell proteomics, we're seeing now reports of 5,000, 6,000. So about half the proteome is now being profiled in single cells, which is just super surprising. Like that first slide I showed of the 125 cells, that was probably like 300 proteins identified from that 125 cells. So it's really remarkable what the mass spec vendor, developers have provided, along with the, pushing the sample prep and the separations and the data analysis. It's like taking all of that to get to where we are. I don't know, sure, maybe there's a way to get to 10,000 proteins per cell in a few years. I think most of us are more concerned about getting to more cells per study, right? So there's a lot of emphasis on throughput, but you want to have your cake and eat it too. And they're both super important. You can't study a protein that is among those that you didn't see, right? So you just can't, you're just blind to those. So if you happen to be interested in protein X, you better have sufficient coverage that gets that one. So yeah, but I don't know. Yeah, we're excited. We haven't seen anything plateau yet, right?
No.
We are here this is the 8th of these meetings. We have not seen anything plateau yet, I don't believe. So thank you.
Thank you. Yeah, lots of exciting things to look forward to. Let's give a hand to Ryan.
Thank you.
All right, our next speaker is Yash Shukla. He recently completed his PhD at UT Austin, and I had the pleasure of hearing a talk from Yash at one of our he gave to our group in a group meeting on a Wednesday, and I was really, really excited by the work he was doing measuring translation in development. Such a kind of important problem for methods that are kind of studying protein synthesis outside of transcription, especially because its transcription is not so active at this point. And so I'm excited for him to tell you all about this technology because I think there's just so much fruitful crossover for people doing proteomics by mass spec.
Hello. Okay, great. Awesome. Thank you so much for that introduction. I will not be talking about any mass spec, but hopefully I can convince you that a C. elegans system is a great system to study proteomics by the end of this talk. So my study looks at the landscape and regulation of mRNA translation during early C. elegans embryogenesis. So all animals start from a universal beginning when I'm showing you a one cell or a zygote of a mouse, fish, and a nematode. These undergo divisions in order to become a two cell and then finally divide to become this multicellular ball. They'll go on to differentiate to become the tissues and organs that you see around you today. However, what really establishes this is this very large egg, which provides these maternally deposited factors that allow early embryogenesis to happen very rapidly. So what I am studying is how maternal mRNAs regulate this early embryogenesis before zygotic genome activation. So a lot of these maternal mRNAs are present early on, and they're carefully regulated to produce protein at the right place and right time. And this happens before this maternal to zygotic switch, where the zygotic genome is activated and produces its own transcripts and proteins and so on and so forth. Now, what we know in many systems of early embryogenesis is that the mRNA amount doesn't quite correlate to the protein abundance. And for a few transcripts, we know that they're translationally regulated to control how much protein is made. But we do not know if this is a general rule or to what extent does this happen. So the question that I'm addressing is to what extent is maternally deposited mRNAs translationally regulated during early embryogenesis? And the model system that we like to use is C. elegans, primarily because it produces an invariant lineage starting from the very first cell division, where you can see in blue that there is an ectoderm that's precursor that is produced from the first division, then the mesendoderm. And by the 8 cell stage, you have all 3 germlines that have been predefined. And the key thing is that these are reliant on maternal mRNA, and transcription is not involved in order for this process to occur. So now you can directly correlate translational regulation to changes that are happening in cell fate as the embryo develops. And the way we go about addressing this is by looking at this metric translational efficiency. And the way we define it is the ribosome occupancy on a given number of RNA abundance. So something that has high number of ribosomes on a given number of mRNA would have high translational efficiency, and something that has fewer ones would have would be low translational efficiency. So in order to do this, we had to measure 2 things, ribosome occupancy and RNA abundance, and that is what we did. So we took embryos at each of these stages, 1, 2, 4, and 8. And to get ribosome footprinting, we obtain these ribosomes intact on the RNA. We apply nucleases to remove all the exposed fragments and get this ribosome protected footprint. And then you can send this 30 nucleotide fragment for sequencing, and that's how you get ribosome occupancy. For total mRNA, you just collect it, and then you send it for sequencing, and you get RNA abundance. Now, for this part, single cell or single embryo RNA-seq has been done before in C. elegans embryos, and it has already been established. However, conventional ribosome profiling would require you to collect at least 10,000 embryos at each of these stages. But in C. elegans, this process happens within a matter of minutes, where they divide in 5 minutes and produce different cells. So it's really hard to actually collect those number of embryos at each of these stages. To get around this, we use this platform called RiboITP, which was previously developed on mouse embryos. This is known as ribosome profiling via isotachophoresis. And so what we do is just collect 9 embryos and then produce these fragments, ribosome protected fragments, and apply it to this microfluidics chip. And apply this to this microfluidics chip that, based off of the ionic mobility, can separate that 30 nucleotide fragment only from a few select embryos. And then you can just go ahead with library prep as usual. And so through this, we were able to move from using 10,000 embryos to all the way to 9. And that's how we got ribosome profiling data set along with RNA-seq. So we did a bunch of QC, and then finally what we want to know is, what does this data actually tell us? So we wanted to know, how does translational efficiency change as the embryo develops from 1 to 2, 2 to 4, and so on and so forth. And so here's the data. To break it down, this is the x-axis is showing you the average count or the abundance of the RNA. The y-axis is showing you the fold change, 2 cell by 1 cell. And in blue, I'm just showing you all the different RNAs, and only the significant ones have been indicated as a point in orange. And as you can see, the RNA abundance doesn't change as much from the 1 to 2 cell stage. However, if you were to look at the translational efficiency, we see these huge changes that are occurring both in up and down regulation as the embryo develops from 1 to 2. A similar pattern was seen from the 2 to 4 cell stage, where we see that there's few changes in mRNA abundance that does occur, but we see these big changes that are occurring in translational efficiency. So this tells us that overall the mRNA content is remaining constant, but it's really the translation that is being regulated of these mRNA species. So the next thing we want to understand is what drives these translational regulation. And I'm going to show you two things. First is the role of RNA binding proteins, and the second is phase separation. And those are the two things in the field that we understand could regulate translational efficiency. So in C. elegans, we have this protein called OMA1 that was previously known to regulate a few transcripts during early embryogenesis, but we did not know the extent to which it regulates it. So from what we understand is that OMA1 would bind to the 3-UTR and prevent ribosomes from binding to it. And as the embryo develops, this protein starts to degrade, and we would then observe translation of these transcripts that were regulated by OMA1. But we wanted to see the extent to which OMA1 actually regulates different transcripts. And the way we went about doing it is that we had this ribo-ITP and RNA-seq data at the wild type 1, 2, and 4. We then applied the same platform on a gain-of-function mutant in which OMA1 does not degrade over time. And then you can compare these at each of these stages to know what role does OMA1 specifically play in terms of translational regulation. What we observed was at the 1-cell stage, we don't see any difference between the wild type and the mutant in terms of translational efficiency, which makes sense because in both cases, OMA1 is not degraded. However, at the 2-cell stage, we see this large decrease in translational efficiency in the case of the mutant. But what's interesting is that in green, I'm showing you previously identified mRNA species from an OMA1 pull-down experiment. But we are observing other mRNAs that were not previously identified in those particular experiments, along with the fact that there's some upregulation that is also happening. So this specific data set of how embryogenesis is being controlled, specifically the 2-cell stage by this RNA binding protein, was addressed through this particular experiment. And at the 4-cell stage, we see a bigger dip in translational efficiency also. Another thing that we want to look at was these phase-separated particles called P granules. So what is thought of in the field is that a lot of these mRNAs would be localized into these granules that I'm showing you in green in the germline lineage, where they would trap these, what we thought was they would trap these mRNAs and prevent them from being translated. What I wanted to assess whether that was the case in our data set. And these granules are not something that's unique to C. elegans. You also see this in Drosophila or even in Xenopus. In order to address this question, we used a prior study in which people had stained 441 transcripts that localized to P granules at various different stages of embryogenesis. And the specific question we wanted to ask was to look at the subset of mRNAs that are localized uniformly at the 2-cell stage and then appear in P granules at the 4-cell stage, whether we see a dip in translational efficiency. So if localization into P granules caused this dip, causes a dip in translational efficiency, we know it's probably because it goes into this phase-separated particles. And here's something that we found that was interesting, was that the dip actually happens one stage before, where the translational efficiency goes down from the 1 to 2-cell stage and remains repressed at the 2, 4, and 8-cell stage. So what this tells us is that first the repression happens, and then they localize into these P granules, rather than going into P granules and causing that repression. And with that, I would like to summarize that what we've identified is translational efficiency remodeling as the embryo develops. We identified different transcripts that are regulated by OMA1. And also this repression in TE seems to be occurring prior to this phase separation. And with that, I'd like to thank my PI and my lab. And also, this paper should be out in bioRxiv. So if you're interested in, and there's more data out there that you'd like to see, you can easily scan this. It'll be in press soon, but yeah, thank you.
So thanks so much. I'll actually start by asking a quick question. So for the sample amount requirements for doing the making measurements, why was previously the amount, I guess, so high? Are you doing like RT-PCR of these 30 nucleotide fragments, or like, can you not just, I guess, say you want to do single cell, is it easy enough to kind of PCR your way to having enough fragments to sequence?
I think the reasoning is also the way you get these samples is you have to do like ultracentrifugation in order to get those particular number of ribosome footprints. And in all that process, there's so much sample loss that by the end of it, you don't have enough to get a good quantitative measurement of ribosome profiling. And so that was predominantly the reason why.
Cool. Thank you. Questions?
Very cool. I think it also reminded me of Andrew's talk with the protein dilution happening. And I was wondering if you see the similar case when you calculate translation efficiency in your ratio. Are you seeing a decrease in those transcripts that increase translation efficiency? Or are you seeing a decrease in the ribosome binding?
Yeah, that would be interesting to see. I guess, unfortunately, the way we're doing this assay is we're collecting the whole embryo in itself instead of the isolated cells from there. But that's something we can do. We can isolate them and collect them and do that. But yeah, that would be interesting to see how dilution could affect the concentration.
Yes. Have you been able to identify other features associated with change in ribosome occupancy, such as sequence motifs on messenger RNAs or microRNAs changing their abundance as another form of regulating translation?
Yeah, so one of the things that we also looked at was look at motifs of previously known RNA binding proteins. And I have a list of them in there, but yes, we identified, at least for the ones where the data was available for this particular motifs, of how much do they correlate with the translational efficiency that we see. We didn't yet do like a motif search to identify which motifs explain higher translational efficiency. That would be sort of a next step that we want to take.
Cool. Well, one other quick question with the RNA binding proteins that seem to affect translation. What causes that change? Because presumably that RNA binding protein was present in the zygote. Do you have thoughts as to what results in the switch?
Yeah, so one of the things that I have two reasons, three that I can think of on top of my head, and for different proteins there will be different contexts. One of the things that happens during this early embryogenesis is there's a lot of asymmetric divisions in which the RNA binding proteins are divided separately. And so depending on which RNA binding protein is present in what stoichiometry, you can define the TE of that particular transcript. There is a lot of phosphorylation and changes that are happening on these particular mRNA binding proteins that regulates the translational efficiency of these different mRNAs. So those are a few that are on the top of my head.
Conceptually possible mechanisms, but it's not necessarily clear for any one particular protein what is driving it.
That is correct. I think that people have not really dissected yet, and I hope that through this data set, people can start really like looking at RNA binding proteins and what are the different species that it regulates together. Yeah.
Thank you.
One additional quick question. Is RNA degradation like muted in some way at that 1 to kind of 2 cell stage, since you kind of need those early deposited, but since you showed those plots showing that like the actual mRNA abundance does not change substantially from like the 1 to the 2 cell stage. And so presumably, I guess if they were kind of being degraded or being degraded at different rates, you would kind of naturally see maybe a lower, because since there's no transcription happening, a decreased abundance of mRNAs that degrade easier. So do you know if maybe this process is not happening in addition to lack of transcription?
Yeah, from all the other data sets that I've looked at have done RNA-seq, the RNA amount stays fairly constant from 1 to 4 cell stage. And it's really after that a lot of degradation starts happening in different cell types. So I guess from the resolution that I have, it seems like it stays fairly stable.
Thanks. All right. There are no more questions. Sorry. Oh, there's one online. Another localization. Wow. Thanks. Yeah, someone is asking about the localization effects other than P granules. So outside of that.
Yes, apart from P granules, we looked at transcripts that are localized on the periphery of the cell, and all of them have a higher translational efficiency relative to the average. And also in terms of localization, we've also looked at transcripts that are localized in the somatic lineage versus the germline lineage. And we observed that in the germline lineage, they seem to be repressed compared to the somatic lineage. So those are the two other localization things that we saw apart from P granules.
Well, in the periphery, do you mean like near the cell surface?
Like on the cell cortex, yes.
Do you think that there are RNA binding proteins that are like somehow also transmembrane or localized to the surface in some way, or maybe interact with things on the surface that could
for some of the transcripts that people have previously studied, what ends up happening is they get partially translated, resulting in a domain, like a domain that binds to the membrane, and then they finish their translation on the membrane in itself. So that's where I think that active translation is happening for them.
Okay, cool.
Yeah.
Thank you. Interesting. All right, let's give another round of applause for
Thank you.
Ashley.
Hey, I'm happy to introduce Dr. Jared here. Jared got his PhD in Indiana University in 2017, and since then, he started with a postdoc at Northwestern University at the Proteomics Center of Excellence with Neil Kelleher, where he joined the ranks of top-down mass spectrometrists. He also was one of the pioneers in individual ion mass spectrometry, so not just single cells, but single ions in Orbitraps. And today he'll be talking to us about some proteoform imaging mass spectrometry. Please welcome Jared.
I had uploaded my talk to the one computer. Don't know where that one went. Let's go. Oh, there we are. Great. Nope.
Right here. Oh, you just have that.
Sure, thank you. Thank you. All right, thank you for the wonderful introduction. Today I'll be talking about probing the proteome of differentiating human skin keratinocytes using a relatively new technique within the Kelleher Research Group, single nuclei proteoform imaging. Before I jump into applications, I'd first like to talk about the underlying technology that allows these measurements to take place. As we all know, mass spectrometry doesn't actually measure the mass of our molecules, right? It measures the mass to charge ratio. When we think of an Orbitrap analyzer, as an ion oscillates around the central electrode, the frequency through a Fourier transform is related to the mass to charge ratio. Now, there's a subsection of mass spectrometry known as charge detection mass spectrometry. It's been around for a few decades, and it uses individual ions rather than an ensemble of ions to make a measurement. So we'll have singular ions that oscillate around the central electrode. Once again, we'll determine its m/z through its frequency measurement. However, now we can look for these individual ions, the rate of integrated signal on the outer electrode of this Orbitrap analyzer. And we find that the signal induced is proportional to the charge of the ion. So now we can accurately measure not only the mass to charge ratio, but the charge of each individual ion, giving us its exact true mass. So you might be thinking, why is this important? If we have a complex sample, such as Kexel lysate, we don't do any separations with it. We do gel-free fractionation, though. We take the 0 to 25 kilodalton fraction, shoot into the mass spectrometer. We expect there to be thousands of proteoforms. On top of this, we get splitting of charge states here over the m/z domain, and we get a really, really complex spectrum. It's so complex, we actually do not see any peaks. However, if we start to consolidate these various charge state distributions into one mass distribution, we go from no data to this very rich now mass spectrum. If you look at the x-axis down here, we're in the mass domain, kilodaltons. We're able to identify through a mass tag approach about 550 proteoforms with these really nice isotopic distributions that are able to match to theoretical distributions. So we'll now couple this technology with the differentiation of skin keratinocytes. We have a really wonderful collaborator in the dermatology department, Northwestern University, Xiaomin Bao. What she'll do is take discarded human skin tissue, isolate primary epidermal cells. I'll find that when she plates this, only the undifferentiated or progenitor states stick to the plate. And then to induce differentiation, it's kind of what we'd see here through the epidermis up until finally the cells are then shedded. And she'll do this with calcium chloride over the course of 4 days. We'll take these two different states, break the cells open, isolate the nuclei from the keratinocytes, and then we'll drop cast them on positively charged slides. Here's just a small picture of what a few of these single nuclei look like. They're about 5 microns in size. This varies from what the keratinocyte cells are, about 10 to 12 microns themselves. We'll then subject this to something we've been doing over the past few years, single cell proteoform imaging mass spectrometry, or SCPIMS. We'll use a nanodesi based approach pioneered by the Julia Laskin lab. We'll have a denatured solvent running through this first capillary here. It'll form a liquid-liquid bridge with the slide. As we raster that across the surface, it'll extract out proteoforms from the nuclei, or just depictions of a cell, into the secondary capillary. And then it'll ionize that into the mass spectrometer where we'll perform our individual ion analysis. And we have 3 different stages here. Before, this is the nanodesi probe, and this is a picture of the cell. We'll be doing this with nuclei though. Where we have before, on, and then after sampling. And we can see in the chromatogram here, we have a little bit of background before and after. However, we start extracting out these individual ions here when we're on top of the cell. We like to call this a cellogram. So to gain enough statistics for our various states, we'll sample 17,000 single nuclei. Half of this is in the undifferentiated state, and then the other half is in the differentiated state. Here's just a subsection of what these extraction profiles look like. Each one of these ticks is the sampling of a singular nuclei. So we have sampled 65, throughput of about 65 nuclei per hour. So it takes us about 11 days to collect these 17,000 data points. We find that for each one of these extraction events, we get about 6,500 individual ion signals they're able to use, and this corresponds to an isotope of an intact proteoform. Now, there's a lot we can do with this data. The first thing we'll do, though, is take everything aggregated together to be able to see what we have present. We are using intact mass tag approach. So we find that with across these 17,000 single nuclei, we'll have greater than 75 million charge-assigned ions here in this butterfly plot that we'll make. Undifferentiated is in red, differentiated is in black. And using our intact mass tag approach, we've previously taken a created a top-down database using the same sample set. And we have 265 high-confidence proteoforms we're able to determine through isotopic matching here. Here's just a few of these insets. You can see what some of these distributions look like. We get a lot of ribosomal binding proteins, a lot of histones, of course. And here's the H4 region, acetylations, methylations. Here's just another inset here of various proteoform landscapes of H2B and H2A. So now that we've determined kind of what we have within the entire data set, we can start breaking it back down, the puzzle back down into its different components. Shown here is just one of the nuclei that we sampled, number 1035. You can see what those 6,500 ion signals start to look like here in the mass domain for a singular nucleus that was sampled. We can start to see then how many ions go into each proteoform distribution. And then we also have a scoring system that we've invented here, proteoform assignment scoring, we call it PAS score. And this looks at, there's a few examples of what some of these various proteoforms look like for this single nucleus that we've sampled here. Do we get the relative abundance that we'd expect toward the center of this Gaussian isotopic distribution? And what's the accuracy of these individual ions to the actual individual isotopes of our theoretical distributions? That all goes into the scoring. Here on the left-hand side, you can see a rather high score. It scales from about 1, which is 100% confidence, down to 0, which is 0% confidence. You can see we populate for our single cell, our single nucleus here in black versus the theoretical distribution in purple. Here we populated pretty well. We get our nice Gaussian, almost like distribution, and our mass accuracy is really well, so we have a higher proteoform assignment score. And then on the right-hand side, you can see we populate more of the periphery here rather than the center, as we'd expect. And some of the accuracy is slightly off, so it has a lower proteoform assignment score. So we do this for all the proteoforms that we have for each nuclei. Then we could take the data set itself, and we've started sequestering it to clustering. So we know what proteoforms go into each nuclei that we've sampled, and then performing clustering, we find in this example we have 5 distinct clusters we've determined. We find that these clusters are populated by both differentiated and undifferentiated nuclei, and so some of these clusters have about a 50-50 mix. Some of them are more populated by our differentiated nuclei, so for cluster 1, and then cluster 2, some of them are more populated by our undifferentiated nuclei that we've seen. And here it just breaks it down to components underneath. Now, there's a lot of data here, and to try to get an idea about what we should start to target, we did run a bottom-up histone panel to see what is upregulated and differentiated here in blue, and then upregulated in undifferentiated here in yellow. And just for the sake of time, I'll show just one example of what we've been able to determine. So we'll look at the H4K20 monomethyl. We can look at how this is expressed within these various clusters. And we find that here for cluster 4, for the monomethyl, it is highly expressed here in cluster 4 and lowly expressed here in cluster 2 and 3. And then if we move to the dimethyl, we find that it is more lowly expressed now here in cluster 4, higher expressed in cluster 0, 1, and 3, and then more lowly expressed still in cluster 2. We can look at the ions, for these specific nuclei and start to plot this back into their mass spectra to see how it corresponds with the plots on the right-hand side. And we find when we compare cluster 4 and cluster 3 for histone H4, the monomethyl is decreased as you go from cluster 4 to 3, as we can see on the right-hand side, but it's increased in relative abundance here in cluster 3 for the dimethyl. Similarly, we can do this once again for cluster 4 and 2, where we find that both the monomethyl as well as the dimethyl are both decreased in relative abundance here for cluster 2. We can also go back and look then at our bottom-up histone assay, see how this relates to some of the abundance that we find there. So the monomethyl here, we find for our undifferentiated is more highly expressed. And then for our dimethyl, we find that although it's significantly more closer, differentiated is highly expressed, which we find that, and there's been some papers on this, that the monomethyl itself is corresponding with cell growth. And then as you move to the dimethyl and trimethyl, you get more gene silencing. So with that, I'd like to thank Northwestern Proteomics, of course, Neil Kelleher. We have a lot of postdocs, biostats folks that have started looking at this. This is kind of our first soiree into clustering and moving toward this style of analysis. So we still have a lot to learn. Thermo Fisher Scientific for all the individual ion work and development. A lot of our first single cell platform development was done in conjunction with the Sweedler group. And then, of course, for our skin sample preparation, Jamin Bao. So, all right, thank you. I'd be happy to take any questions.
Thank you very much for a great introduction to our single cell imaging. I'm curious, so of course histones are very abundant in most of these cells. Have you guys looked at different solvent mechanisms for potentially biasing to different parts of the cell, like, specifically membrane ones, more hydrophobic, or detergents in your solvents to kind of extract that information?
Yeah, so that's a wonderful question. We've really started thinking about coupling, and I know other groups have done this too, kind of the single cell and then single nuclei based sampling to kind of increase proteome coverage. Our depth is still not that deep. I mean, we're 265 proteoforms deep. We're not in the thousands. But yeah, we found that, changing, even denaturing solvent, adding more acid, you can bias different histones and modifications. So yeah, there's a lot to explore there. We just haven't done it yet. Yeah. Mm-hmm.
Any other questions?
If you manage to fish out a goldfish out of a pond, What would you ask it to give you in terms of improving the power of single ion imaging on the side of sample prep, instrumentation, data analytics? What would be your first wishes that the goldfish can grant you?
So good. So you're talking about next steps then? Or
That's right. Next steps and sort of thinking really big picture, not being too concerned about resource. Let's say that Thermo and Bruker are willing to devote all of their R&D budget on the instrumentation side.
Right.
A little bit giving you the opportunity outside of practical constraints. What do you think are the biggest opportunities, promises that you would like to see realized?
Yeah, well, I mean, one of the biggest limitations that we have, of course, are databases that we use and matching. So intact mass tag approach, there's only so much you can do. We try to boost all of our identifications with taking the SAM data type and running top-down analysis or using previously determined bottom-up data sets, but having enough sensitivity to be able to do tandem mass spectrometry or something like that to actually have the sample and then try to analyze what exactly those proteoforms would be. Amazing, right, from what we do. So that's a sensitivity, and we just don't have enough ion counts to be able to do fragmentation, correspond that back. But I mean, that would be the dream, right?
Thank you.
Thank you. So in your 500 to 600 protoforms that you see in nuclei, how large those protoforms can be? Are you limited in the molecular mass of those protoforms? And if you funnel them down to proteins from protoforms, then what is the representation of the proteome? 100 proteins, 50 proteins? What? Give us, please, more detail and sense.
Yeah, so for a lot of what we see, I mean, I showed a few examples. I mean, histones are highly modified, right? So the amount of proteins that we see, I mean, are probably, especially with this, less than 100. With cells, rather than just nuclei, it's higher. Sometimes we see just one proteoform per protein, depending. But yeah, as for the limit itself, I mean, we've shown using this style of analysis in a joint paper with Julia Laskin that we can go up to 70-ish kilodaltons. So we're a little bit higher. I know sometimes with the LCMS, top down, we're limited to maybe 30, 35 kilodaltons. So we can start to access a little bit higher. As we take these charge states and start to piece them together into one mass distribution, it really increases the signal to noise. So we can sample higher into that proteome. As for the depth, we just need to get, we usually have lower injection times for our analysis event, and we just need to be able to make it so that we can start sampling some of the more lower abundant proteoforms. We just aren't there yet. Yeah.
So what is your explanation for this limitation by molecular mass? So is it ion suppression? Is it like channeling into multiple charge states? How do you explain it?
Yep. So absolutely. So right, the larger it is, the more isotopic channel and charge state channels it has to fill. So for us to even vary that, you probably need a higher concentration to be able to see that, right? It's just too down in the weeds. The concentration that there is in the single cell just probably isn't high enough.
Can it be also ionization suppression when ionizable histones suppress everything else?
Yep. Yeah, absolutely.
Thank you.
So I think I saw on one of your slides that you estimate about 6,500 ions per nuclei is what you're observing. What is causing a limitation to that number, and how could it go up by like 100-fold? Because there's presumably more than 6,000 proteins in the nucleus.
Yeah, so especially for some of our top-down work, we're very sensitive to adduct salts, and that will also lead to ion suppression. So we've started increasing washing protocols and working through a lot of that. So we just need to get better at cleaning our sample, funneling all the ions directly into the mass spectrometer. It's probably just a transfer is what it is. It's just low abundance to begin with, and to transfer in and getting the ions out and up and in. It's just where we're currently limited. Yeah, there should be significantly more. Right, of course, proteins, proteoforms that are present. Right, in all the other talks we've seen, there's thousands. Right.
Great. Well, let's thank Jared again.
Good afternoon. It's my pleasure to introduce Dr. Nesvishki. Dr. Nesvishki serves both as Godfrey Doris-Stobie Professor of Bioinformatics and as Director of Proteomics Resource Facility at the University of Michigan, Ann Arbor. He is founder and CEO of Pragmatics, which leads efforts to commercialize computational algorithms for mass spectrometry data analysis. He also serves as senior editor in bioinformatics and biostatistics for the Journal of Proteomics, sits on the editorial board of Molecular and Cellular Proteomics, and is a member of the Scientific Advisory Board at the Swiss Institute of Bioinformatics. Please join me in welcoming Dr. Nesvishki.
Well, thank you, Nikolai. I think all the organizers for inviting me to come here. It's my second time at this conference. I have to put some disclosures required for me to put here related to conflict of interest and financial interest related to licensing of our tools, MS-Fragger, IronQuant, DiaTracer through our company, Pragmatics, as well as other relationships here. So FragPipe is really a great computational platform because you can run it on the cloud, you can run it on a cluster, on a desktop. We have about 14 to 15,000 downloads for each version. We just released a new version, FragPipe 23. And it's free to academics, and as I already mentioned, it's commercially available to industrial partners. So before I get into single-cell proteomics, I give a little bit of a brief introduction to the tools themselves. But why, I mean, one advantage of FragPipe is that we have this system of workflows where you could basically load the workflow of interest, whether it's a DIA with spectral library building, direct DIA spectral library building at quantification, or it's a TMT or SciLac, chemoproteomics, immunopeptidomics. Just load the workflow and everything is going to be set for you. You just need to load the workflow, the data, the database, and click run. And what I'll talk today about some of the workflows that involve single-cell proteomics data, although the same workflows that we use for single-cell, you would use for other data sets as well. So we don't have actually single-cell specific workflows because we found that most of the strategies are actually pretty much the same as for bulk proteomics data analysis, as you'll see from my presentation. We also have a FragPipe Analyst, a downstream tool that's super convenient for you to quickly visualize results of your quantitative workflow, again, whether it's a DIA or MS1 with match between runs or TMT workflow. You can quickly visualize volcano plots, PCA plots, do enrichment analysis for experiments that involve differential expression analysis. Perhaps not for single cell so much, where you use dedicated tools like Sam is developing here, but for a lot of standard differential expression analysis workflows, this is a very, very powerful tool. So some of the updates that are relevant to this presentation in FragPipe is that we have a new tools for DIA data analysis, new DDA plus tool for chimeric peptide identification from MS spectrum. We have deep learning-based rescoring. We have visualization tools, like I said, in addition to FragPipe Analyst, we integrate with Skyline, MSStats, and we have a lot of new PTM workflows and glycoproteomics workflows and so on and so forth. So a lot of different things, but I will get now more specifically to things that are related to single-cell proteomics. I mean, I actually like really the single-cell proteomics community because it's really pushing the envelope and helps us computational scientists to develop new tools and new strategies. So even if at the end, the way how we analyze the data for single cell is not necessarily that much different for analyzing bulk proteomics data, it's actually pushes us to optimize the workflows because everything that we optimize for single cell benefits pretty much every user. So we need methods that work with single cell as well as with bulk proteomics data sets, ideally through the same workflow, the same pipeline. And we have to pay extra attention to things such as match between runs and false discovery rate analysis related to match between runs, especially when you use bulk data or 100-cell data to build the libraries boosting runs and then transfer the identifications to single cell runs, and some of the additional challenges with how we do deep learning-based rescoring, or some of the new TMT strategies that people explored for single-cell data. So things like this really pushed us to develop new things in a pipeline that, again, at the end benefit pretty much everyone using the tools, not only single-cell community. So, for example, one thing that we were really pushing hard is this development from MS Booster rescoring tool that utilizes deep learning-based predictions. So what's shown on this slide here is a typical workflow where you would identify peptides from MS spectra using a search engine. In our case, it's MS Fragger. And then it would go to percolator for rescoring, and then downstream protein inference analysis and so on. But if you can predict retention times and spectra using deep learning-based predictions and use those predictions in scoring, you can significantly boost your identification rates in challenging data sets, right? In the regular trypsin digest data sets, you're not really going to gain anything. But for challenging data sets, and we thought that single cell would actually be one of those data sets, and indeed, this is where it helps, that if you include deep learning-based predictions and incorporate them in scoring, that would improve the number of identifications. And MS Booster is our tool that's doing exactly that. And again, single cell analysis was one of the motivations for developing these tools, actually. So MS Fragger, MS Booster basically will predict, as I said, retention times, spectra, and ion mobility values if it's a timsTOF instrument, and then would add additional scores based on how concordant the predicted retention time and an empirical one, how well they agree. So a peptide with a big difference would be penalized, and the same for the spectral similarity. Initially, in the early days, we thought that actually we would have to retrain the deep learning model specifically of single-cell data. And Sam had a paper that I think alluded to that point, that single-cell spectra would be would look different from bulk proteomics data. So kind of now it's really depends on the data set. I would say that we see some single-cell proteomics data sets where conventional predictions work well, and we see some other data sets where spectra look very different than what you predict using existing predictors trained on bulk data. So in general, we do not, for example, on our tools now use any single cell specific sort of deep learning model for predicting spectral retention times. And still we get 5 to 10, sometimes 15% increase in the number of identifications by utilizing predictions when applied to single cell data. So here is, for example, one data set where if you look at the spectral similarity between the predicted and experimental spectrum for the single cell run versus 5 cell versus bulk cell, you see that the spectral similarity is pretty much the same, even for single cell data as for bulk data. But as I said, in other data sets, you'll see that the similarity between the single cell run and the predicted spectra would be much worse than for bulk data versus predicted spectra. So it really depends on the data set. So, but to make it more robust, because we don't want to be sort of at the mercy of how the spectra look like or retention times in a particular data set, to benefit no matter what, we also work on creating a system where you could actually utilize multiple different predictors. And we worked with Matthias Wilhelm lab on this Koina server. Koina is a server that hosts multiple deep learning models for retention time spectral predictions from PROSIT, from AlphaPepDeep, from several other tools. And the idea of Koina is that in your pipeline, in our case, it's FragPipe with MS Booster, you could send peptide identifications. You can send peptide sequences and spectra to this Koina prediction server, and basically try every model there. And we automated in FragPipe in MS Booster, so we send a little bit of data, not the whole data set, and test all models that's available on Koina. And then we find the one that for the particular data set we're analyzing gives the best similarity and the best retention time agreement. And then for the full analysis on all data, then we are using that best model. So we basically created this easy to use infrastructure for finding the best prediction model. So even if one predictor doesn't work well on a particular data set, probably another predictor from another tool would work better. So that improves the robustness. This being said, again, I don't think there is any predictor that was trained specifically on single cell data. But I find that with this Coinage server now, especially in multiple models, we actually get robust results even for single cell data sets, no matter what the data set is. So it improves the number of IDs for single cell data, but it also improves in other applications, for example, HLA peptidomics, and we do ton of sort of collaborations in this HLA immunopeptidomic space, and their predictions are also very useful. So basically, any type of experiment where there is a large gray zone sort of between true and false identifications, a large source space or challenging data, this type of predictions have the most impact. And the regular trypsin digest, it's minimal. So quantification, and then we again worked a lot over the last 5 years on developing tools for quantifying peptides with the best accuracy and precision possible, whether it's a MS1 or a label-free MS1 with SILAC or TMT-based workflow or DIA. So we developed a tool called IonQuant for MS1-based quantification with match between runs. And actually adding match between runs in IonQuant was one of the motivations is single cell proteomics data. Because when you build, and it's the same with DIA, and Vadim actually adopted a lot of, I collaborated a lot with Vadim Demichev, and he also adopted a lot of the concepts in match between runs for DIA there. So the concept is that you can analyze your single cell data along with your boosting runs, library runs, right? Maybe 10 cells, 100 cells, if not even bulk data, which is not ideal for sure. So you have these boosting runs. So you identify a lot of peptides from those boosting runs because it's a lot more starting material. And then you build a library that has a lot of peptides. And then you go back to single cell runs and you look for those peptides, right? Now, so whether you do it with MS1, with match between runs, or you do it in DIA, where match between runs is sort of, inherently built in the tools, it's the same challenge because you can FDR control the library well, but you go back to each run and you're transferring still a lot more IDs from your sort of bigger library to each individual run, and you have to control the error rates at the run-specific level. So for MS1, we developed this match between runs in IonQuant, and this is part of these papers here that we described. And I'm not going to talk too much about MS1 now at the moment, except I'll say that in FragPipe 23 that we released just a few weeks ago, we actually made a lot of improvements in IonQuant as far as the quantification accuracy and precision of MS1-based quantification. So if you download the latest version of our tools, the IonQuant and match between run and MS1 data is significantly improved compared to FragPipe 22 in previous versions. Now, again, as I mentioned at the beginning of my presentation, why I like single-cell proteomics communities, because people are trying all sort of different things, right? Trying to find improvements, and we work a lot with Ingju, and he's a great collaborator, and he's trying a lot of stuff, as many of you here. And so, one paper that he approached us was on the TMT single-cell proteomics analysis that came out a few years ago now, where we all know how you do TMT with single-cell proteomics in that scenario, but what they were trying to do is to see if they can do everything in the linear ion trap and not using orbit trap. So to speed things up, to increase the throughput, sort of generate more data and get more IDs. And so we worked with him to support basically quantification on the linear ion trap data, basically low-res quantification in our tools. And in FragPipe 23, we also work, we basically added support for Astral TMT data, as well as TeamStuff, as well as TMT35 plex data. And I can tell you that one motivation for TMT35 plex, again, we have a collaboration with people using TMT35. And I'm super excited about Astral with TMT35, because that, I think, is going to be a super powerful strategy, including, hopefully, for single-cell proteomics data. So anyways, our tools now supporting Astral and supporting 35 plex data. Then others here, so Ryan was Kelly was working on at the time, I guess, no longer interested in wide window acquisition MS2 for single cell analysis. But we thought, okay, well, if people are doing that, how do we develop our MS-Fragger search engine that can most effectively, in the very, fast search MS2 data for wide window acquisition data, right? 4, 5, 10, maybe 20 Dalton window, but acquired as MS2 data. So not DIA, but DDA MS2. So Chimeris was capable of processing the data, but was extremely slow. So this work we published a few months ago on DDA Plus, where sort of single cell focus kind of faded in a sense because the field transitioned to other data acquisition strategies for single cell. But in applications where DDA MS2 is still useful, and there are plenty of applications, actually we found that people really love the ability of MS-Fragger DDA Plus to search MS2 data as a full window acquisition search, right? So identifying all chimeric peptides in that data acquisition window. That comes from our open search strategy that we enabled through the indexing in MS-Fragger. So we basically treat DDA data as if it was like a basically open search, in say 2 Dalton window. So anyways, we developed this full-fledged sort of DDA plus mode of MS-Fragger. And even though probably you don't see it applied now to single-cell proteomics data, as I said, there was a motivation, but now we have other uses for this strategy, and I think it's very successful. So here's a preprint of that work. And then on this slide here, we demonstrate that we are identifying a good number of identifications through this full isolation window search. And it's orders of magnitude faster than Chimeris, which was an alternative tool for processing this data. So another point I want to make, again, this is a work from Inju, who is at Genentech now, but they want to do a benchmarking study related to spatial proteomics analysis, where they looked at tissues and cutting the pixels out of those images, but they wanted to compare basically label frequentification and TMT strategies for TMT, MS2 based and MS3 based and various combinations. But how do you really compare different strategies if you use different tools? So actually FragPipe again was the only tool where we could do everything consistently, right? Everything would go through the same strategy, including percolator and full discovery rate scoring, whether it's a LFQ or even DIA. So I think the advantage of our pipeline is one advantage is that it allows you consistently compare different strategies that you may be applying as part of your optimization protocols or when you're deciding on what strategy to use. So as part of this work, basically, they benchmark different strategies and again, label free with MS1 based quantification versus different TMT strategies. And FragPipe provided a comprehensive data processing and analysis, but most important for this, I think, application was that it was enabling consistent comparison between different data sets, right, between different strategies. So, if you're interested, just to summarize what they've done, is that, and that's an interesting paper because the conclusions are, of course, basically the main conclusion is that, the best method depends on what you're trying to achieve, but the label frequentification actually with MS1 based and match between runs actually offered a deep protein coverage, 3,000 to 4,000 proteins. Again, this is not a single cell, this is spatial proteomics analysis, but it had a really great dynamic range, and it was really great for quantifying specific markers. Of course, at the expense of a limited throughput, relatively limited throughput. On the other hand, TMT, MS2, and MS3 strategies were much higher throughput, but they had, a little bit more challenging as far as the quantification accuracy. And MS3 approach didn't significantly improve over MS2 due to lower sensitivity, but provided some other complementary information. So I think the conclusion is that, all of these techniques are complementary, and all of them have a role in the single cell proteomics study. But I think my point here is that by having consistent tools that we develop as part of this collaboration and previous collaborations I mentioned for single cell proteomics data, we have a we have basically a toolbox that allows us to process data from any strategy in a consistent way. So the sort of this benchmarking becomes meaningful, and hopefully the results are good, and you would adopt them for your single cell proteomics workflows. Now, as far as the DIA, data independent acquisition analysis, so we have a lot of things that we worked in the DIA area. So as you know, we were the first group that developed direct DIA strategy, and then sort of when everyone was thinking that DIA is a targeted proteomics workflow that requires spectral library building from DDA data and so on. So we had a tool called DIA Umpire, and now we support all major workflows in FragPipe, and we still have our DIA Umpire for certain applications. We still use it for immunopeptidomics data on Orbitraps, for example. That's our HLA non-specific DIA workflow that's used DIA Umpire. For Orbitrap data, including Astral narrow window DIA, we have a new tool called MSFragger DIA that does direct search of the data, followed by targeted sort of peptide-centric rescoring, all part of MSFragger DIA. But for dia-passive data, we also have now sort of the next generation DIA Umpire called dia Tracer. So we have a tool for dia-passive data, and I'll show you one example in the context of, again, spatial proteomics data, how we can use this tool to analyze the data very effectively. But no matter what strategy you use, again, whether it's MSFragger DIA, DIA Umpire, or just DDA data, what the advantage of our strategy is that everything goes through the consistent full discovery rate analysis. Everything will go through MS Booster for deep learning-based rescoring, followed by percolator and Q-value estimation, target decoy strategy. So everything becomes really comparable. It's not apples to oranges, right? Because everything goes through the same well-established workflows where, if you maybe choose some other tools to use, it's a black box, right? That may give you, 12,000 proteins in 5-minute runs, right? And of course, it's exciting, but is it real or not? It's a good subject for a discussion. So with all these workflows, we basically would build high-quality spectral libraries, and then we would pass those libraries for quantification, extraction of quantification, either to Skyline or Diane or Alpha DIA, another great tool that you'll hear later in today's program. So, and one difference between, for those of you who are into DIA stuff, between how we approach things and the pure peptide-centric scoring is that the question is, when do you start using deep learning-based predictions? In peptide-centric strategy, you basically say, oh, I can predict retention times at the beginning for the whole database. And I'm going to go to my data, and very, very complex data, with a very, very narrow retention time window. And only going to look on that retention time window and find something, and then kind of quickly generate a decoy and test there. And that's really a dangerous strategy, because you're not looking for modified peptides. You're not looking for other artifacts that are in the data. You just look for a particular unmodified tryptic peptide in a narrow retention time window that the software predicted for you. So that has a problem with the sort of FDR estimation. But also it's a very time consuming to do predictions for everything at the beginning if you do immunopeptidomics or PTMs and so on. So in our strategy, we do MSFragger sort of direct DIA search first, or DIA tracer for MSFragger. And then we do predictions only for top scoring peptides. And if the prediction is not good, so maybe the retention time is totally off because it's a modified peptides and so on, that's okay. It's just prediction's not going to help us a lot in rescoring, but they're not going to penalize us too much, right? The bad prediction not going to penalize us and not going to create false positives, where if you kind of rely on the predictions at the beginning, that has an issue. And, there are ways to deal with this, transfer learning and so on, but, I'm just giving you sort of, trying to give you a bigger picture here to understand all the complexities of different strategies for analyzing the data. So the sort of spectrum-centric first strategy that we are using in our tools has a lot of advantages when it comes to large sort spaces, PTMs, any other experiments where it's hard to predict peptides for the whole proteome accurately. So coming back to timsTOF data, I mean, this works really just beautifully how we did this, I think, because again, we take the dia-passive data and we convert it to a DDA-like data in DML files, right? We basically detect the precursors, detect fragments, correlate them, generate pseudo-mass spectrum, precursor fragment groups. Of course, it's all about signal processing, how you do it. It's really a lot of work went, actually years of work went into optimizing this just for ion mobility data, not to mention the early DIU work. But at the end of the day, when it works now is that you generate DDA-like data from your diapass of that data. And that data can be searched against using MassFrag or any other search tool. For PTMs, it can be searched in a regular way with PTMs and triptych, semi-triptych search, something again that's hard to do with other tools. And then you can, once you build the library, you can pass it to Diane, or the library when compatible with Spectronaut, or again, Skyline or Alpha DIA. So you build a library, and then any tool can extract quantification from the original DIA files, from the single-shot DIA files using that library. And you can build a hybrid library as well. If you have DDA data or GPF DIA data, you can build a hybrid library. So in this particular example, we applied DIA tracer and FracPipe computational platform to allow input spatial proteomics data from the study listed here on this slide. And it's 148 microregions from 4 areas of human tonsil, right? And as part of the original work, they generated high input library containing 46 runs to generate high input library from a lot more starting material, and then use that library to do quantification from this single shot, single microregion data. So we demonstrated in our DIA tracer paper that came out a few months ago, is that we can basically, so yes, if we just look at the total number of proteins quantified, we would get less from a direct DIA method, only using low input data, compared to using the high input library. However, if you filter for missing values, basically saying that you quantify your data with a certain degree of completeness, for example, at 80 or 90% of completeness, the results are very, very similar already. And of course, as I said, the best strategy would be to build a hybrid library. So, if you do have high input data, and you have your single shot, low input, single cell, or like in this case, sort of the microregion data, why not build a hybrid library? Direct DIA coupled with in this case, the high input data was also DIA. So it's all direct DIA, but high input plus low input hybrid library. And then you do it all in one analysis with well-aligned retention times between all runs, very good high quality filtered library. So that, in our view, gives us the best results. So we were able to replicate the results of the original study here using DIA tracer and FracPipe computational platform on this timsTOF data set. And again, with just a few clicks, you can visualize the data using FracPipe Analyst as far as the differential expression analysis between different type of regions in the human tonsil. And thixial cluster, this is a PCA plot, and a volcano plot, and the known markers that differentiate between those different regions. All of this you could visualize and quickly validate just using direct DIA approach, only low input data, coupled with DIA tracer, MassFragger, a FracPipe, and then FracPipe Analyst at the end for visualization and differential expression analysis. Similarly, of course, we applied this also on astral data. It works just fine. Actually, for astral data, it works really well. It works well for all workflows, but for astral, because it's a narrow window, it's really a great match to how we approach things. Except that, again, this is from Jasper Olsen lab, just some single cell proteomics data from the astral data. In this case, they used 20 cell boosting runs. And if you compare different tools, in this case, Spectronaut, Diane Library Free, or FracPipe with Diane, but only for quantification, you'll see that the numbers vary than the total number of IDs in the matrix file. But it's very misleading what each tool reports, because you have to look at how the data is filtered. And if you look at the number of proteins quantified without missing values, or with just a few missing values, a low percentage of missing values, you'll see that in our pipeline, we actually get a really good results. Because we control, again, the error rates in a run specific way very rigorously. And when you use these tools, just an advice for you to actually check how the data was filtered. For example, if you open the Spectronaut file, you will probably see that the Spectronaut results are filtered to 5% FDR in a run specific way, right? Which is completely unacceptable for single cell data, because you take the data built from 20 cells, right, in this case, transfer it to single cell data, but apply only 5% FDR for the run specific, right? So that's just wrong, right? So you always have to make sure that you consistently filter the data if you're comparing different tools. Just something that I always try to tell people. Not often people listen, but I keep saying that. So And again, I mean, this is for a discussion, but the need of stringent filters, especially on ultras or astrals type of data, where if you actually, because we integrated everything with Skyline, that's beautiful because you could run FracPipe, just say run Skyline, it will generate Skyline document file, you can open it, and you'll see all these traces. And you'll get a heart attack if you look at this ultra or astral data, because you'll see that a lot of IDs have like one fragment here, one fragment there, they never coalute, and somehow it passes to Q values, right? So in our case, we prevent this from happening or try to minimize this type of magnifications by requiring that you identify a peptide by at least 4 or 5 fragments in the same MS2 scan, right? So we basically require that. And we also require by default that we detect MS1 signal. So we don't report, and you can then check it, but by default, you only will see IDs where you also found MS1, right? So this is we on purpose would rather report less IDs, but know that it's at least we detect the precursor, so we know it's not unmodified and modified peptide that somehow co-fragmented in the same window, and we're getting the shared fragments misidentified, and also making sure that at least there is some coelution of fragments, right? So I think that's important to think about when you do these comparisons, and if you start comparing the numbers, it gets into these listomics discussions, and I think we, as a community, especially a single-cell community, where there is still a race to show the highest numbers, as high number as possible, that we pay attention to how the data is filtered, and we make comparisons between different tools in a consistent way. And how do those comparisons, I don't know, the CVs are not really good metrics, and I'm not a big fan of multi-organism data sets, but I don't have a better suggestions to provide here, or significantly better. Now, so our strategy is to make sure that we provide visualization tools also to inspect the data. I already mentioned that we can build a Skyline document file, so that just you can generate them, and then you manually can inspect the data at least. And if you're into PTMs, and we work a lot with PTMs like glycoproteomics, so we want to resolve between different glycoforms. So that's really great because you could actually visualize the data in different isomers and so on. Maybe less of interest for single cell proteomics data at this point, all this glycoproteomics and stuff. But maybe it is, because I'm hoping that as the technology improves, single cell field will also go in the direction of PTM analysis and chemoproteomics and all this cool and exciting stuff that actually requires advanced searches of the data, looking for labile PTMs, unknown PTMs, and so on. Because this is, for me, for my lab, is of great interest, right? We work on open searching and all these methods for a long time as a way to, MS Fragger for me is like a dark matter telescope to really study all these unmodified peptides in this data. And with DiaTracer, for example, coming back to what I talked about a few minutes ago, you can generate MZML files, DDA-like files, and search them for PTMs in unconstrained way using MS Fragger Open Search. That's one advantage of using DiaTracer, MS Fragger-based pipeline and FragPipe for DiaPassive data. So in summary, what I hope to tell you in this presentation is that FragPipe is a comprehensive proteomics platform that supports all sort of workflows, different quantification workflows, and many biological applications. And today, of course, I focus on single-cell proteomics data, but we support a lot of different things. And our team is always open for cutting-edge collaborations in all areas, including single-cell proteomics, but not only anything that's challenging, that's really pushing us to further improve and develop the tools. And I think it's just, repeat what I just said a few minutes ago, that we need to focus on the quality of identifications, quality of the analysis, and not just the number of identifications coming out of these studies. So thank you. I will acknowledge my lab here and a few collaborators already mentioned, and Ingju in particular, since I used some of his work here today, and my lab. So thank you.
Great talk. I'm a huge fan of MS Fragger Open Search. I know you didn't talk much about it, but I have a question on one of the different things that you mentioned. You said that you throw out identifications if you don't find a precursor. Do you have a sense of what proportion of the time that's happening?
Well, a really good question. Yes. So the question, yes. So we throw out identifications by default, right? You can uncheck that, but I would say that in Orbitrap's data, in Orbitrap DIA data, maybe you would get at most a few 5% boost in number of IDs by accepting identifications where you didn't see an MS1 precursor. So not a whole lot. In Astral data, we actually get less. I think because the MS1 is such a, there is just so much stuff in there already, so much noise that having MS1 as a filter, okay, we don't see the precursor, we not consider this peptide, right? Actually helps us, right? So we're not even losing, we're only gaining. And diapassive data, we are basically at the beginning, we start with detection of MS1 feature to assemble a precursor fragment group. So there we depend on detecting MS1 and that strategy. It doesn't have to be super rigorous MS1 detection, not like you have to see 5 isotopes with the right shape. We can relax the criteria, but we gotta see at least some MS1 signal.
Thank you.
So if I understand right, in FragPipe, the default DIA pipeline is to do something like DIA umpire, where you create a pseudo MS2 spectrum and then send that through. Did I understand that right?
For diapassive data. For diapassive data, yes. For Orbitraps, it's an option and not the default option. Our default option now is MS-Fragger DIA method, where we first search MS2 scan for all candidates without assembling precursor fragment groups, without what DIA umpire is doing. We flip the order of events. We first detect all candidates for each spectrum, like as if it was a regular database search. Then we take those candidates, create MS spectra, MOZ ions of the fragments, go and trace them in the data in a peptide-centric way. So that's our preferred strategy now. And DIA umpire only for certain applications like immunopeptidomics.
Alex, you are a very, very important person for this community here. I do believe that quality is really the challenge for the single cell now. But in the reality, the number of the fragment is getting less and less, even in the DIA. So I'm wondering, is it possible that you can use less number of the fragment ion, but combining with other factors, for example, the monoisotopic distribution, to increase the confidence of identification, instead of say 5 fragment is a minimum? Is that possible?
Yeah, I mean, I think everything is possible. It's we're going through a lot of these optimization experiments constantly, right? So we, for example, in MS-Fragger, we do de-isotoping first, the first step for high resolution data. We first de-isotope fragments and convert them to singly charged fragments, right? And we actually search the data only with the singly, we assume that we got with the de-isotope decharged the fragments, right? So we're not searching like 2+, 3+ fragments. We try to reduce the search space. So in a sense, like we're matching less fragments, but the high quality fragments in a way, right? And it tends to help us. So for single cell data, again, I think more optimization needs to be done, and things are really improving really fast, and how you generate the data and the quality of data is changing. So I think it's still kind of a moving target, but yes, we are going through steps like this always to see how we can improve the tools.
Sorry, one more question. Today, like Ryan talked about the very fast gradient, like 0.5, different kind of short gradient. So in the DIA computation, for this kind of different gradient, do you think the spectral library can be universal? By using the reference peptide like LRT for calibration, is that robust enough?
Well, I think it's a really complicated question. I'm not able to give an answer in 5 seconds. I'll just comment that if you build a sample-specific libraries from your data, you're always going to be the best libraries than relying on something that you build from very different gradients or from bulk data, right? So that's why we prefer building direct DIA plus some data that's generated on the same instrument, in the DDA mode. But I think with short gradients, the more interesting question to me, or equally interesting, is, should we do quantification from MS1 data, for example, right? Identify from MS2 or quantify from MS1. In our pipeline, we try to report both types of quantification. And I think with something like Astros, or Ultra, probably the same, short gradient data, the quality from SMS spectrum is not going to be super great. You can still identify the peptide because you can build a good library, but MS spectra are not great. But because the signal is split between many fragments, but MS1 often looks better. So we prefer to actually quantify from MS1. And again, in our pipeline, we rely on observation of that MS1 signal. So yeah, there's a lot of things to compare and contrast. It's yeah.
Alexei, I think your point about focusing on quantitative accuracy rather than identifications is very, very resonant with many of us. Certainly, I strongly agree with that. And you discussed the challenges with defining metrics for it. It is very difficult, but I think unless we have metrics, it will be very difficult to shift the focus. Whatever is measurable, people optimize. If something's not measurable, it is difficult to advocate for it. So I think it is very important for us to, as a community, to discuss what are these metrics that we are happy with so that we can replace some of the low bar metrics such as consistency of detection or repeatability as quantified by CVs. I agree with you that that's not a very good standard for anything. We can be consistently misidentifying the same signal, consistently misquantifying it, and getting exceedingly low CVs. It's just not a good metric. But we cannot only say what doesn't work. I think we need to find the time to have the discussion of all of the metrics that are deficient in some ways, which are the metrics that are the most useful, and advocate for those, recognizing that they may not be ideal, but still we have to argue for them. I'll make one suggestion. I'm not saying that this is the ultimate and the best. I think this really has to be a wider discussion, but one metric that resonates with me is the consistency of quantification of the same precursor from different scans in, for example, Plex DI data. We normally have multiple MS1 level scans, multiple MS2 level scans, that can allow us to quantify relative abundance changes across multiple Plex DI samples. And I think the consistency between those can be a useful metric. But I'm also very interested to brainstorm with you and with other people in the community and to see what are these metrics that, as imperfect as they are, we want to advocate. Because if we don't advocate for those, people are just going to use CVs, whatever is the easiest thing to use, the lowest common denominator.
I would also add to that I think the importance of how you post-process the data. So we had a tool, MapDIA, actually, that everyone forgot about, but that was a tool in the early days of DIA that would basically correlate fragments to each other across multiple runs and to the precursor to the peptide and even to the protein level as a way to throw away the outlier fragments and peptides that basically show discordant pattern. And as a way to find high quality fragments and use only those for quantification. Of course, there's a challenge that is actually maybe biologically relevant, isoforms that, things like this, but as a filter of low quality data, that was really great. So I guess what I'm trying to say here is that, once you quantify your fragments and peptides, what you do next is equally important. And that's sort of a little bit underdeveloped area at the moment. There was an effort before and kind of now, I think it will come back to sort of looking at post quantification, sort of like the next steps for filtering out, finding the right fragments to use for peptide and protein level quantification up from the initial search.
So let me ask another. I have many questions, so just ask one more so that there is time for others. And this one is more of a challenging question, but curious what you think about it. When you do MS1 level quantitation, certainly in DI data, one of the main sources of error is interference, and that interference tends to come from highly abundant ions nearby that we also quantify. And we usually throw away the contaminated signal, but there's another possibility, which is maybe we can try to estimate that interference because we measure both the source from which it's likely to have come or potential sources, and we measure the ions that have been interfered with. Do you think it is practically feasible to think of ways to model that and at least partially diminish the effect of that interference?
So it's a good suggestion, an interesting idea to pursue. It's a challenging one. I'll give you an example, a simple example where it's actually turned out to be very challenging, like TMT data correction for the isotopic impurities, right? Sounds like a simple problem. You have so much of this channel leaking into this channel, right? And you solve the linear equations and you correct for the data. Well, it turns out that if you do that, despite what maybe Thermo would tell you or others, that actually you make things worse, because for low abundant peptides, where you have the really challenging low abundant data, you start correcting for these things and you make things worse. It works beautifully for high abundant peptides. You can demultiplex, correct for all of that. And then you start getting outliers and you look at them and it's a low abundant signal, you overcorrected, it created differential expression. So it may work really well for good, maybe even for 90% of the signal. The question is, will you start creating artifacts in the data for something at the noise level that perhaps what you're most interested in, accurate quantification of low abundant things. And this is where, unless you do it, you wouldn't know how that would work, right? So, but yes, it's an interesting direction to think about.
Thank you.
How to deal with this shared peptides, like how this tool will consider these shared peptides and how to give weightage for this?
Oh, this is my favorite paper, and it's 20 years now that I wrote a paper called the protein inference problem, right? That talks exactly about that. So we use a tool called protein profit in our pipeline. Again, no matter what, whether it's a DIA, DDA, SILAC, whatever, everything goes through the protein profit, and we assign peptides to proteins with most evidence through the protein profit algorithm. And that's how we approach that. And we have this razor assignment, and most other tools do that. And we use DDA strategies for DIA data as well. Now, what's done in other tools, they say that they also do protein inference and try to sort of assign peptides to protein accession numbers and genes in a consistent way. But it's a little less known what exactly those tools are doing. But yes, no matter what, you have to deal with this protein inference problem of shared peptides. And, we often in proteogenomic studies, and you do, a ton of proteogenomics as part of CPTAC consortium, we often end up summarizing to the gene level, not to deal with the problem, especially if you integrate with RNA data. Of course, if you really need to resolve between protein isoforms and so on, you have to deal with this problem, and it becomes a bigger issue, and you have to find what works for you. But at least in FragPipe, we have a consistent way to assign peptides to proteins. Consistent given the evidence that we have. Unfortunately, we often don't have enough evidence, and we just have to present it to the user in the most transparent way possible, the ambiguity.
I wonder about prediction. So it's relatively easy to predict retention time, cross-section, or fragmentation of MS2. And I wonder how useful it would be to be able to predict MS1 intensity, so we know which peptide from a given protein should be observable, and we don't see them, we know they are modified. And can we also use this information for scoring and other things? I wonder what do you think about MS1 prediction?
Yeah, great. Do you know Parag Malik? He was my friend. He was a postdoc at the same time as ISB as I was in Rudy Abrasov lab. So Parag had a paper 20 years ago also on the flyability and trying to predict what peptides would be observed. And others also worked on that. So, there are several tools, and we tried actually incorporating in MS Booster a prediction of whether a peptide will be observed or not. So it's not exactly the same as predicting the intensity, but predicting whether you will see the peptide and sequence it, right? So it correlates, of course, with the MS1 intensity of the peptide. It had a minimal impact using the tools that we tried at the time, which is a few years ago. I see new tools coming out, and maybe they will benefit the analysis, but I think it has to be a certain type of transfer learning applied, because you can't trade it on gazillion of data from average proteomic study, right? You kind of see what proteins expressed in your particular study, and maybe based on that, figure out what should be the abundances of proteins that you have questions about that's not really clear identifications, right? So I think there is something to that, but it has to be in the context of transfer learning that looks deeper in what you can confidently identify in your study. Learn from that, predict the intensities or observability of proteins in that particular sample, right? For the questionable ones that would benefit from this risk scoring from the predictions. So something along those lines may work.
Well, let's thank our speaker again.
Thank you.
Our next speaker, Dr. Wally, earned his PhD from the University of California, Davis, and serves as a professor in the Department of Plant Biology and Microbiology at Iowa State University, where his lab investigates the molecular mechanisms that underpin plant growth, development, and environmental stresses. The Wally Lab specializes in mass spectrometry-based proteomics to globally quantify protein abundance and post-translational modifications. Please welcome Dr. Wally.
While we get this fixed, I just want to thank the organizers for this conference. It's been great to actually be here in person. I learned a lot over the years online, and it's been a great community with like open tools and resources that's really helped us get this running in our lab. So today I'm going to kind of talk about, does it work on the? Yeah. There we go. Great. Yeah, so to give some context, my lab in general is interested in how plants perceive and respond to environment and tailor their growth for, under these conditions. And we focus a lot on, what are post-translational modifications, acetylation, ubiquitination, and phosphorylation that drive these signal transduction processes, resulting in altered both transcription and protein abundance through non-transcriptional mechanisms. And a lot of what we do involves integrating these data through network reconstructions. So here is integrating kinase signaling, protein abundance, and transcription. And we like to then follow these networks up with high throughput phenotyping. So this is a robot we use to kind of image plants under different conditions. And then we can test these large scale models with specific mutations of different genes that are in these predictions. Or another paper here where we found altered number of meristematic stem cells in the root, and I'll come back to these stem cells later. But the idea of what we do tends to be measure lots of things, measure lots of PTMs, and these are usually starting with milligram quantities of proteins, and we decided why not try something a little more challenging and go down to single cells. But really the motivation is, these approaches are all grinding up cells from all different cell types. So what are we missing when we grind up a whole organ or organism? And, in the context of where we want to end up and go, you can think about single cell RNA sequencing where, 6 years or so ago, this is a Arabidopsis root single cell papers, and they're at 5,000 to 10,000 cells being quantified. You fast forward a few more years now, and in the same root system, and I'll come back to this study in a bit later, but we're now measuring over 100,000 cells in a study. And, I think that is the goal for where single cell proteomics should be. But, obviously there's really cool biology measuring hundreds of cells. But when I'm thinking about the context of hundreds of thousands of cells being measured, that kind of shaped the approaches we wanted to pursue when we picked this up in our lab. And, Nikolai's lab has had a number of great papers documenting differences, and you all have developed wonderful tools. So we really focused on these TMT DDA approaches for multiplexing, so we could start building on scaling samples. We're excited for some of the Plex DIA that's coming out that'll enable more and more DIA-based multiplexed methods. So again, we focused on these TMT methods. And you all know in this room the Scope 2 workflow. And with our system that we had available, we're using MPOP, not NPOP. But one thing I wanted to point out the difference is for plant samples, plants are dirty. So often the carrier and reference in these scope styles is comprised of actual cells deposited in a well. What we did is collect bunches of cells, I think it was like 50,000 cells, and used normal protein extraction methods to get clean peptides for the carrier and the reference to help avoid column and ion optics contamination. And then we just aliquoted the equivalent amounts of peptides that you would expect from either a single cell or 5 cells or 100 cells, what have you. And the other thing I want to point out with plants is I'm saying cells. What I'm actually referring to are what we call protoplasts. So, plants are multicellular organisms. Their cells are surrounded by cell walls. To release them, plant biologists, whether it's RNA sequencing or anything single cell approaches, will use enzymes to digest away those cell walls. And what you have is a plasma membrane enclosed protoplast. So it's actually protoplasts we're working with in these studies. As we went through our journey to get this up and running, our efforts in this started and stopped over the years. But we tested lots of different methods, and kind of the focus point here is we measured on the average in a Arabidopsis root cell, they have about 100 picograms of protein per cell. So if we have a reference, single cells, in either 100 or 200 carrier cells, we're looking at 20 to 37 nanograms of protein per plex. So we tested different strategies, and ultimately we settled on acquisition studies from Nikolai's lab. We also are using a QE+. Theirs was from a QE Classic, but very similar. We used a MicroPack 200 centimeter column. We used a trap column as well, and some different gradient tests. So that's kind of where we ended up for acquisition settings and throughput. And now I'll get into the actual single cell study that we worked on, and it's asked a very simple question: could we differentiate the proteomes of 2 really related root cell types using single cell proteomics? And to orient you here, these are Arabidopsis seedlings that have germinated on a plate. And you can see the roots are growing down. So roots grow from the tip. So this very endpoint here, that's the new cells being deposited from the root apical meristem. And what's kind of nice about this system is that the meristem contains populations of cells that differentiates into all the cell types in the root. So these stem cells are, again, in the meristem, that's at the tip of the root growing down, and they're differentiated into the different cell types within the root. You can see that here in this video where the new cells are color-coded. So every time a new cell is deposited, it's color-coded. And you can see new cells show up in this region here. And then once you get it past this zone, the cells start expanding. So there's no more division in this region, it's just cell expansion. So you get root growth by the combination of division and expansion of the cells. And what this sets up is that the root is a developmental timeline. So young cells are at the tip, old cells are at the top near the soil. And the other feature of this is that the cells are deposited in concentric, or cell types are in a concentric ring. So each ring in a Arabidopsis root is a different cell type. So you get this mix of spatiotemporal division where you get all the cell types in a cross section and age as you go along the axis of the root. And then if you look closer at the 2 stem cells or 2 types of cells that we focused on, it's the endodermis and the cortex. Endodermis and cortex here are in yellow and blue, and they originate from the same meristematic stem cell. So these are 2 very closely related cell types. They differentiate again from the same stem cell, ultimately. And so what we asked is, can we tell the proteomes of these apart using single cell proteomics? And what we approach we took here is we took marker lines. The other nice thing about the Arabidopsis root is every one of those cell types, there are marker lines where you can get and color code the cells in that different cell type. This is showing it in one root, but it's actually 2 different marker lines, so they were sorted separately. And nonetheless, Trevor Nolan and Philip Benfey's group at the time sorted these cells using fax, deposited them to 384 well plates, and then Christian in the audience here took over and developed a OpenTrons OT2 workflow for digestion and trypsin labeling. We used single cells. This is TMT 18-plex throughout a couple of the channels. 14 cells, 7 from each cell type were in each multiplex set, 5-cell reference, and with the amount of peptides we had, we estimate that we were adding about 135x carrier then in the end. And then these software tools developed, DOMS, Dart ID, and the SCP package were all used at different steps in this workflow, whether for optimization or for identification and quantification. Oh, I thought I hid this slide, but this is the OT True in action. So Ryan Kelly's lab has also been great with tools. So this was getting the OpenTrons was spurned by the Autopot system, and we also got the TCAN Uno he's described. So where did we go with the data? So the SCP package spits out some nice metrics here. So some of the things we looked at were cumulative sensitivity. You can see both the cortex and the endodermis cells, the different cell types, the cells are showing similar metrics, also in terms of data completeness and local sensitivity. And then if you look at card index, also showing that the two cell types are very similar. So this SCP package was also a very nice tool to really help us get through the data analysis and benchmark that we're getting decent quality data, and you can preload it with other SCP studies out there, and it was a really nice feature. So what we ended up with is, we processed about 770 single cells. We could identify about 3,700 proteins or so across all those. And we did a number of filterings past that. So coefficient of variation filtering was really important for us to get high quality quantitative data. And what you can see here is I'm showing here the stringent filtering where we ended up focusing on 81 out of these 700 and some cells. And that was based on this CV filtering approach. And I'll come back to why I think that filtering removed a bunch of our cells. But nonetheless, we had about 590 some proteins that were then differentially expressed when we do all these filterings between the endodermis and the cortex cells. And this was done on a Q-Exactive Plus mass spec again that was on its last legs at the time. So what were we able to identify, or what types? This is coming back to the Shahan et al. Single cell RNA sequencing paper where they quantified about 110,000 single cells. This was from the same group that grew the Arabidopsis plants for us into the sorting, so that the plants were grown in the same conditions in the same lab's growth chambers. So we took their single cell transcriptome data in order, it's kind of cut off at the bottom here, but we did rank order abundance of the transcripts on the x-axis and then transcript abundance by CPM on the y-axis. So this is the most abundant transcripts to the least abundance, and then we color coded whether or not we detected the proteins from those given genes. So the same G product from same gene. So you can see we see proteins from all the way from high to low transcript expressed genes, but obviously we're more likely to detect high abundant transcripts. So if you look at the histograms here, there's a lot of these lower abundance transcripts where we don't detect the protein as expected. And then what kind of proteins? Everybody always asks us, do you detect stuff that we find interesting? And that's often kinases and transcription factors. So among those 3,200 quantified proteins, we have about 175 quantified kinases, a similar number of transcription factors. And here's a similar graph, but now it's rank protein abundance and rank protein intensity. And you can see that both kinases and transcription factors also span high to low protein abundance groups. That's great. We can quantify stuff. Is it what we would expect based on what we know about the biology of these two cell types? So in these MA plots showing average intensity versus fold change, where stuff that's enriched in the cortex is up here and stuff enriched in the endodermis is down here. We asked, do we find proteins we would expect in those different cell types? So the first one we looked at is this auxin response factor 5 transcription factor. It's enriched in these cortex cells where we had expected it. It's known to drive cortical cell division. Similarly, there was a couple other proteins that were known to drive different aspects of maintaining hormone distributions and stuff in the cortex where we would expect them. Conversely, if we look at stuff enriched in the endodermis, we have proteins like this Casp1, which drives Casparian strip formation. So this is this superized file of that's deposited along the endodermal cells to help form a barrier between the xylem and the phloem, et cetera. But it's where we would expect it to be. This protein involved in redox signaling is also enriched in these endodermal cells. So we find stuff where we'd expect them, and then we asked if we could differentiate the cell types. We did this both with PCA and UMAP. You can see nice separation of the two cell types. Similarly, if we used hierarchical clustering and clustered by the cells on up here, you can see largely the endodermal cells separate from the cortical cells. So I think what we wanted, it kind of ended up benchmarking for us that yes, these two really closely related cell types can be differentiated. And coming back to this idea of variability between cells, we believe that much of this is biological. So if you notice in the video, the cells at the younger cells are very small, and as you go up the root, you get very large cells. So you have this very large distribution of cell sizes along the gradient of these roots. And we think this is what's driving some of that variation in what we're quantifying. And a number of different studies have shown in various systems that histones can normalize for some of these cell size artifacts. The complication in a number of plant tissues, including plant roots, is they undergo extensive endoreduplication of the genomes. So you have like 32 N cells, for example, within the root. And so you'll have lots of histones that don't necessarily track with these cell size differences. And what I had hoped to actually talk about in this talk was to find proteins to normalize this, but our work has gotten bogged down this year. So maybe another year we'll have a better answer of what is actually good for normalizing for cell size, and also addressing, is it really the cell size that's causing us to throw out a number of these cells? And then what I'm kind of hopeful for where we're going is I think that single nuclei proteomics has some promise, and I'm interested in that. I think we need to get to these large organ and organism level cell atlases. And I'd be hopeful we can pair this with single cell RNA sequencing data. And I was talking with Hannah at lunch today, and hopefully she'll have some nice data in the next coming years able to do this. And then, once we can pair these different data types, I love integrating these different types with networks, and that's where I hope we end up. So with that, I'll thank people that did the work, namely Christian, who's here if you have more questions. Trevor was a postdoc with Philip Benfey, and he now has his own lab at Caltech. And then the Iowa State Plant Sciences Institute funded a lot of this work, as well as the College of Ag and Life Sciences for buying mass specs. So with that, I'll take any questions.
So I'm always curious about trying to normalize out size.
Yeah.
Is there a reason beyond QC that you don't want to know why big cells are different than you're developing just born cells?
Because the protein content is very different. So you could tell cells are different along that gradient, but then I think it gets hard to find meaningful biological differences. Is it just driven by really abundant proteins, So it's a tricky thing. And the other, issue with plants is the cells become heavily, they have vacuoles that are largely water. So then that's another contributing factor. So also cell size doesn't necessarily contribute to protein content per se.
What I understood from the morning is that if the cell is bigger in size, then we'll get a more number of proteins in the mass spec, even if it is single cell, then compared to the smaller cell, because you said that these two cells are very different in size. I'm curious, is there any protein or is difference between these two cell lines?
So I'll clarify. I think at a given position along the route, longitudinally, the cells will be similarly sized. It's just that even within a cell type, the cells that are young are going to be very different from the cells that are old. So even within the cell type itself, there's differences in sizes. So it's not necessarily the cell size of one cell type versus the other. It's more the position along the route.
I love to see single-cell proteomics applied in plants, especially in such a cool development system where you have cells differentiating. And I wonder if you can exploit this to infer a pseudotime trajectory, which in this particular case, you're perhaps in a position to validate based on the size of the cells, based on additional tools that you have for Arabidopsis. This seems like a particularly cool analysis for the model system that you're focusing on.
Yep, and that's with Trevor, he's continuing to do a lot of the single cell RNA sequencing and has done the pseudo-time trajectories for all the different cell types in the root. So we're hopeful to be able to build enough cells with proteomics data to do that exact question.
Great, thank you.
Any other questions?
Just want to follow up on the previous question about the trajectory analysis, because you already have the single cell RNA-seq, and that's maybe not a good question that I'm asking at the Proteomics conference, but what do you expect that proteomics can provide, like beyond what this single cell RNA-seq can in this type of analysis? There's of course a disconnect, but do you think that's going to be like new biology that you're looking at, or like what's the overall goal?
I think that it can be new biology, and particularly there's a number of proteins that are known to be from genes that are transcribed in one cell type, and the protein's translated and moves into adjacent cell types. So the trajectories of where the RNA and the protein are, we know for a number of these key developmentally regulated proteins are cell type distinct. So if you're looking at the RNA of them in one cell type, the protein's actually acting in the adjacent cell type, but has moved. Thank you.
All right, so after some exciting science, we've earned another break, I believe. We're going to take a 30 break and then return at 5:45 for the final session of today's afternoon.
Good to see you.
Yeah, I'm gonna go right now. Okay. You did set it up. It's all set. Now you just have to make sure.
Thank you. Bye. I'll see you after 7.
Of course.
If someone is sure. But I think it needs to be like So now you well, we don't get this. Yeah, although we raised the standard. Well, not today, this is the. Yeah, no, but I think there was like. Anyway, Jason. Yeah, actually, I'm gonna but I can still so then you can like draw here, like officially. English makes it they're like we want you to look like somebody's you go to almost in place. So they also, I don't know, I was living here too, or like You get the house rotating, but it's like, you realize it's like, like question is. I was like, oh, wow. Yeah. No. If somebody called me. Well, they're like. Yeah, I'm trying to figure out what to do. Like, I feel like I work on it. You know, it's like I found myself thinking, oh, that's really fun. Yeah, there's all signs. Talk to the guy that I was well, legit in some ways, but then you know. So like yeah, no, are there? Well, I don't actually think it's weird because I think like but you guys convinced me, so you have people who do one count with these weights and even any individual right? Like sometimes like the first one you know it's like you never jump more than the second one you're like so you would like put these data and it would just be like these four. But these are all my great right. Yeah, so I did aspect, you know. Right. So yeah, I should write to yeah. Somebody who's in my 75% of their time. Okay. Yeah, it's like half the yeah. So, general sufficiently. I got transitioning so that he's tired. So I got there, he was no longer. That's I age and that he won't interest in just in the end very fun about. Yeah, these are the people I brought in who also signed. You could build right in strong. Yeah. See, you got your own super cool. Yeah. Sorry, it's not to me, it's not like well, yeah. But some works on two. People, it's still like I would say like I usually but we think about We had on the board. I see. No, it's still the same. So, yeah, it's like 2010. They could just say. Oh, yeah, that's all right. Thanks for. Yeah. Yeah. That's not so, whatever you hate, like, people, you know, like, so. They actually, they're what? Feeling that. I think that's my just have to. Yeah. How are you? Good. What's that? Right there on that book. Yeah. Yeah, yeah. Slowly. How are you? It's always not a struggle. I was going to say that's not right. I think that's shiny slides. Yeah. It's gonna be like like formula style. So possible. She going to be other. Third. I'm also the record. Yeah, that's time. It's a really good feeling. I'll still like a bit of wrap up work. Yeah. Yes, of course you. Yeah, so I'll be wrong, but another like days or 3 days. Okay. I will start being strong. Yeah. Maybe also. Yeah. It's what I used to know this. So yes, I don't care what you like the yeah. Yeah, well, but you can have some sense some easy to take the battle and also we don't have them especially if they want to take a high utility or every stuff outside. there's one problem when I build.
Yeah.
Just the water.
Yes.
So we speak of all the functions. So individualized, that's just a little bit. So, yeah. So there are side species. So on we have fixed. Yes, well, it's a milestone, yes. It would be nice to have some savings. Extract it out. 2 ions. Force the transit from eyes. Slope your charging. But it doesn't. Same thing. It's always slope. Slope. So yeah, should we go? So the charge was so very plus 1 minus 1 charge should have been passed.
So I think she has them on here. And right. Yeah, that's me. You can just grab that. And I see that's right. Presented. Yeah, probably. But I think. Can that show the presenter view?
Oh, we have not been able to figure that one out, unfortunately.
Yeah, we'll be fine.
You sure? Sorry, unfortunately. Okay.
It will be fine.
And there's a clicker here if you prefer to use that, but it's.
There's a.
Yep.
This one.
The mic is on. We should be starting soon. Let me go upstairs. Sure.
Usually introduce yourself. What do you need to introduce you? Who do you think you are?
But the mic is not functioning.
Yeah, tell me. Ask for the ring. What is this? This is constant terrible. That's me. I was going to say it's going.
I don't understand. You just put it on a Windows computer, and then you just run it for 8 days.
Big old Windows. If it breaks down after 7 days,
Just do it again. What are you talking about?
PhD student
This
harder.
is the problem with you people, with you computer people. You know what I mean? You just make up stuff that nobody needs.
I will explain why we can't do this.
I mean, being ridiculous. This is trying to test its level of message. All our data sets, okay, are like 800 DIA runs, run in one giant diad search for MBR. And yeah, it works out most of the time.
Works out every time, 80% of the time, every time.
Yeah, I'm a drunk, so I did it on the cluster, but everybody else just did it on the Windows computer. But I already did it on the cluster. I don't need this.
Other side.
It would be terrible if we did this. But just check it. It's on. Yeah, it's on. Oh, it's supposed to be. I think of all of it.
You have to, so go up to the.
Sure, of course. Yes, I'll introduce you to all of them and certainly all of the out there. He's going to be taking notes. Hello. Okay. Hi, everybody, welcome back from coffee break. It's now my distinct pleasure to introduce Georg Wallman, or as he asked me to call him for 10 months, George. And as I called him, Jorge, whichever you like. So Georg has a long history with SCP in the Slavov lab. So he came to us for his master's thesis and was with us for about 10 months. And it was a very, very, very successful, very good time, I think, very productive. Georg authored a paper sort of extending the DUMS framework for DIA data. While doing that, he also got into understanding how search engines work, feature identification, and many more things. After his thesis was done, of course, Georg moved forward and went to the Mon Lab at the Max Planck Institute, where he's been a PhD student now for a couple of years. And he has, again, continued a lot of computational workflows. His sort of main publication was Alpha DIA, which is an open source, I'm calling it main, hopefully you agree, Alpha DIA, which is an open source search engine end-to-end that uses transfer learning. And yeah, today Georg's going to tell us about exploring the limits of large-scale DIA search. Looking forward to your talk, Georg.
Thank you so much, Sat, for the introduction. Yes, so I'm a PhD student at the TS Mans Lab, working on all sorts of problems involving DIA search and how we can improve it, how we can make it accessible to the community. And part of this is also kind of adapting to the changes we've seen in the field of proteomics over the course of the last years. And this is that we have achieved substantially higher throughput, so throughput to a level which we wouldn't have imagined a couple of years ago. So more than 100 samples per day measured on a single instrument. We have these very sensitive TOF detectors like the Astral and the TIMS-TOF, which allowed a very short gradients, very deep proteome coverage. And this has resulted in quite large cohorts and larger, ever larger data sets. So be it single cell proteomics, but also plasma studies, tissue studies, where we start seeing more than 10,000 samples being quantified in the same run. And single cell proteomics and also kind of the projects and data sets we've seen today is a major driver. And this brings the challenge about how we can continue processing these data sets and how we can, adapt the tools we need to not hit a roadblock at some point. And I can imagine most of you had a large study, maybe a couple hundred samples, maybe a couple thousand samples. You put them on some Windows computer, some workstation. You started to search, and after 4 days, 5 days, 6 days of work, it crashed out of memory, and you then just went forward, did it again. Maybe you were lucky, maybe you bought a bigger computer. But of course, this is a work mode which doesn't scale. And so what I'll be talking about today, kind of very briefly for the last session, is to give you some perspective how DIA search works, kind of what steps happen in between, and how all of this translates with computational complexity and how we can adapt it for distributed search so that we are not limited anymore with the number of samples we can process. And in the end, I will give you the perspective how we've used this with some of our largest cohorts, which is like a plasma study, which is about to be published with the Bill and Melinda Gates Foundation, where we've processed and quantified more than 40,000 proteomic samples, so multiple years of astral measurement time in a single run, successfully with label-free quantification. So the platform we are using is AlphaDIA, it's hard to introduce. So it's an open source DIA search engine, which is user accessible, which allows kind of everybody to process their files. And we've released the first version as a preprint last year. The paper will come out briefly or shortly. And we are kind of continuously developing like an open ecosystem, which where we want to, of course, offer cutting edge tools, but where we also want to make transparent how the processing works. And we want to offer kind of the community the tools that they can adapt and improve existing methods. And that kind of, especially with DIA, where this data is so complicated, we move away from this black box processing approach. And what AlphaDIA gives us is full control over the processing workflow. So we start with the raw files in the beginning, be it from the SciEx instruments, the Astral instruments, or also from the timsTOF instruments. We have the spectral library prediction with the package PepDeep from our AlphaX ecosystem. We handle libraries, add annotations, add modifications, perform mask calculations, and then throw everything into AlphaDIA. And following search and FDR correction, we end up with our FDR-controlled precursor list, and in the end get our protein quantities from direct LFQ. And one of the nice things is that this runs on all, it's mostly Python-based with some kind of high-performance improvements, and it runs on all different infrastructures. So we can run it on Windows, on Linux, and we can also run it on the cluster. And the story I'll present you today is how we've adapted it for very highly parallelized distributed cluster use. So how we can, the example I will be using, So what are the steps which we have in DIA search? The first step, it's kind of obvious. We know that most search engines do it by default. If we're talking about peptide-centric search, in a way how Diane performs it, Spectral performs it, or also AlphaDIA performs it, we start with in silico library prediction. So we're building a library of 2 or 3 million precursors potentially detectable based on a FASTA file. Happens once per analysis, and it's usually a pretty fast process, takes maybe 20, 30 minutes, and the output is our spectral library. We then go on, and then kind of the computational complexity starts using a first search with a fully predicted library. So this means that we are quantifying and extracting precursors at 1% local FDR, and we have to perform this process once per raw file. And this is usually what is the computational bottleneck if we perform large-scale analysis, because it's performed sequentially. So if we have thousands of files, we have to wait for one file to finish for the next one to begin. So we went with a modularization approach where we could do these searches all in parallel, and the precursor quant, which is then generated in the end, could be collected. This precursor quant is then collected in the third step, which is a consensus step, which means that we start gathering all of this information, all of these quantified precursors, and combine it in an MBR library. And at this step, we also perform the protein inference, we perform false discovery correction, and we perform spectral library building for the next step. What is the challenge about this? Computationally, gathering all of this information is not very, very challenging, but we have to load everything at the same time into memory. So we are limited by space, not by time. Then we go into the second step of the search, which is the MBR search, with the goal to reduce the missingness in the data and to increase the completeness, and also to help identification of locally abundant peptides, especially in the context of single cell, or to in the context of single cell or plasma. We perform this step again once per raw file, so we have the challenge that we have to distribute this load, but because this is somewhat independent, we can take the same strategy as from the first search, take different quant files, and then kind of combine them in the final consensus step. And here the challenge is really that we want to perform the label-free quantification, where we compare kind of like with like, so we compare the same precursor across all different samples, and this means that one last time we have to load everything into memory at the same time, and we're limited with space complexity. We went forward and implemented this distributed search approach, and we have the tools online. You can check them out if you want to get started yourself to parallelize this in a cluster or in a cloud setting yourself. So you can use it with your Slurm cluster, your research cluster, to have a search which runs much faster and can also be continued if it stops in between. So we went forward to test this with the project of a colleague of mine, Marvin Thielert, who's also here today, who's working on some of our single cell data sets, using a data set of 1,670 single HeLa cells, and acquired or processed with the cell-in-one, then measured with the Wisp-Zoom gradient ATSPD on the Orbitrap Astral. And this data set, couple of hundreds of gigabytes of data, we wanted to process on our local cluster with 50 different nodes, and then 2 jobs per node. And this wasn't any kind of high performance computing setup. You can imagine if you have such a cluster and you clog it up with jobs for the next 2 weeks, your colleagues will be mad. So we actually used kind of the oldest partition of that cluster. So it's only 10 cores, 10 different CPU cores per job, and 64 gigabytes of memory. So it's not that you need like the most up-to-date hardware to do this well. So this is what we used. So for the first step, predicting a spectral library, as I mentioned, it's kind of sequentially, this was easy to do. So this would have taken, or this took 20 minutes in the whole workflow. But then kind of the major bottleneck was, as I mentioned, the first search. Within the first search, what we are doing is we take the library in a peptide-centric manner. We look for each and every precursor if there's evidence in the data. If we can score that precursor, if we can gather the evidence for that precursor, and if it then kind of survives in the FDR competition. And what we see is that this step consists of 2 different substeps. The first one is that we have to identify all of the hyperparameters. This is what I name, or what I call here, a search space optimization, where we calibrate the retention time predictions, where we reduce the retention time limits, where we reduce the mass limits you need to quantify a precursor with a given mass accuracy. And what we see is that these steps on average took 20 minutes per single cell. And if we look at the correlation between the duration of the optimization step and also the number of proteins we got, we see, interestingly, a clearly negative correlation. So if you have lots of input material in a single cell and you can identify a lot of precursors, it's much faster to perform the step. But the lower we go, the fewer proteins we find, the longer it takes for the optimization. And this is something you might know kind of intuitively from running searches yourself, that the fewer material is in your sample, the longer it takes kind of for example, Diane to identify anything, and you can end up kind of with it just searching for hours in the same data. In the end, kind of the quantification was pretty fast, was like 8 minutes per cell for the whole library. So we ended up with a relatively slow process of 28 minutes per cell. But because we had used 100 different jobs in parallel, we were able to use to search 3.7 cells per minute. So we were able to search the whole data set in 7.5 hours. Then kind of we started the first gather operation, where we created the metropolitan run library of all precursors, identified the 1% FDR, and we went into the second search. And as the search space was already reduced very much at this stage, so we were only looking for 40 to 50,000 precursors across the whole data set, it was much faster and much easier to quantify. So the search space optimization, where we found the right search parameters, only took around 2 minutes. And you can see that on average, we quantified around 4,000 proteins in these single cells. And the quantification itself, the extraction of the quantitative information, only took around half a minute. And this also tells us that the second step in this 2-step searches is so much faster, which means if we even if we have 1,000, 2,000, 5,000 cells, it might make sense to perform the library building with a spectrum-centric tool like MS-Fragger, or only with a subset of the library, which still kind of exhaustively covers all precursors which can be potentially identified, and tend to use only this fast peptide-centric search for the quantification. So kind of processing this whole data set with that library only took 60 minutes in total. And if we would now reprocess it, for example, with an existing library or in a library which was empirically acquired, this is something you could easily do kind of with a click of a button, and you would have the results 1 hour later. And kind of speeding this process up, of course, gives the researcher then the ability to iterate much faster and test hypotheses and find out what issues there might be with the data processing. The last step was then the label-free quantification, which kind of was pretty straightforward, took 50 minutes. So we were able kind of to process this large data set in around 10 hours of total time. So how far does this scale? As I brought up, all of these different processes can be slow, but we can parallelize them pretty well. But if we do this, there's another issue. And this is that these steps in between, so these kind of consensus steps where we combine all data, can be a limiting factor. And the reason for this is that we have to combine all of the information on one machine, which means that we might be limited by memory. And when we worked on this large plasma proteomics study with more than 40,000 samples, we actually weren't able to quantify everything in Diane, even if we used a computer with like a terabyte of memory, because it was just beyond to kind of what you could gather on a single machine. So we looked into our internal LFQ implementation, direct LFQ of a colleague of mine, Konstantin, and we realized that that how LFQ works can actually be exploited to scale further, even to these very large studies. And the idea is that during search, we parallelize across these rows. So if you think about the matrix you want to get out of the quantification, the LFQ matrix, we have a matrix of individual runs as rows, and then individual proteins as columns. And what we're interested in as an output is the protein quantities in that matrix, and as an input, the different precursors and fragments which give quantitative information to this. So during search, we can easily just look at one slice across the row, and we don't have a very complicated problem because we only look at one file at a time. But then kind of during classical LFQ, we load everything into memory. At the same time, we only, for quantifying a single protein, we only need the information from that single protein across all different runs. So we went with a vertical slice and adapted our internal direct LFQ workflow to operate on single proteins at a time. Or batches of proteins at a time, a couple tens, a couple hundreds, and then kind of combine this information in the end into a single table with a delayed normalization kind of to account for different total intensities or different kind of ionization efficiencies. So this turned out to work very well, and also kind of to be not to scale much beyond what might be 40,000 samples. So this is something which will be then published, or which is about to be published with the manuscript for that plasma study. So what is the takeaway? We can scale these proteomic workloads optimally, facilitating ever larger cohorts, even going to tens of thousands of samples. And the takeaway is also that library building optimization are still computational bottlenecks in such a DIA search workflow. So the search itself is somewhat simple, but finding the right parameters, understanding the data your instrument has produced and how it was produced, can be challenging. And also that label-free quantification has been a memory bottleneck, but we think we have found a solution how to scale it out of memory kind of. So yes, all of this has been a collaboration with friends and colleagues from our lab, working on kind of bioinformatics topics, but also on some applying these methods. And we're also supported by lots of sponsors. So I'm open to any questions you might have.
So I have a technical question about your second pass search, which you call match between runs. Typically, match between runs means that you only use MS1 information. here it kind of sounds like that's not the case.
Yeah, it's a very good point. So I use the term match between runs in the same way as it's used by Diane, for example, in a way that you have a focused library.
It's a two-pass.
It's a two-pass search. Yeah, I feel like this is one of the kind of the naming issues which have persisted in the field, and then somebody started calling it match between run, and it has kind of sticked around. It's a very good point.
Hey, Georg, as always, very clear presentation. When it comes to the building of the spectral library, the optimized spectral library for very large data sets, let's say that we analyze 10 million single cells, it seems to me that at some point there will be a sub-ensemble of the cells that is large enough to give you all the benefits of building that spectral library, so that we don't need to build it from every single cell that we analyzed. We can just take a group of maybe 2,000 or 5,000 single cells, and that will give us all the benefit if we sample it. So maybe that's one approach of determining at what point the marginal gains of including more runs in optimization are beginning to level off, and it's not worthwhile pushing the limits on memory.
That's a very good point. So this is for sure one strategy, and the question is, when do you start leveling off? And one thing we've seen, I don't have the plot on the slides, but we've done this experiment with this very large cohort of 40,000 plasma samples. And interestingly, we see that it never really levels off and becomes flat, but it kind of keeps increasing at a very slow pace. So we think that there are some FDR issues hidden, at least with the tools we've used, which in this case was Diane, on the protein group level, which only emerge or which only manifest itself if you go into the range of thousands, maybe tens of thousands of samples. But there's also another answer to kind of what this number can be. And this is, we all use data completeness filters before we process our data. So for example, we filter our proteins to 60, 70, 80% data completeness. And this also kind of gives a natural, I mean, you can do the statistics. If you have an 80% data completeness filter, you can, after 10 or maybe 20 samples, it becomes highly unlikely that you haven't covered any of these proteins just by random chance. So the number of samples we need for generating a library can be surprisingly low. I think even like a 20% of the samples can be sufficient. Yeah.
Thank you. And I have another quick question. I know that you have thought a lot about, and I think it would be of benefit to this community to discuss. And namely, we optimize the parameters of our spectral libraries on the same data that then we go into research. And there is this possibility of circular logic. And I know we all want to avoid that, and you have thought about it. So if you can share with us your thoughts of what do you do to avoid that?
Yeah, that's a very good point. So what I think I think what's important to use is use the best practices from machine learning. So when we try to apply these methods from machine learning, we also have to use kind of, it's not only about kind of using all of the amazing libraries and all of the capabilities, but also kind of taking the kind of the how to put it, like the necessary kind of steps you have to take. And this is using, for example, kind of meaningful and good test and train and validation splits to make sure that the data you're validating on, or that you're validating on a portion of the data you haven't used in the training of the model. This is one thing. The other benchmark we've run a lot with the transfer learning, for example, is that we've used transfer learning in combination with entrapments. And we've included the false positive identifications from the entrapment search in the transfer learning training data to see if they are being memorized in the next step for the library prediction. And even if we do multiple iterations of that transfer learning, we see that the number of false positive identifications in the entrapment FDR actually goes down and not up. And most likely for the reason that for these false positive identifications, the model cannot generalize, and it needs the capacity of the neurons to model the real true behavior of the peptides, where it can lower the loss compared to overfitting on some spurious data. Yeah.
Thank you.
So for building the spectral library, because we are starting with a theoretical spectral library, then in the first pass, we are making a library from the data. So my question is, like, what is the need of keeping a quantitation at that point? Because in generally for other softwares, when you build a spectral library, you never have a quantitation values there. So what is the need of there? And if you do a quantitation, like is it like MS1 level quantitation or MS2 level quantitation, or keeping a both? And I mean, what is the need of, because anyway we are quantifying the peptides at the second level. So can we reduce the time?
Yeah.
Because
It's a very good point. So we don't need any protein or peptide level quantification. We need precursor level quantification, for example, for predicting the charge state and fragment level quantification to train models for the spectral library. But we don't need any protein level quantification. That's so. This was maybe kind of a bit not quite clear, but kind of the LFQ is only needed in the last step and not in the middle. Yeah. Thank you. Yes. Yeah.
Any more questions for Georg? Okay, great.
Thank you.
Hi all. Today I'm excited to be introducing Dr. Yuju Chen. Dr. Chen is coming to us from Academia Sinica, and she is also an adjunct professor at many universities in Taiwan. Today she will be presenting to us on nanoscale to single cell phosphoproteomics. Welcome, Dr. Chen.
Can you hear me now? Can you hear me in the back? Okay. Thank you very much for the invitation. We are kind of newcomer in the field of single cell proteomics, and my lab is interested in clinical proteomics for long term, and accidentally entered the single cell proteomics field. So today I'm going to share with you our very recent study on the single cell phosphoproteomics, and the story is it? I cannot move. Yes. Cannot move. Okay. So okay. Okay, so the story starts with our favorite activities on the protein modification, especially on the cell membrane. And I started to go into the proteomics when I know nothing about biochemistry. So at that time, I thought, oh, cell membrane must be very important, but I did not realize it's so challenging. So if you look at the cell membrane, on the outside of the membrane, we have a lot of extensive modification on the glycan structure. And because of unique structures, so that is important for a lot of affinity between cell-cell communication or the protein-protein interaction. But after the cell receives the signal, the next step is signal transduction. And it's interesting to see that they are coming from the same protein. So there's a lot of interesting questions remain. For example, on the same protein, whether the glycosylation may also change the protein phosphorylation. But unfortunately, due to the technical challenge, most people still work on a single modification, either glycosylation or phosphorylation. So a lot of questions actually still remain. For example, I'm so interested to see what is the site-specific regulation. This may be extremely challenging for even glycoproteomics. And we talk about signal transduction. What is the absolute amount of the quantitation that is achieved or essential to activate the cellular function? This is still not known. And finally, I'm also interested in the synergy and dynamics of this kind of modification. So far, we have a lot of biomarker, we have a lot of target therapy using receptors. But even for the case of EGFR, for example, one of the most popular target therapy, there's a lot of unknown questions. For example, what are the signal transduction of EGFR for the patient level, and not to mention the single cell level. So I think this is a great space that we still need a lot of effort to understand what is happening on the cell membrane. And we actually spent some years to learn the technology, to develop technology required to answer these questions. And this is a kind of an old story we learned from nature. And my best friend, Guo Yilin, is a very excellent B-cell biologist. And one day she told me that she found something interesting. When the B cells start to have accumulation of ocucination, she discovered that the B cell will go from activation to apoptosis just by accumulated ocucination. So we perform very extensive dynamic phosphoproteomics and also the ocucination to analyze what is happening at the cellular level. To make the story short, and finally, we discovered that the single modification on LSP1 will change the B cell from activation to apoptosis. So once the B cell receive ocucination, and that will further recruit other kinases like PKC on the LSP1. And the PKC is a kinase to further do phosphorylation on LSP1. So it's kind of amazing that by two modifications on the same protein, the cell fate is totally changed for the B cell. So, the nature is so amazing. And encouraged by this story, we further pursued whether this is also helpful at the patient level. And this is an example on the CLL patient analysis. We try to analyze the phosphoprotein profile of the B cell purified from the human, from the CLL patient, and compare to the healthy control. And one of the protein we discovered is CAP1. Is block of layer, serine phosphorylation on the 473. And Guoyi spent a few years, 2 to 3 years, from cell line all the way to the animal, and discovered that this is an important site to regulate cell cycle progression for the CLL. And then I'm much more interested to see whether this is useful. And by patient analysis, we discovered that this specific phosphorylation site is very, very dependent on the inhibition of the drug and also recurrence of the drug. But during the process, you can see that we just recently published, but this project started, I think, more than 5 years ago. The challenge is it's so difficult to analyze the phosphoproteomic profile from the limited cell. And even we collect the sample from the patients, there are only a few micrograms of the sample. That is so challenging to see sufficient depth of the phosphoproteomics. So even this is useful, we realize the sensitivity may not be good enough for the clinical proteomics. So we start to see whether we can try other approach. And then we start to be very interested on the data-independent DIM mass spectrometry. So we come up with the idea, if we are able to collect a big library using different kind of cell line, different kind of tissue from the same disease, maybe we can use this kind of global spectral library to enhance the sensitivity with a single shot manner. And this was done many years ago. So at that time, we are so excited that we are able to get many thousands of the protein and many thousands of phosphopeptide in a single shot DI approach. So with this success, you can also see that it works so well, even for the phosphoproteomics. So we use 165 synthetic phosphopeptide from the non-small cell lung cancer pathway. And you could see that the quantitation performance is excellent. So encouraged by this approach, we start to see, oh, DIA is a wonderful tool. But then we encounter another challenge again. From the cell to the animal to the human tissue, even for FFP tissue or biopsy tissue, everything is perfect. But when you come to the even more challenging sample, like for example, the tumor microenvironment, the cell-cell, immune cell, cancer cell interaction, you need much, much better sensitivity. But I also want to comment that when we count the protein identification number, it really depends on the depth of the disease or the biology. You cannot tell, oh, we must need 1,000 protein, 2,000 protein. The truth is we have to see the core of the network of the specific biology or disease. So this further inspires to explore the possibility to see whether we can have more sensitive approach for the clinical proteomics. And again, so that comes to the discussion about the current state sensitivity. And recently, we just published a review article. We try to summarize all the sensitive approach from the community. So the blue line, the blue circles means the single cell proteomics. And you can see that there's a lot of advancement for the single micro scale all the way to the single cell proteomics. And I think a lot of invention are from the experts sitting here. But if you look at the pink circles, which are the phosphoproteomics, the base approach is only 100 cell level. And this was published by Dr. Jiafeng Cai, who is also my former student. But this is the beautiful and nice work he published in the PNNL. So you could see that compared to the rapid advancement of the single-cell proteomics, the phosphoproteomics still lag behind because the total abundance of the phosphorylation is so low abundant compared to the peptide, the free peptide. And there's a lot of challenge, not only the low abundance, and it's also very dynamic in the case of the protein phosphorylation. And the phosphopeptide is more negative charge compared to the free peptide. So overall, there's a lot of challenge associated with a single cell level. And so we start to think whether we can have a better approach to take care of this. And then I met my colleague, Dr. Xiaolin Tu, who is the expert for microfluidic device. And so we thought that maybe this is an approach that can be fully automated to minimize the sample loss with a lot of flexibility. So for example, this microfluidic device can be, the battery is low. Is that okay? The battery is low. Okay, and so with this micro device, you can have a very flexible design for different kind of configuration. And in the beginning, we can even do cell culture. We can do drug screening method. You can use a microscope for everything. And following the proteomics approach, we can also design different kind of material, for example, functionalized nanomaterial, titanium oxide bead, to do the enrichment for different kind of protein modification. So we thought that maybe this is a way to go for the modified protein analysis. And then we started to test the microfluidic device for the single cell proteomics. But initially, we actually designed this for patient analysis. I thought maybe this is nice for fully automatic analysis. So we started from 5,000 cell, 1,000, and finally we realized, oh, that's great for single cell. That's how it started. And you could see that even at a single cell resolution, it's very, very nice to see that we are able to do observe the EGFR signaling pathway in a very comprehensive manner. This is really, really amazing. But then when we want to test this on the single cell phosphoproteomics, and not to mention single cell glycoproteomics, we encounter a lot of challenge. Compared to the single cell proteomics, the phosphoproteomics or other PTM usually require enrichment of the modified peptide for better coverage. So if you look at the conventional workflow, there are several steps that we are not able to use the microfluidic device. For example, to do the cell lysis for trypsin digestion, sometimes we need to do the desalting, we need to change the buffer, and we are also not able to do the centrifugation by microfluidic device. So that means we have to modify the process. So first of all, we test any reagent that can be used for lysis digestion in the same tube without any desalting. So we compare the 4 types of the reagent, and we find out that SDC is actually working perfectly with the microfluidic device. And we further find out that the digestion time can be further reduced to only 1 to 2 hours. This is very complete without any missing value. And finally, we also add the DDM. This is suggested by Dr. Jiafeng Cai in his paper to minimize the non-specific absorption on the tube. So after the protocol is optimized from the micro scale, you could see that the coverage actually improved a lot. And following this process, the whole process can be completed in 2 hours, very, very fast. And we also use the library, DRA library, to improve the sensitivity of the coverage. And you could see that we actually generate different kind of library. The small library means that the size to construct the library is comparable to your sample. Because when you do the DIA library search, the peptide distribution in the retention time domain should be very similar. That can give you better identification coverage. And in the large library, even you have much higher number of the peptide precursor, but remember that they are in the different complexity in terms of the peptide composition and also the interference from other peptide. So we call spectral match, sample size comparable library. So from this, you could see that at the microgram level, you are able to actually see beautiful biology. You are able to see the depths of the biology very easily. And we just published these papers, and this is a very simple method using single tube. And the protocol is extremely easy, and you are welcome to check out the protocol for up to some microgram of the sample. So we adapt the whole protocol to the microfluidic device. I think it's run out of battery. Is that right? It's okay. I just started to talk about the single cell part. Any question for now? Or I use my own computers. Would that work?
Hey, Yuju, you asked if there were any questions. So,
okay.
So I may have missed, I think I did miss it, but so you are doing enrichment or you are not doing enrichment?
We are doing enrichment.
Okay.
Yeah.
Do you think there is a point at which you
lose more from enrichment than you gain, like at the single cell level, because you don't get perfect recovery when you enrich, right? So do you think there's kind of a sample size where enrichment no longer makes sense?
Yes, this is a great question. We actually compare the enriched sample and without enrichment. And actually, a few years ago, there's a group published a paper. They used a few hundred samples to search the single cell phospholipid. And they are able to get like hundreds of the phospholipid from hundreds of the single cell data. But based on our enriched sample, with the Eclipse, we don't have the extra yet or the timsTOF yet. So based on Eclipse data, we are able to get few hundreds of phospholipid at a single cell level. So I do believe that enrichment is much, much better. But if you talk about the nanoscale or the microscale, there's a huge difference. So enrichment is still very important. And in the case of the glycopeptide, it's the same. Say if we have 1 microgram of the sample, the serum sample, we are able to get like thousands of 1,000 of the glycopeptide. But if you do not do any enrichment, then you probably get much, much less. But this is based on the Eclipse data, Lumos data. And I think now we have much faster speed of the mass spectrometer like Ultra or the timsTOF. So that might be another level. Yeah.
No, thank you very much. And is there, since most of us don't have access to the microfluidic devices, right? Is there an alternative workflow like phage tip or something like that you could do instead for enrichment?
Okay, for the one-part protocol I just mentioned, for the phospholipid level, we are able to get like 0.2 microgram. So that's few thousands of the cell. But for the protein level, it can be go down to hundreds. But we are not able to get even more. So I think the microfluidic device has the advantage of the sample loss, minimal sample loss. Yeah.
Thank you.
Thank you. To follow up on Ryan's question, you started talking about the interplay of phosphopeptides and the signaling through glycopeptides to phosphopeptides and so on. Did you also use any enrichment tools for glycopeptides or glycans? Maybe you didn't get to that yet.
Great question. Great question. If you asked me the same question 2 years ago, I will tell you no, different material. Glycol and phospholipid are both negative, but they have the different hydrophobicity and also different degree of the negative charge. So it's kind of difficult to use the same material to do the enrichment. But recently we have work using something called Trilic, which is a hydrophilic material for conventionally used for the glycopeptide. And we use that for glycopeptide for some years, but we have difficulty to get highly purified phospholipid because of the low hydrophobicity of the phospholipid. But recently we discovered that if we add the metal ions into the Trilic material, that material alone can be used to enrich both glycol and the phospholipid in a sequential manner from the same sample. So this is one of the methods that can enrich both. Yeah.
Did I understand correctly? You used helic?
Trilic. It's not helic.
Not helic?
Are you a chemist?
Yeah.
Okay, great. For helic material, there's amine and also phosphate. Okay, and in helic material, the positive charge is inside, and negative charge is outside. So if you have a bit and positive charge inside, that means your negative peptide has to be going to inner side to do the interaction, to do the binding. So the efficiency is not good. But for Trilic material, the polarity is switched. So you have the positive charge outside to extract the glycopeptide or the phospholipid. So that has much better efficiency compared to the helic. material. It's called tritic, C-helic.
Oh, okay.
Yeah, it's ionic reagent. Yeah, so we just spike iron into the tritic, then it can be used for enrichment of the both glycol and phosphopeptide.
Thanks.
Yeah. Yeah. Or I can use my computer.
I'm wondering if you've worked with, for example, high pH fractionated libraries.
Where's the microphone?
Here.
Oh, okay.
Sorry. I'm wondering if you have tried, for example, to work with high pH fractionated libraries of bulk samples that have been phosphopeptide enriched to help like a little bit the DIA software to find more IDs.
So can you rephrase your question?
So when you have like a library,
yeah,
of a phosphoeptide enriched sample, like let's say a bulk sample, yeah, use that as reference for the search with the software in single cell samples.
Yeah.
If we try that out to increase or improve the hits.
Yes, yes, that is actually in my talk. This is actually a very important question. When we work on the single cell proteomics, the library is not an issue. You just try to, like Alex mentioned, you need to have a high quality of the spectral library, and that can enhance the single cell proteomics. But when we work on the single cell phosphoproteomics, we encounter the challenge that I don't know if I can use the name. Okay, spectronaut. Even we pass all the FDR control of the spectronaut, and you find out that some of the peptides do not have the good quality on the mass spectra. You know, usually we will check the MS fragmentation, the number of the fragment ion, whether the phosphosite can be located for site determination. And we find out that some of the low abundant peptide, phosphopeptide has very low number of the fragment ions, but they were still identified by spectronaut. That's why we also consult Alexi to use the FreePy to confirm this part. So I think for this kind of modified peptide, the computational tool still need a lot of space to further improve the confidence of the identification. Yeah. Okay. Thank you. Okay, so we adapt the protocol into the microfluidic device, and this is a very simple concept. So you do the titanium oxide first, and the reason that we use the titanium oxide because it is a bit material. It can be easily packed into the column, and then we do the cell captures. It's all fully automatic, and we also have a microscope to look at the imaging of the size or in the morphology of the cell, and you can do the cell lysis in the same chamber together with the protein digestion. So after everything is done, is ready for mass spectrometry. You don't need any desorting at all. So that, because this is a simple step, so that will minimize the sample loss for single cell phosphoproteomic data. So as you can see from here, one of the advantage of the food device is you are able to adapt any kind of accessory to the chip. So we can use a microscope to look at the real image of the single cell. You can also count the cell. And one of the revision compared to our previous version is that the cell utility efficacy is very high. That means you don't need to have tens or hundreds of cells flow into the chip. You just need to very small very small number of the cells flowing to the chip. It's nearly 100% of the capturing efficiency. So that is very nice for the limited sample. And following that, we use the DIA library-based DIA to improve the coverage. And so this slide explains why I mean the sample size comparable library. So on the left-hand side, you can see that we actually generate different size of the library using different amount of the sample. And then when you use the data set to test the different library, you could see that for the smaller cell number, the small library actually improved the coverage because of the similar peptide space in the LC-MS-MS domain. But when you have the higher cell number, the larger library is important. And sometimes people ask me, how do you know which library works better? Actually, we find out that if you use the sample amount like 2 to 5-fold higher, that usually is the best. But it still depends on the sample type. So I think this is a very important point of building the spectral library. And so it's kind of amazing that from 10 cell to 1,000 cell, I forgot to mention. So even with the 1,000 cell, you could see that we are able to identify more than 10,000 phosphopeptides. That is already sufficient for a lot of biology of your interest. And even with the 1,000 cell, we are able to see many, many kinases. And also, you can see the very comprehensive coverage of the non-small cell lung cancer pathway. So for example, for EGFR, the tyrosine site, these two tyrosine sites are the so-called autophosphorylation site. That is the indication of the EGFR mutation, the driver mutation. So at the 10 cell level, it's amazing that we are able to see that. But after 10 cell, then we start to have the challenge for the single cell phosphoproteomics. So this is the one I just mentioned. We use the spectrum now to do the library search. But even everything past the confidence level and FDR control, you can still see that a smaller group of the peptide is in the low range of the normal distribution. So that suggests that this phosphopeptide may not have the good quality, or at least the confidence is not high enough. So we're struggling this for a lot of time. And then we find out that we probably need to do a more stringent control on the confidence of the phosphopeptide identification. So we follow the, first of all, we follow the monodistributed distribution of the precursor. We make sure the precursor is correct. And we also use the higher cell number to verify if the phosphopeptide pattern is correct. So by using this, then we reduce some number, but we want to make sure all the identified phosphopeptides are highly confident. So I think this is still issues that we really need the help from the informatics expert. And thanks to Alexi and Fang Qiao, the great student and now postdoc there. And we also use the FlatPy to confirm the identification of the single cell level. So you are able to see that we have a few hundreds, not high yet, but few hundreds of the phosphopeptide at the single cell level. It's kind of amazing that even at such a smaller, very low coverage, we still see a lot of phosphopeptide that have been reported in the literature related to cancer biology or the drug resistance, etc. So I think a lot of people said that the important molecule must be low abundant. But in the case of the single phosphoproteomics, we may not necessarily agree with this kind of argument. We see a lot of phosphopeptide, important phosphopeptide are still there. That means they are in the basal level. But with such a low abundance, we really want to see whether that is likely useful. So we tested on a very interesting and important question in Asia. EGFR is high percentage in Asia countries. So our government have a lot of money paid to the reimbursement of the target therapy, all free for our patient. But eventually, nearly every patient will develop resistance to the EGFR target therapy. And then they can only perform chemotherapy. So the question is, for this dying patient in the late stage, is it possible that we are able to suggest any potential drug as a next line treatment for this dying patient? So we thought that most of the target therapy are kinase. So phosphoproteomics should be a good approach. So we tested this analysis for 3 patients from the stage 4, and they already developed resistance to the 3rd generation EGFR target therapy. So with 10 cells, we are able to see about 1,500 phosphopeptide. And it's very amazing that with such a low coverage, we actually observe the biomarkers for the 2 most abundant lung cancer cell type, adenocarcinoma and SCCC. And these 3 patients are actually from the same clinical staging and same EGFR mutation. No difference at all in the clinical diagnosis. But we observed that this specific patient, 157, has the biomarker for HCC, but clinically, this person was diagnosed as the ADC cell type. So we went back to the hospital and consulted my collaborators whether there's a wrong annotation or something is wrong there. And then they discovered that this patient actually has a very low amount of the SCCC composition. So I think this example demonstrates that this kind of cell heterogeneity was not able to be detected by the clinical tools. So this also highlights the importance of the small cell population analysis at the patient level. And for these 3 patients, we actually see a lot of difference between their phosphoproteomic profile. So for example, this female patient has the activation of the catenin complex, especially the YAP1, which has been recognized as an important regulator for the drug resistance. So finally, we want to see whether this kind of analysis is useful. And this is not single cell level yet. This is the 10 cell level. We are able to see that 2 phosphorylation sites on the EGFR are activated. And we also see the phosphorylation site for the ERBB2, the HER2 therapy. So we talked to my colleague whether he is able to test the drug for the patient. So he used the primary cell from the same patients from the lung pleural infection, and he confirmed that the drug obtainable targeting both the EGFR and the ERBB2 is useful for this specific patient. So we are very happy to see that this kind of research assay is likely be useful for the patient to indicate the potential next line drug treatment for the future. So I think this, again, highlight the potential utilities for this research. But unfortunately, we don't have the most high-end mass spectrometer, so the throughput is really an issue for our current research. And we hope that we have the new mass spectrometer coming later this year to really enhance the throughput. And finally, I think we are very happy to make our dream come true for the single cell phosphoproteomic study. And even though we achieved the first goal, but I think there's still a lot of challenge associated with this kind of single cell protein modification. And the data deconvolution from DIA, I think, is still an issue. And the site localization is still very, very challenging, given the very low number of the fragmentation pattern for a lot of phospholipid. But with this approach, I think we are able to ask more interesting questions. For example, I think we start to have the tool to ask how does the site-specific regulation at the individual cell level. And we may be also to start to ask the single cell signaling issue, whether the site-specific phosphorylation occurs on the same cell or different cell. Because what we observe from the past is the accumulation of the overall result. So I think this likely open another field. And I also talked to my student. When I tell him to do the project, he was angry for 1 year, and he always argued with me because this is something impossible. But now I think maybe it's time that we can pursue like single cell glycoproteomics, but we still need to demonstrate the utility of this kind of research. And finally, I want to thank my colleague. Dr. Tu is the expert to make the microfluid device. So if you are interested, you can talk to him. And Taiwan is the kind of empire of the semiconductor industry. So chip production is kind of easy and very, very cheap for our countries. And we are hoping to really push the tools for the practical utility in the future. And finally, I also want to thank the patient. And clinically, there's a lot of questions that might be useful for proteomics. So I'm hoping that we are able to really push the utility for the patient in the future. Thank you very much for your comment.
Thanks. Thank you, Dr. Chen. So I think that we have time to take one question. So I'm here.
Yeah, so I'm a little bit interested in the microfluidic system for single cell sample preparation. I mean, I'm an old microfluidics engineer, actually. I did that early in my PhD. So how much of an issue is like bubble formation? Because I guess that is usually a problem, that bubbles get in and they get stuck and they can.
How to get it started?
Yeah, how do you deal with bubbles?
I don't have the slide to show. That is actually the most challenging question in the beginning. So my colleague, Dr. Tu, used a high pressure to push out the bubble, and also the surface, the design of the size of the channel is also highly important. You have to make sure the fluid mechanics, is in the most optimal condition. So you can check out our design. And I think he has all the detail in the publication. Another thing is the surface chemistry. You have to make sure that after you do everything in the clean room, everything on the surface has to be very clean. Otherwise, you introduce a lot of detergent reagents into the mass spectrometry, so the data will not be good. Yeah, so I think these two are most critical.
Can I ask just one small question? Okay. I mean, of course, when you prepare on a tip, this is something you can dispose of afterwards. Are these microfluidic systems, are they reusable? Like, how do you clean them efficiently so that you can load a new set of single cells and make sure that you're not contaminating?
Yes, we tested the chip for, I think at the current stage, the base utility is like 5 times. But because it's so cheap, so we don't usually reuse the chip.
Okay.
Yeah.
Oh, thank you.
Thank you.
Transcription factor cheat codes, that one. Right. Let's see here. There we go. Okay. Amazing. Yes, great. Well, nope. Okay, there we go. There we go. All right. Yeah.
Hi all, thanks for your attention and participation so far. This will be the last talk of the day. I'm pleased to introduce Dr. Katie Galloway. Dr. Galloway is a professor of chemical engineering at MIT, where her research focuses on connecting basic research questions in gene circuits, genome architecture, and cell fates to the development of synthetic biology tools. Please welcome Dr. Galloway.
So it's a real pleasure to be here with you all. I'm going to definitely be switching gears a little bit. This will not have a lot of proteomics or mass spec, but I hope I'll give you some motivation to think about why transcription factor levels really matter, what we've been seeing. My lab works at the intersection of synthetic biology and stem cell biology to program cell fate. And so oftentimes we're thinking about how do these cells jump across these transitions, like what's required. These cell fate transitions have been extensively studied through RNA sequencing technologies. And that gives you one picture of how programs turn on. But a lot of what we're seeing, especially now, is that the regulation at the protein level is extremely important. And I'll give you a few vignettes, and I'll try to keep it short so we're not here too long. So just to give you a little bit of idea of what these transitions look like, you're probably very familiar with making iPSCs from fibroblasts. If you think about a cell fate transition, this is very canonical. You can use the Yamanaka transcription factors, overexpress them in diverse ways, and then that makes iPSCs at some frequency. You isolate those cells, you can then take small molecules and growth factors and differentiate those cells with pretty good efficiencies, and these protocols vary, but you can make neurons very nicely. You can do something alternatively where you overexpress the transcription factors that make neurons. And that's actually really efficient. We love doing this. It works really well. But the levels still actually matter quite a lot. And then we can do the thing that we like to do the most, which is actually go direct or do direct conversion, where we take somatic cells, so fibroblasts, and convert them directly using transcription factor overexpression to make these neurons. And this saves time, but it's a huge challenge. And when I started this working in this system when I was a postdoc, I remember being like looking in the dish and being like, wow, this is not looking good because we have like 50 cells. And here was actually one of those images. You can actually see this is a transgenic mouse model where the spinal cord is glowing green because it's an HB9 GFP mouse. And so all the motor neurons are labeled with green. This is useful in a dish because you can start to see the cells. Of course, this is a sad picture because you can see there's 2 cells that have turned on this reporter. And of course, there's actually a sea of cells there. So the question is, why have some of these cells reprogrammed, some haven't? And why does one of them get stuck? One's a neuron, has beautiful processes, soma, beautiful. Other one, obviously not a neuron. And so the question is like, why did one of these cells respond and the other one didn't? And so many of them completely were non-responsive. So some of the things that we've learned about this process only came through a lot of trial and error, which is, at least one path forward. Importantly, we've been able to get a lot more insight as we've increased the rates of conversion. The hard thing is if you want to improve a process, or understand a process, most of the time you have to do both at the same time because you don't have enough events. You can't actually tell with statistical certainty if any perturbation is actually improving that process or impeding it, or if it's just noise. So we did that first hard thing and basically figured out how to increase the reprogramming rates through this chemogenetic cocktail. And this was quite useful. And we've actually subsequently improved it even more and improved the reproducibility, which is important. So with that, we then wanted to actually be able to ask, can these cells be useful therapeutically? And we were able to get to a scale where we could ask, okay, if these are going to be cell therapies, can we implant them? Because again, scale really matters. We can now do this for mouse motor neurons. We can do this, and we can do this for human motor neurons. And this is directly converted. So people have been able to make iPSCs or ESCs into neurons for a long time. But going direct from skin at scale is really hard. And now we've been able to actually engraft these into the brains of mice and see that they actually survive and can start to send out projections, which is really exciting because, again, when we started this, like looking at 50 cells in a dish, and I'm like, what do you do with 50 cells in a dish? Because even after single cell isolation, you run that on a 10x and they're gone. It was not good. So one of the things that we realized when we actually got to enough scale, we could start to ask questions about how do our transcription factor levels themselves actually influence the rates of conversion. And so one of my first grad students, Nat Wong, came in and said, I really want to know how these individual transcription factors, like them alone, are going to contribute to rates, like higher level, more conversion, lower level, worse conversion, and no one really knew. And even here, we're kind of getting close to this. We've done some staining where we're actually looking at the transcription factors. But here, what we have is an NGN2, which is essential for conversion, a T2A tag in Ruby. And so that we can actually just look at what's the level of expression. But this is a 2A, so we're not looking at directly at the levels. But this gives us a way of not interfering with the protein. And so what Nat did is he went in and made a bunch of different constructs that had different levels of NGN2, and then actually exploited the variability of reprogramming to build this axis, where what he could see is that you see this axis here of basically Ruby, which gives us a proxy for NGN2, and you see there's this positive correlation with conversion and NGN2 levels. Great. And we've been able to see this is generally true with NGN2. Now, the other transcription factors, there's 2 others that are actually essential, and you can see, let's see here. If this works. I don't want to touch anything and break anything. Without NGN2, you get no reprogramming rate. But you'll require 2 other factors to make this happen. And when we go and tag those, what we can see is that this positive correlation isn't for everything. Like if you overexpress anything, it doesn't positively correlate, which is good because then we have more confidence. So there's no correlation with ISL1 and LHX3. But you can see if you drop these out, the rates drop precipitously. So they're necessary, but their levels aren't correlative with our reprogramming rates, which was something that we were surprised to see and thought was pretty interesting. Okay, and I guess the one other thing hiding here is this is specific for a very specific cell state. So these are hyperproliferative cells, so cells that have rapidly divided. We can measure the levels there. And if we measure in cells that have not rapidly divided, the trends are more obscure because the cells that are going to reprogram are the ones that have rapidly divided. So we have this dynamic timeline where you have to isolate the cells that are actually going to convert, and the measurements of those cells and their transcription factor levels are predictive. and other cells are not. So we have a couple of different challenges in this system. And so the levels really matter. What we again found was that these cell state, and by cell state, I'm going to be talking about proliferation history. So the history of cells having divided. It's very hard to measure absolute level of proliferation at any given time. So we measure how much cells have divided over time with a die. And so what Nat had basically found was that the cells that have gone through this hyperproliferation, so rapidly divided, are more receptive than to transcription factors and convert at high rates. But we still didn't know entirely whether you actually wanted really high levels of transcription factors and or high levels of transcription factors and proliferation, or how basically the cells are going to interpret those. And so what Nat did then, wait a second here. All right, was he went in and wanted to actually measure the levels and the proliferative cells and ask the question, how does proliferation change the levels of expression? And the expectation is, given the hyperproliferative cells had previously been shown to convert at higher rates, the expectation is the transcription factor level should be higher. But you guys were all here today and you got to hear Andrew's talk. So your all expectation and what you know is going to be true is actually the levels are going to be lower. But we had to go show that and actually prove it. So Nat actually labeled these cells hyperproliferative, non-hyperproliferative with this dye, and then sorted the cells out and watched, basically looked at the levels with these different tagged proteins. And again, this is a proxy, but we've gone in and actually measured the proteins as well for some of the ones that have good antibodies. Again, what we saw here is that the levels in the hyperproliferative cells relative to the non-hyperproliferative for every single one of these genes is lower, suggesting that it's not the transcription factor levels that make these cells more able to convert. It's actually the cell state that is then telling the cells how to interpret the transcription factor cues, which is important. And so we're like, well, if that's true, then we can do the next hard experiment, which is actually go in and measure and isolate cells with different levels of NGN2, because again, that's the one that has the correlations. So we take cells with low, medium, and high, and we look at the cells that have either a history of low proliferation or high proliferation. And what you can see immediately is the ones with high proliferation convert at higher rates. But what's really interesting is that even the cells with really high levels of NGN2, if they haven't rapidly proliferating, are converting at lower rates than cells that have lower levels of transcription factors. Again, indicating its proliferation history and the transcription factor levels that potentiate the ability of the cells to convert to a new cell type. And that's sort of summarized here, where again, the transfer function for NGN2 and the cell receptivity is dictated then by this proliferation history. Okay. And again, that was really wild because we kept on asking the question, how can the cells reprogram if they have lower levels of transcription factors? And we're like, we still don't know the answer to that, but they do. And probably it's because over time, the levels come back up. And yes. So this is sort of the picture that we have here, where these hyperproliferative cells are then able to be more receptive. The levels of NGN2 basically are lower in these hyperproliferative cells, but then as they increase, basically you're getting higher rates of conversion over time. Okay. So the last piece, and I was glad I got to talk after this, the talk on phosphoproteomics, is we started asking the question, how is proliferation at all connected to what's going on with our transcription factor levels? Or is there any connection, or is this just all dilution? Could there be any active regulation? And so a while ago, a group had published that there was some phosphoregulation of NGN2, which is relevant because NGN2 should come and bind different targets of neuronal genes and then lengthen cell cycle. Eventually, these cells are going to be totally post-mitotic, right? So they're going to slow down their cell cycle. What happens is that it previously had shown that there's phosphosites on NGN2, and that MAPK signaling can actually lead to the phosphorylation, which either inhibits DNA binding, some people have shown that, or leads to degradation. And if that happens, then you basically have no binding of the neuronal genes and then cell cycle progression. And so we wondered, could this actually be happening in this system? People have shown it in other ones, but in primary cells, a lot of things don't always translate. And so Brittany went in and did sort of this Western and did, this is the wild type here, NGN2. She also took a phosphomutant, so it has alanine, so it can't be phosphorylated. And normally, what you can see is the patterns change significantly. So this is, in theory, the non-phosphorylated NGN2. When we add in MAPK signaling, which is what we use to drive cells to rapidly proliferate, what you can see is this band disappears almost entirely. Seemingly, it's being degraded. Again, the mutant basically doesn't have that. And then if you turn the MAPK off with an inhibitor, you see that this pattern basically gets reestablished to what you had before. suggesting that this is actually what's potentially happening in this model over here. So we went in and actually overexpressed the mutant and the wild type that has the phosphosites and looked at the proliferation rates. And indeed, when we have high MAPK signaling, as you see here, there's a lot of cells that are proliferative, and basically all the other conditions where you have these different isoforms, you see that basically there's no real substantial proliferation. What's interesting is that when we go look at the conversion, though, you can see that the conversion is, as we expected, quite high, but actually this mutant does pretty well. It impedes proliferation, but for some reason it's actually quite potent, depending on the day, in terms of inducing conversion. And in iPSCs, it's known that this mutant is actually more potent than the wild type, probably because it's able to resist all the different mechanisms of MAPK signaling to impede its ability to find its targets or be degraded. And so what we'd love to be able to do is actually build a circuit where we could turn this on in the beginning, drive proliferation, and then ultimately turn on the second mutant, which would allow us to then evade any sort of cell cycle processes. The last thing I'm going to tell you is what we see is that transcription factor levels don't just affect these cell properties like the gene regulatory networks. You can actually see this at macro scale. This is just a normal MEF in the nucleus here. You can see lamin B and H3K27 trimethyl. Here, when we add in these transcription factors, that's L and I, you can see the nucleus gets much larger. Interestingly, when we drive cells with proliferation with this cocktail called RID, the nuclei look smaller or relatively smaller compared to transcription factors. And again, that might be because NGN2 is being probably degraded in this state, which is, again, quite interesting. And this also seems to correlate with the state of lower transcriptional activity and histone modifications. Okay, so in summary, the thing I wanted to tell you about is, depending on where cells are in their history, they can have very different properties and receptivities to transcription factors. You need to measure the levels, but the levels alone aren't enough to tell you whether a cell is going to convert. And so we have a really complex problem. Transcription factor levels matter. The history of the cell matters, and we have heterogeneous populations of cells that are going to asynchronously basically transition. And so single cell proteomics seems like a really useful opportunity for us to start to dissect where are cells in this process and when are they advancing or not in conversion. And hopefully we can start to then figure out how to build circuits that can actually can do this type of reprogramming in vivo, which is our goal. So that's one side of my lab that does our reprogramming. The other side then figures out how do we actually build these circuits so that we can run them reliably in cells. And of course, we've always thought that we were controlling protein levels really well. I think I've showed you that we're doing it okay sometimes depending on the cell. And there's a lot we could learn about how these proteins are regulated to then build circuits that work better. My lab, again, is split between these two sides if you want to know more. I know Nikolai actually initially asked me to talk about a different synthetic biology project, but I thought this might be more interesting to you all. I hope it is. If not, you get your money back from me. And this is my group. We started in the fall of 2019. It's been really a pleasure to build this lab over the last little while, and especially grateful to Nat Wong and Brittany Lundy, who contributed to this, and also Adam Bites on the reprogramming team. We have wonderful collaborators as well, our funding sources, and happy to take questions if we have time.
I think we have time for a few questions.
So, Katie, listening to your talk, all the time I was thinking how useful a metabolic pulse could be to tell you about the proliferative history of the cells and the different rates of protein synthesis and degradation. But let's say we could make all of the measurements you could possibly dream of.
Yeah.
How would you use those to optimize your differentiation protocol? There are sort of two different challenges here. One is measure things at single cell resolution, time resolved manner, which is very, very challenging. There is a completely separate challenge. Once you have these data, how do you use them to optimize the differentiation protocol?
So that's a very good question, because I think it's being one. There's the question of how is this actually working, and then how could you actually use tools to guide cells at all. Can you? I actually think that there's probably things that we could do, some of which are, I mean, we've already seen just, happily somebody else had studied this NGN2 mutant. And I think one of the things that we would see is like, well, if you know that there's a phosphorylation state that tends to then emerge upon different perturbations, could you imagine designing something that's resistant like this mutant? I think there's probably a lot more things in there like that. One of the things when our cells actually, when we add in transcription factors, the nuclei get larger. The cells are generally larger too. And then when we add in this cocktail, basically that drives proliferation, the cells become smaller. They're less metabolically active. We don't know what the protein levels look like, but we know that the RNA levels are lower, and we know that the rate of transcription is much lower. They almost look like they're in a quiescent-like state. And again, we haven't measured protein synthesis, any of these things yet, but I think the question is, in terms of this driving, how do you build a circuit that's actually resistant to some of the things that we see that are going wrong here? And that's what I think we'd be building towards. Yeah.
Thank you. Questions? I know that we actually might have gotten one in the chat.
Yeah, I don't know. And stop share, yeah.
Okay. Okay. We can ask this question first and then go to Andrew. So there's a recent Nature paper screening yeast TFs for downstream effects. I was wondering if the cases in mouse and human is much different due to the different TF to genes ratio and networks. Could you please comment on this?
So the networks themselves would definitely be different. I'm not sure if that's exactly what the question is being asked. But yeah, so I don't know. Maybe you're asking a question also about the connectivity or the degree of connectivity. I mean, the difference between yeast and mouse and human regulation at the transcriptional level is pretty large. So I would imagine there's going to be an enormous difference between them, but I can't comment too much more beyond without more information. Yeah.
Hi, great talk. So I have two questions. One quick one first. So do you see any differences either in like the gene expression or phenotypes of the direct converted neurons versus ones that you go back and so like yeah, is there any...
That's a great question. So we haven't looked at that ourselves directly. Other people have reported on this, asking the question like, well, if direct conversion is so hard, why even bother? And the answer to that came through a couple different papers. One, Fred Gage's lab at oh gosh, now I'm forgetting where he is. Anyway, he's in San Diego. They showed that if you could directly convert neurons, you'd actually retain signatures of aging, whereas when you went through an iPSC intermediate, you eliminated the signatures of aging that they had identified, which were specifically a nuclear cytoplasmic deficit. The reporters that they work with are actually fairly difficult, and I've looked at the correlations; they're not strong. But other people have come up with different signatures of aging and seem to see the same thing. I will say it's hotly contested, and it's not a fight that I'm interested in getting involved in. However, I'll say this: the advantage is we haven't measured this in our system at all. I figure if we retain signatures of aging, it's great for disease modeling, and if we erase them, hey, we did rejuvenation, great. So you have to find the silver lining no matter what.
And then, sorry, this is partly just from my understanding. So that transcription factor that's getting phosphorylated and then degraded, so when that's bound to essentially somewhere in the DNA that's preventing the transcription factors that you want to express, essentially from being able to activate the programs that are needed to.
So NGN2, when it's bound to different targets, normally, especially, in a forward programming, it's binding to the promoters of or enhancers of neuronal genes, so activating neuronal programs. It may also function as a repressor. I don't think that's its major function. It's mostly thought to be a transcriptional activator. And it also does bind with different partners. Is that answering your question?
Yeah. So do you mostly see like the lack of conversion as just making it harder essentially for the transcription factors you need to like get to the sites they want to? Is that the main challenge, or?
So I don't think it's so. I think our one of our experiments where we just blasted the expression and they don't allow them to proliferate actually, or they don't proliferate that much, actually answers that question. High levels are not sufficient to drive conversion at high rates. So you have greater probability of converting if you've rapidly divided and gone through that hyperproliferation phase than if you just have high levels. And so seemingly the thought is that potentially, actually I was really interested in the idea of entropy because it could be that especially as these RNA levels, protein levels, everything dilutes down, you create a scenario where your transcription factors are then a larger fraction basically of the proteome space as you're cranking out these with these transgenes. And that creates a space where basically you can displace and events become more probabilistic. We haven't modeled that. It's an idea. I don't know if it actually is true.
Thank you.
All right, thank you.
Hey, so with this we've approached the end of our program for today. I enjoyed tremendously the diverse presentations, questions, and discussions. And I'm very much excited and looking forward to another day tomorrow of exciting presentations and discussions. And there will be a lot of questions that were asked today that I think are going to find their natural expressions in more discussions tomorrow. So without further ado, I'm going to let you go and rest, enjoy dinner, and look forward to seeing you all tomorrow for another wonderful day.
All the lights.